dignitaries who have joined with us for day 3 of SNJB PharmaCon 2021. We are really happy to have Dr. Srinath, Dr. Uduppa, and the chairman of the first session with us. I, Dr. Atish Mundada, coordinator of SNJB PharmaCon 2021, on behalf of management of SNJB and all my colleagues of SNJB Sriman Suresh Dada Jain College of Pharmacy, welcome you all and welcome all the delegates to the day 3 of SNJB PharmaCon 2021. Dear delegates, we have seen a very knowledgeable sessions on day 1 and day 2. Session 1 was delivered by Dr. Vandana Patravali. Session 2 was delivered by Dr. Jitendra Wagh. Yesterday, we had a knowledge feast being delivered by Dr. Vaibhav Patel and Dr. Ravindra Dhumar. Today, continuing our tradition, the way we have renowned speakers on day 1 and day 2, similarly, today also, this is going to be a real knowledge feast, Sunday feast we can call it as, which will be delivered by Dr. Srinath from US, and the second technical session will be delivered by Dr. Saranjit Singh sir, a brand ambassador of SNJB PharmaCon. So we are really happy to welcome you all on day 3 of SNJB PharmaCon 2021. The first technical session is going to be delivered by Dr. Srinath, and for do the honor, I call upon Dr. Ganesh Basarkar to carry the session forward. Over to you, Dr. Basarkar.
Okay, sir. Okay. Thanks, Dr. Atish Mundada. First of all, good morning, all of you. A warm welcome on behalf of Sriman Suresh Dada Jain College of Pharmacy for the third day of SNJB's International Conference, that is PharmaCon 21. Indeed, as Dr. Atish Mundada said, that today is a feast for a technical knowledge. I can say that all we are very much fortunate to have the two biggest academic stalwarts today as speakers, Dr. Srinath Palakurthi and Dr. Saranjit Singh, to whom I need not explain, from whom we can gather as much as technical knowledge today. So as Dr. Atish said, that today we are going to have two scientific presentations. The first one will be from Dr. Srinath Palakurthi. So for this, I am very much delighted to introduce the chairperson for this session, Dr. B. Vijay Kumar. A warm welcome to Dr. Vijay Kumar on behalf of Sriman Suresh Dada Jain College of Pharmacy.
Thank you and good morning.
Let me give a little bit introduction about Dr. Vijay Kumar. Dr. Vijay Kumar earned his doctoral research in pharmaceutics from Department of Pharmaceutical Sciences, Kakatiya University, and presently he is working as a professor as well as the principal at Janagama Institute of Pharmaceutical Research at Warangal, Telangana State. Indeed, he is more of a technocrat rather than academician. He is very much instrumental in setting up two pharmacy colleges at Warangal of Telangana State. He has a vast experience of more than 30 years, which includes both academic as well as industries. And he has co-authored a number of publications and presented his research findings in various international as well as the national conferences. And his research interest includes novel drug delivery systems, dosage form design, and especially oral dosage forms. And he is a recipient of numerous awards, which includes the Best Teacher and Award of Excellence from Indo Global Education Summit Expo 2017 and Mahatma Jyoti Rao Phule Best Teacher Award. With this short introduction, I welcome Dr. Vijay Kumar to chair this session. Over to Dr. Vijay Kumar. Please take this session forward. Dr. Vijay Kumar.
Yeah. First of all, good morning, everyone. And thanks to Basarkar for a brief introduction about me. And in fact, I am very much thankful to Dr. C.D. Upasani, convener, SNJB PharmaCon, and coordinator Dr. Atish Mudunda, and the management of SSDJ College of Pharmacy. It is my pleasure to introduce today's speaker, Dr. Srinath Palakurthi. He has studied in UCPSC Kakatiya University. He might be our junior, 2 years to us, I think. He has completed successfully the B Pharmacy course from Kakatiya University in 1986 to 91. And then he moved to M Pharmacy to Dr. H.S. Gaur University, Sagar. And then he has completed his PhD in the years 1995 to 2000 in the prestigious Indian Institute of Chemical Technology, Hyderabad. Later on, he has completed his postdoctoral research in University of Alberta, Edmonton, Canada. And then from 2003 to 7, he worked as assistant professor of pharma sciences in University of Alberta. Then from 2006 to till date, he now presently is the professor in Texas, Irma Lerma College of Pharmacy, Texas A&M Health Sciences Center. So Dr. Palakurthi research area, mainly they will focus on drug delivery and targeting in ovarian and breast cancer, nanoparticles for inflammatory bowel diseases, topical ophthalmic drug products. And one more thing, because of time constraint, I am shortening his biodata. Milk exosomes. He is presently doing his research in milk exosomes. Milk has been exploring milk-bred vesicles for cancer drug delivery, as bovine milk is suitable natural source for developing a vehicle in pharmaceuticals. These vesicles are very efficient in transporting the drugs that are difficult to deliver, poorly soluble drugs. Even the milk-based exosomes themselves, that means without the drug, have shown inherent anti-cancer activity when used alone. So we know that after chemotherapy, cancer chemotherapy, there will be a lot of side effects, neuropathic pain, etc. And they are doing their research mainly on this. That means developing a combination of standard anti-cancer agent with FDA approved pharmaceutical excipient to enhance safety and efficacy of the therapy and reverse the drug resistance. So with this brief introduction, I am handing over the session to Dr. Palakurthi.
Thank you so much. Can you hear me?
Yeah.
Okay. Thank you so much for your introduction. At the outset, I would like to thank Dr. Mandada, Dr. Basarkar, Dr. Vijay Kumar, and all the conference organizers and the participants. So do I have the administrative rights right now to share my screen? Can you see the slides?
Yeah.
So, good morning, everyone. Thank you very much for the invitation to participate in this conference. And I'm really thrilled to see the video about your college and the facilities there. I'm very glad to see that, you have such great facilities and you are regularly organizing this kind of conference. For me, it looks like I'm coming back to my home, I did my, as Dr. Vijay Kumar mentioned, I did my Bachelor's of Pharmacy in Kakti University and, Master's in Sagar University and then PhD from IICT. So, looking at these facilities, gives me a feeling that I'm coming back home. So, good morning again, and I would like to start my presentation now. Our research lab has been working on a variety of nanoparticle systems. And in fact, Dr. Vijay Kumar has mentioned a few of them. But for today, I will be presenting some data on targeted chemotherapy of ovarian cancer using nanoparticles. Okay. I would like to give an outline of my presentation, which not only includes the topic of today's presentation, that is the targeted combination chemotherapy of ovarian cancer using cisplatin and cytodomycin. In addition to this project, we are also developing Z nanoparticles. Z is a protein that is extracted from corn. So this is an edible protein. And we are developing nanoparticles using this Z. As you know, unlike the polymeric nanoparticles, protein nanoparticles are a little bit tricky. And since this particular protein has mucoadhesive property, and this is also an edible protein, which has a generally regarded as safe status by FDA, we have chosen this particular protein to deliver certain immunosuppressive agents such as cyclosporine and Janus kinase inhibitors for the targeted therapy of inflammatory bowel disease. In addition to that, we're also doing a nano emulsion formulation of benzenidazole for the treatment of Chagas disease. Chagas disease is a tropical disease, induced by a parasite, Trypanosoma cruzi, mostly in the tropical countries such as South America, Mexico, and other tropical countries. So we have developed a new formulation, nano emulsion formula of benzenidazole, and currently in vivo studies are in progress. In addition to that, we are also working on cytodomycin for proliferative visceral retinopathy, generally for the in case of ocular injuries, followed by these injuries, scar formation is a serious problem, which leads to sometimes blindness. So to prevent that and to treat
Palakurti sir, sorry to interrupt you. Your voice is muted, I think.
Okay, thank you.
Yeah.
So this is the outline of my presentation. In addition to the topic of today's presentation, the targeted combination chemotherapy of ovarian cancer using cisplatin and cytodomycin, we are developing Z nanoparticle. Z is an edible protein that is extracted from cod, which has the GRAS status, generally regarded as safe status by the FDA. So we have developed the Z nanoparticles loaded with immunosuppressive agents such as the cyclosporin A and Janus kinase inhibitor for the targeted therapy of inflammatory bowel disease. In addition to that, we're also developing nanoemulsion of benzenidazole. Benzenidazole is a drug that is used for the treatment of Chagas disease, which is caused by a parasite called Trypanosoma cruzi. And this disease is quite prevalent in tropical diseases such as South America, Mexico, etc., and other tropical countries. We're also developing cytodomycin formulations for the treatment of proliferative vitreoretinopathy. Proliferative vitreoretinopathy is a condition where after the generally the ocular injuries, whether it is blast injuries or burn injuries, etc., development of a scar on the eye in the retina, and that leads to sometimes total blindness. So we are trying to develop a formulation of cytodomycin both for the topical purpose as well as the intraocular delivery of cytodomycin. So these are some of the projects that we have been working on using different types of nanoparticle systems. Coming to today's presentation on the combination chemotherapy using cisplatin and cytodomycin combination for ovarian cancer. I will be discussing how we have developed the dendrimer cisplatin complexes, dendrimer cytodomycin complexes, and it's the combination efficacy in SKOV3 xenograft model, and the basis of cisplatin cytodomycin synergism, and hyaluronic acid conjugated complexes, dendrimer complexes of cisplatin and cytodomycin, and testing their efficacy in organoid model, xenograft model, and patient-derived xenograft model. And finally, I will conclude with some toxicity data of this combination therapy. So in addition to these academic research projects, we are also working on industrial projects. Currently, I serve as a consultant for several pharmaceutical industry, both in USA as well as elsewhere. We have developed in vitro drug release testing methods, IVRT, as most of you will be knowing it, for the topical products that includes the emulsions, suspensions, and gels, both for the application onto the skin as well as for the ocular purpose. In addition to that, we're also working on in vitro release testing methods for the long-acting injectables. I cannot give much details about these projects since these are the industrial projects. Okay, so these are some of the research projects that are going on in my lab. But for today's presentation, I will be discussing the data on the cisplatin combination chemotherapy. Okay, so ovarian cancer. Ovarian cancer, as based on the statistics given by the American Cancer Society, about 21,000 women will be diagnosed with ovarian cancer this year, and out of that, about 14,000, these patients will be dying with ovarian cancer in 2021. And we know that there are multiple therapeutic modalities that are existing for the ovarian cancer, right from the surgical therapy, chemotherapy, radiation therapy, etc., and combination chemotherapy, etc. So despite these multimodal approaches, the 5-year survival rate of these late stage ovarian cancer patients is only 11%. Okay. So to give a perspective of the prevalence of ovarian cancer in India, I looked into some of the data that is published in Asian Pacific cancer journals. It says that about 59,000 around 59,000 ovarian cancer incidences are estimated in 2020, and by 2035 these number of patients with ovarian cancer are expected to be about 371,000, with a death rate of about 254,000. That is about 65% of the patients that are diagnosed with ovarian cancer will be dying of this cancer by 2035. So this is a huge challenge. And so this is a serious gynecological cancer in women, right? So this is a serious problem. And as you can see, there is a significant difference between the cancer incidence as well as the cure of ovarian cancer between the American data versus the data from India. Perhaps because there are significant differences in the therapeutic approaches and the diagnostic margins. Okay. So leaving that aside, irrespective of the treatment modalities, the multiple therapeutic approaches, ovarian cancer is a serious problem. Okay, so the first line therapy of ovarian cancer is cisplatin. Okay, and this has been the first line of therapy for the past several decades, and it's not generally ovarian cancer is a chemosensitive malignancy. So generally, it can be the moment the chemotherapy is started, the looks like the tumor would be regressing pretty soon. But the problem is not that. The problem is, since the ovarian cancer does not present any significant symptoms in the patient, most of the times, by the time the patient reaches the physician, sees the physician, the tumor might have already metastasized to other areas, mostly into the peritoneal cavity. So once this tumor has metastasized, it is very difficult to control the spreading of this tumor, and moreover, the development of the resistance. So as you know that the mechanism of action, cisplatin is a DNA adduct formation. Basically, it would be forming an adduct with the DNA, and therefore it would be preventing the proliferation of the cell. So, but the efficacy of cisplatin would be reduced upon multiple cycles because of the development of resistance primarily, and also the dose-dependent toxicity. Mostly it is the neurotoxicity, nephrotoxicity, and autotoxicity that is induced by cisplatin because the higher and higher doses have to be given because of the development of resistance, right? So most of the times, once the dose is increased, and toxicity will be seen, and that results in the discontinuation of the therapy. Okay. So why this resistance is developed? Resistance to cisplatin is developed because of several reasons. One of the primary reasons is inadequate uptake of cisplatin into the cells. Cisplatin is a small molecule. It will be diffusing generally into the cells by simple diffusion process, based on Fitzlock diffusion. But what happens is, there are certain transporters that are involved in the uptake of cisplatin. In addition to diffusion, copper transporters, CTR1, CTR2 are the transporters that are involved in the uptake of cisplatin into the cells. And these transporter proteins will be mutated, and therefore the extent of uptake of the cisplatin into the cells is hindered. Two, the tumor cells will be developing nuclear excision repair mechanisms. Therefore, even if the DNA is broken down, still the repair mechanisms will be overcoming the cisplatin efficacy. So these are the two main mechanisms by which the cisplatin resistance would be developed in the cancer cells. So to overcome this cisplatin resistance, generally a combination chemotherapy is used, right? So more than one chemotherapeutic agents are combined. Therefore, their additive effect is seen as, would be improving the efficacy as such, right? However, when we are going for the additive therapy, additive combination therapy, the efficacy might be increased, but at the same time, the additive effect on toxicity would also be enhanced. Therefore, that is a serious concern, right? So ideally, to develop an efficient therapy for late stage ovarian cancer, we want the therapy should be targeted, and it should target the various populations, the tumor cell populations in the tumor. As you know that the tumors are generally heterogeneous in nature, which contain the sensitive tumor population, resistant tumor population, and the cancer stem cells, right? As we go along, I will show you some of the slides showing how these, the cancer stems will differentiate into tumor cells later, but tumor is a heterogeneous tissue. So we need to target different tumor cell populations, right? And we should increase the cellular uptake of cisplatin or the combination drugs into the cells. We need to reduce the toxicity of these combination drugs. So that would constitute an ideal therapeutic margin, right? So we will address these challenges one by one to develop an ideal therapy for ovarian cancer. So from the drug delivery perspective, I was mentioning to you earlier that one of the primary mechanisms of development of resistance is inadequate cellular uptake of cisplatin into the cells. So to address this issue, we have asked a question that can we develop a nanoparticle formulation to increase the cellular uptake of cisplatin into the cells, right? So to address, before addressing that issue, in addition to developing the formulations, most of the pharmacy graduates there might be aware of the concept of tumor targeting. Generally, we use nanoparticles to increase the cellular uptake. And when we use nanoparticles, that would elicit certain targeting effects, right? So in that context, I would like to introduce the concept of enhanced permeability and retention effect that is possible with the nanoparticles, right? So this is a schematic showing how the nanoparticles will be accumulated into the tumor tissue. So this is a tumor tissue, right, surrounding the vasculature. And when nanoparticles are administered by the intravenous route, right, these nanoparticles, as you can see, this side of the vasculature, as you can see, this is the normal vasculature, and where the endothelial cells are relatively tightly packed, there are not much gaps between the endothelial cells here, right? But in the vicinity of the tumor, you can see gaps between the endothelial cells, right? So when nanoparticle formulations are administered, these nanoparticles would be leaking through these gaps and gets into the tumor tissue, right? So these gaps are generally to the tune of around 150 to 400 nanometers. And nanoparticles, because of their smaller size than the gaps that are created between the endothelial cells, would be extravasating or leaking into the tumor tissue, and tumor cells would be endocytosing these particles, and the particles would be broken down, and the drug would be released, would be showing the cytotoxic effect, right? In addition to that, in a tumor vasculature, in a tumor tissue, lymphatic system is fully developed, and therefore the lymphatic drainage is poor. Therefore, the particles would be accumulated in the tumor tissue here, right? And this effect is called enhanced permeability and retention effect. Enhanced permeability because of the gaps between the endothelial cells, and the retention of the nanoparticles because of the poor development of the lymphatic system in the tumor tissue, right? So this is about simple nanoparticles. In the insert, I have shown you the nanoparticles that are coated with or conjugated with a ligand, right? The small bucket-like structures. And when we use, and once these nanoparticles are loaded with the drug molecules, and when we use this formulation in vivo, the ligand would be binding to the receptors that are expressed on the tumor cell surface, right? And would be undergoing receptor-mediated endocytosis, and endosomes will be formed. And when these endosomes, when they fuse with the lysosomes, the particle will be broken down and the drug would be released and will be showing the cytotoxic effect of it, right? So even when we use a simple nanoparticles, or when we use ligand conjugated, ligand coated nanoparticles, they would be showing the targeting effect. When we use simple nanoparticles, we call it as a passive targeting or targeting due to EPR effect. And when we are coating with a ligand, it is receptor-mediated endocytosis effect or the receptor-mediated endocytosis targeting effect of the nanoparticle, right? So to increase the cellular uptake of cisplatin, what type of nanoparticles we would like to use? Well, in the literature, there are different types of nanoparticle systems, such as the polymer-based and the liposome lipid-based nanoparticles have been reported. But unfortunately, the drug loading in these nanoparticles has been less than 5%. Okay. So we were thinking, what kind of nanoparticles would be ideal for loading cisplatin into the nanoparticles. So we have selected dendrimers as the nanoparticle structures, and dendrimers are these are the synthetic particles, synthetic macromolecular structures, and which are synthesized by iterative chemical reactions, generally starting with a core molecule. It could be ammonia or ethylene diamine or any other amine containing molecule. When it is reacted with methacrylate, right, followed by ethylene diamine, from one amine you would be getting two amines, right? And again, this is one set of reaction. And when this is reacted with again methacrylate and ethylene diamine, you would be getting from each terminal amine, you would be getting two amines. So, the number of amine groups or the functional groups would be keep increasing. So when we are reacting with methacrylate, the terminal groups will be COOH, and when we further react these functional groups with ethylene diamine, we will be getting amine as the function or the terminal function groups, right? And therefore, by increasing the number of set of these reactions, the number of surface function groups would be increasing, right? In this particular case, we have used polyamine dendrimers, which are synthesized from ethylene diamine as the core molecule. What has been shown in the literature is that because of the presence of the amine groups on the surface, right, and because their small size, generally a polyamine dendrimers of generation 4 is about 5 nanometers. Okay, as the number of generations increase, the size would be slightly increasing, but it would be generally less than about 10 to 20 nanometers. And these dendrimers have been shown to be efficient carriers for enhancing the permeability. We know this much of information from the literature. Since the presence of multiple functional groups on the surface is increasing the permeability, perhaps loading the dendrimers with cisplatin might increase the permeability of this tumor cell surface and increase the uptake of the cisplatin. So that was the idea behind selecting dendrimers as the carriers. So what we have done was, initially, we have loaded the PAMAM dendrimers, polyamine dendrimers, with cisplatin, and we want to see if the uptake of cisplatin is increased into the cells. Is it increasing the level of DNA adduct formation? Is it increasing the survival of that particular tumor model or in vivo model? So we have done some in vitro and in vivo tests with different cell lines as well as in vivo models. Here I'm showing you 4 different cell lines: SKOV3, OVCAR3, and A2780 CT7. So SKOV3, OVCAR3, A2780 are the ovarian cancer cell lines which are sensitive in nature, means they respond to the cisplatin. CP70 is the cisplatin resistant cell line, right? So as you can see from this data, I think, I'm sorry, the y-axis here is not quite visible here. This is the amount of platinum per amount of DNA, nanograms of platinum per nanograms of DNA, right? And the other y-axis is the amount of platinum per amount of protein. The amount of protein means we are talking normalizing the data in terms of the number of cells. So as you can see from this data, in all these cell lines, this is the control. So when we are using COOH dendrimer or amine dendrimer or NH2 dendrimer or a biotinylated dendrimer, the amount of cisplatin, the amount of DNA adduct formation has significantly increased in these two cell lines, except this particular cell line. This is also a sensitive cell line. But in a resistant cell line also, you are seeing a significant increase in the DNA adduct formation, both for NH2 as well as the biotinylated dendrimer formation. So overall, what we can say is that the amount of cellular uptake of cisplatin when we are using dendrimer cisplatin complexes has significantly increased, right? So more DNA adduct formation is expected to lead to more, the cell death, right? So after confirmation that the dendrimer cisplatin complexes have increased the DNA adduct formation, we want to test if this adduct formation would be translated into in vivo therapeutic efficacy. So what we have done is we have developed SKOV3 xenografted tumor model in athymic nude monkeys, and we have done the pharmacokinetic and pharmacodynamic studies. In pharmacokinetic study, in a separate group, we have tested at 4 hours and 24 hours. We have collected the ascites as well as the blood samples, and we have tested the cisplatin concentration in these samples. As you can see, comparing with simple cisplatin, the concentration with the amine terminated dendrimer and the biotinylated dendrimer has been increased both at 4 hours and 24 hours. Though it is to some extent a good news, but it has a concern. We will address that in a minute. So at these time interval, at 24 hours, we have collected different tissues, such as the liver, lung, etc. Right? Liver, lung, heart, kidney, all other different tissues, and where you can see here increase in cisplatin concentration with the dendrimers as compared to simple cisplatin, perhaps because of uptake of the nanoparticles by the RES system, that is liver, spleen, kidney, etc. In its back, the important result is that as compared with the control, the accumulation of cisplatin in the ascites cells, the fluid that is accumulated in the peritoneal cavity, the concentration of cisplatin is higher comparing with the control, and the same in case of the blood too. The concentration in the blood is more than the compared to the control. So here the concern is that while the concentration in the ascites is more, the concentration in the blood is also more. What it means is that since permeability of the dendrimers is more, perhaps the dendrimers are more permeable also across the peritoneal cavity and getting into the systemic circulation. And if there is more concentration of cisplatin in the blood comparing with the control, what it means is perhaps it might induce higher cytotoxicity, which is a big concern for us, right? So putting it aside, does it really translate into increasing the efficacy in the tumor model? Well, we have checked the survival of the animals following the administration of cisplatin and dendrimer complexes, biotin-related complexes, comparing that of the control. With the control, the survival rate was the number of days that the animals were surviving was about 25 to 26 days. And with simple cisplatin, it was about 32 days. But with both the dendrimers complexes, the survival rate was significantly increased. Okay. While the survival rate has significantly increased, the concentration of the cisplatin in the systemic circulation was also high. Therefore, it might lead to systemic toxicity. Right. So that's the reason we need to go for a combination therapy or a targeted combination therapy. So when we are talking about combination therapy and targeted combination therapy, what kind of targeting would be ideal for this kind of cancer? Well, in an ideal targeting module, we need to target all the different types of cells, different types of cells in a tumor. So we need to select a ligand that can target different tumor cell populations. The different types of tumor cell populations that are present in a tumor are the resistant tumor cells, the sensitive tumor cells, as well as cancer stem cells. So what we looking into these different types of cells, what we have thought is, well, let us select a ligand that targets all these populations. So we have selected hyaluronic acid, right? Hyaluronic acid is present in, all the tumor cells, many different types of tumor cells. And it binds to CD44 receptor, which is expressed on the tumor cells as well as the tumor stem cells. So thinking that it can bind to CD44 that is present in different cell populations, we thought hyaluronic acid is a suitable conjugating agent or the ligand. So, but before going further, we need to test what is the level of expression of CD44 in different tumor cells, right? So we have selected different types of these cell lines. CHO, which is Chinese hamster ovary cells, that is a control group here. SKOV3, OVCAR3, OVCAR8, OVCAR3, A2780, and CT7, right? So we have seen all these different types of cell lines, and we have seen the CD44 expression. This is the target that binds to hyaluronic acid. And what we have seen is the expression in SKOV3 and OVCAR8 was significantly higher comparing with the other different types of phenotypes, but definitely it is significantly higher comparing with Chinese hamster ovary cells. Okay. After knowing that CD44 expression higher in these cell lines, we want to do the Western blot to see the expression in these cell lines. Okay. So we have compared the SKOV3, OVCAR8 with that of CHO. So even using the Western blot. And as you can see here, in these cell lines, the CD44 expression, the intensity, fluorescent intensity was significantly higher comparing with CHO. So what we have seen is in these tumor cells like OVCAR8 and SKOV3, the CD44 expression was significantly higher. So we can use hyaluronic acid as the targeting ligand. Now we have seen that dendrimers will be increasing the cellular uptake, and hyaluronic acid will be binding to CD44 receptors that are expressed on the different tumor cell population present in the tumor. Now, what kind of combination therapy we would like to use? Well, we want to use a combination of cisplatin and cyanomycin based on the literature reports. Cyanomycin is a coccidiostat and that has been used in the poultry industry. And in 2009, Gupta's research group has identified that cyanomycin has specific affinity for the cancer stem cells, and at nanomolar range, it would be killing the cancer stem cells, especially the breast cancer cells. Based on that data, what we were looking was if saronomycin could be a suitable combination agent with the cisplatin. So we have, put assembled the mechanism of the cisplatin and saronomycin to see if these drugs act complementary. As the schematic shows here, cisplatin would be causing the reactive oxygen species. The mitochondrial damage leads to apoptosis and cell death. And cisplatin also working through mTOR pathway, and it also inhibits the p38 MAP kinase and arrests the cell proliferation. And its main mechanism is a DNA damage, DNA erect formation, and inhibition of p53, leading to apoptosis. That is the common mechanism of cisplatin. Coming to saronomycin, as you can see, it has several different kinds of mechanisms. It is basically a potassium ionophore. It will be working through inhibition of BCL2. It will cause ROS, reactive oxygen species. It will be inhibiting nuclear factor kappa B factor, leading to apoptosis. It will be inhibiting p38 MAP kinase, right? So looking into, and it causes the autophagy, right? And inhibits the vascular endothelial growth factor receptor, leading to, inhibition of angiogenesis. So looking into these multiple aspects, multiple targets here, what we can say is that for both cisplatin and saronomycin, there are some common areas, common mechanisms. For example, ROS, right? And also the p38 MAP kinase and DNA damage, right? So these are some of the areas where cisplatin and saronomycin, they have the same mechanism of action. In addition to the same mechanism of action, saronomycin has other complementary mechanisms that promote the efficacy, promote the cytotoxicity of cisplatin. Given that these two drugs are complementary to each other, perhaps using a combination of cisplatin and saronomycin will increase the efficacy. And if they are targeted, perhaps we can reduce their dose, and therefore we can reduce the toxicity also. So that was the hypothesis that we have used here. So as I was mentioning to you earlier, that whenever in a typical, chemotherapy, right, the conventional chemotherapeutic approach, if this is the tumor containing normal tumor cells as well as the tumor stem cells, when we're going to the conventional monodrug therapy, it is possible that the chemotherapeutic agents will be killing the normal tumor cells, leaving the cancer stem cells, and these cancer stem cells will differentiate and form into the tumor again. But when we are going for a combination therapy that targets both the cancer stem cells as well as the tumor cells, basically all the tumor cells, all the different tumor populations will be killed, and the complete tumor regression will be taken place. That is the hypothesis here. So having seen that the cisplatin and saronomycin can act complementarily, what we have done was we wanted to prepare the dentamere cisplatin complex, dentamere saronomycin complex, and use them at a particular ratio. So here is the schematic showing that when these dentamere complexes are conjugated with the hyaluronic acid on its surface, these dentamers are loaded with different drugs, cisplatin and saronomycin separately. And when we use them at a mixture at a particular ratio, this ligand would be binding to CD44 receptor expressed on the tumor cell surface, undergoing endocytosis. and right, and then the complexes will be released here. And as you know, that nanoparticles are not the substrates for the efflux proteins, therefore they will not be effluxed out, and the cisplatin will be showing its efficacy by DNA adduct formation, and salinomycin would be showing its efficacy through non-nuclear pathways, showing a combination chemotherapy efficacy. So that is the hypothesis. So before going further, though we are trying to develop a combination chemotherapy using dentamol cisplatin and dentamol salinomycin complexes, we would like to test the efficacy of this combination therapy of salinomycin and cisplatin in different cell lines. Okay. Here we have seen three different cell lines. XLC496, which is a ovarian cancer stem cell, CP70, cisplatin resistant cell line, and 2008C13. This is also resistant cell because the target population, tumor population here is the resistant cells and the cancer stem cells, right? As you can see, increasing the concentration of one drug with the and keeping salinomycin constant at its IC50, and similarly with other different cell lines, as the concentration is increasing, this percent viability is significantly decreasing, right? So we are seeing a significant decrease in cell viability. So cell viability in all these cell lines, in the stem cells and in the resistant cells, right? By using a combination of cisplatin and salinomycin. So based on this data, what we have hypothesized is that if a targeted delivery of the cisplatin and salinomycin combination is used in the form of dentamol cisplatin, dentamol salinomycin complexes, their cellular uptake would be enhanced, cellular accumulation would be occurring, increases the DNA adduct formation, and promotes the apoptosis and reduces the stemness of the stem cells and reduces the toxicity of these combination drugs. So this is what we have hypothesized. To test this hypothesis, we have prepared the dentamol cisplatin and dentamol salinomycin complexes that are conjugated with hyaluronic acid, and we have tested their efficacy in vitro, in vivo, ex vivo, as well as in vivo. So initially what we have done, we have collected the primary ovarian cancer cells. So these are the cells basically from the patient. The tumor would be taken out, would be minced, and also trypsinized to get single cell suspension, okay, from the tumor. And these are the ascites tumor cells. You might be knowing that in the late stage ovarian cancer patients, the patients will be developing ascites fluid because of the lymphatic drainage is blocked. Therefore, fluid will be accumulated in the peritoneal cavity, and sometimes this fluid may be anywhere from half a liter to 5 to 6 liters too. Okay, so huge volume of ascites fluid, and which contains a lot of tumor cells, right? So generally, ovarian cancer patients would be visiting the physician to tap this ascitic fluid, right? And at the same time, the chemotherapy or the radiation therapy would be administered into the peritoneal cavity. So what we have done is we have collected the tumors from the patient, and we have made into single cell suspension, and we have collected the ascites fluid from the patient. We have centrifuged it and collected the tumor cells. And these tumor cells were subjected to a combination of cisplatin salinomycin complexes at different ratios. As you can see, this is the dentamol cisplatin combination. Okay. These are the individual the complexes. So when we are using a combination of dendromus cisplatin, dendromus aranomycin complex, the aranomycin complex, the cell viability has significantly reduced. Not only in the primary ovarian cancer cells, but also in the ascites tumor cells, the combination therapy has significantly reduced its viability. Similarly, we have developed the cancer stem cells in SKOV3 cancer stem cells. The way we can develop the cancer stem cells is that we induce tumor into the mice. Once the visible subcutaneous tumor is developed, sacrifice the animals, collect the tumor, mince them, right, and culture them. And from there, once the tumor cells are grown from the xenograft, we will be subjecting them to cancer stem cell separation by using the Aldifluor assay, so that you can separate by flow cytometry, you can separate the cancer stem cells based on Aldifluor assay. And these cells were cultured and then subjected to combination therapy using hyaluronic acid conjugated cisplatin, hyaluronic acid conjugated dendromus aranomycin, a different combination ratios. As you can see that dendromus cisplatin, dendromus aranomycin complexes have shown higher cytotoxicity. Similarly, this is the syngeneic model, that is not a human model. So basically, a tumor cell that induces cancer in the animals, in the mice, and those cells were used to induce tumor in the animals, in the mice, not in the athymic nude mice, but in the normal mice. And once the tumor is developed, the tumor is taken out, cultured into the in vitro, in the culture flasks, and the cancer stem cells were isolated. So irrespective of type of the tumor cell model that we are using, whether it is syngeneic model, cancer stem cell model, whether it is primary model or the ascites tumor cells, the combination therapy of dendromus cisplatin, dendromus aranomycin has increased the cytotoxicity. Then what we did, we used the A2780 and SKOV3 stem cells, and we have done the serial dilution assay. What we'll be doing in serial dilution assay is we'll be diluting the cancer cells, to achieve a number of approximately arbitrary number of 50, 20, 10, and 5, and 1. And when they are subjected to this combination therapy, if those cells are not responsive to the treatment, then the cell number will be increasing. And since these are stem cells, they would be forming into spheres as shown here. So in case of A2780 and SKOV3 stem cells, right? When these cells were treated with vehicle, you are getting a big sphere of tumor. When it is treated with simple cisplatin complex, the sphere is bigger. Simple salaromycin sphere is relatively small, but similar to the of cisplatin. But when a combination therapy is used, the size of the sphere is significantly low. The same results were seen in case of SKOV3 derived cancer stem cells. So what this shows is that when we are using a combination of cisplatin and salaromycin dendromer complexes, the sphere formation assay, the cytotoxicity assay have significantly shown that the combination therapy is more effective than individual agents. Then what we did, we want to see the metastasis of the tumor when a combination therapy is used. So here, for the imaging purposes, we use the ES2 cell xenograft model. Okay, so in this model, these are the cells that are tagged with green fluorescent protein luciferase. Basically, these are the fluorescent tag. So when we have induced the tumor into the ovary, it is called orthotopic model. Basically, we will be injecting the ovarian cancer cells into the bursa, the superficial layer of the ovary. And once the tumor is developed, we will be imaging them. Since the cancer cells are fluorescent in nature, they are luminescent in nature, right? We can track the metastasis of the tumor cells by doing imaging, okay? So after a few weeks, once the tumor is developed, generally when we induce tumor, it takes about 3 weeks for the tumor to develop. And we have started imaging the animals. And as you can see here, this is the vehicle. And then once the tumor is developed, we will be administering different treatment agents. In case, and periodically we will be imaging these animals. In the group where the vehicle is only used, the tumor has spread all over the peritoneal cavity. But when cisplatin and salidomycin alone are used, the spreading is there, but most of them are localized, but still, you can see the metastasis. And it's when we have used simple cisplatin and salidomycin, not the targeted formulation, but simple combination of chemotherapeutic agents. Also, we have seen the metastasis, but when dendrimer cisplatin complexes conjugated with hyaluronic acid have been used, the metastasis is almost negligible and the tumor has reduced very, very significantly. The same thing is shown in terms of the tumor volume and the tumor weight. As you can see, the tumor volume has significantly lower comparing with other combinations, right? So when we are using a combination of cisplatin and salidomycin in the form of dendrimer complexes that are targeted with hyaluronic acid, the tumor has completely regressed and its metastasis has significantly reduced. So it is very common to see if this kind of therapy is efficacious in tumor organoid model as well as patient-derived xeno explant model. So what we have done was, these are the tumor cells. We have isolated them and put it into the Petri plates and treated with the different agents, the combination therapy as well as the individual therapy. And this is the organoid model. And we have checked the patient-derived and the viability of the organoids at different time intervals using the cell titer flow assay. As you can see, with the vehicle, the cell viability is very high and it has significantly reduced. It is almost negligible when a combination therapy is used. And similarly, as you can, the same thing is visible here. When the vehicle is used, the tumor has grown into, a sphere here, but the number of tumor cells with the combination therapy is significantly low. And there is another assay here called explant model, wherein basically in a Petri dish, you will be using the gelatin sponge on which you will be using the ovarian cancer tissue that are collected from the patient. Generally, these are 1 to 3 millimeters, very small tissues, and this is the nutrient medium. And once these the tumor explants have been treated with different treatment agents, we'll be measuring the Ki67. Ki67 is a nuclear shuttling protein, an antigen whose expression increases as the cell proliferates. So it's an immunohistochemical assay. So we have seen that as the combination therapy is used, the Ki67, which is a marker for cell proliferation, is generally, its expression is significantly lower comparing with all other treatments, simple agents alone. The same thing is shown in terms of the the imaging with the tissue imaging in histochemistry. So the histology slides have also shown that the Ki67 expression has significantly lowered when combination therapy is used as compared to the other the treatment model. Finally, we wanted to test the efficacy of this combination therapy in patient-derived xenograft model, wherein we'll be collecting the patient tumor cells, and these tumor pieces would be administered, implanted into the athymic mule mice, the skid mice, and once the tumor is developed after 3 weeks, we'll be treating these mice with the combination therapy at this time, and at different time intervals. The tumor volume will be measured. As you can see here, the combination therapy has significantly reduced the tumor volume, and when the end point comes, basically we have sacrificed the animals and we have collected the tumors, and as you can see, the size of the tumor is significantly lower as comparing with the individual agents as well as the simple vehicle control. So we have seen that the cisplatin cytotoxin complexes have significantly increased their efficacy, and there is a reason for use of cisplatin cytotoxin for the ovarian cancer therapy based on their complementary mechanisms. However, we need to find out what exactly happening when cisplatin cytotoxin have been used. Is there a synergism, right? So generally, these mechanistic aspects will be taking several years to elucidate the mechanism. We have just started with SKOV3, and what it looks like is MAP kinase as well as the P53 genes have been involved in the synergism. But this transcriptomic genomic analysis has been going on, but we have done with only one cell line, so I'm not showing much data about it, but just I wanted to show that different proteins have been, gene sets have been analyzed, and we are further processing it. So we have seen that cellular uptake is increased, efficacy is increased, and even in resistant cells, the efficacy, and the stem cell cells efficacy has been increased. What about the toxicity? So what we have done was we have tested the toxicity of dendritic cisplatin complex and the dendrimers, their chemotoxicity, and what we have seen was at a concentration of less than 10 micromolar, the RBCs were, the hemolysis was very minimal. The cell viability was, similar to that of the control. Okay, and in the in vivo models, we have collected the tissue, the kidney, and we have done the histopathology studies, and the kidney tissue, the histology of the groups that are treated with the combination therapy is not significantly different from that of the individual agents, indicating that by using a combination therapy, there is no increase in toxicity, which is a good sign. So, however, we have to further analyze the dose-dependent toxicity and long-term toxicity of this combination. So those things still we are supposed to do. So in conclusion, the dendrimer cisplatin complex increases the cellular uptake and the DNA adduct formation, and also the cytotoxicity, even in cisplatin sensitive as well as the cisplatin resistant cell lines. And because of the small size of the dendrimers and the dendrimer complexes, their stability as well as the good drug loading capacity. Generally, we have seen a drug loading capacity of around 11%, as opposed to about 5% with other type of carriers. So then dendrimer cisplatin, dendrimer cytotoxin complexes have been characterized, and they appear to be optimal comparing with other carrier systems. We have seen the combination therapy in different cell lines. In cancer stem cells, the combination index, which is a representative of synergism of 0.54. If it is less than 1, it is called synergism. So we have seen a combination index of 0.54, and in cisplatin resistant cells, it is 0.6. And in the primary cells, combination index is 0.3, and the tumor ascites cells, it is 0.4, indicating that all these combination index values are less than 1, showing that they are highly synergistic in nature, right? And we have seen the efficacy in other ex vivo and organoid models. We have done the efficacy in 2 in vivo models. One is the ES2 cell xenografted model, where we have shown that the combination therapy is superior to individual complex, and metastasis is significantly reduced. And even in patient-derived xenograft model and explant model, the efficacy with the combination therapy, targeted combination therapy, is significantly higher. And however, the final pharmacokinetics of these complexes, and especially toxicity studies, have to be further done in detail. But what we have seen from the mechanistic studies is that genes that are regulated, such as the P30 and MAP kinase and p53 gene sets, appear to be significantly affected in one cell line, that is SKOV3, but further we need to analyze with other cell line models also. I think I will stop here. This is one slide that I would like to show in terms of the in vitro release testing using thoughtful products. We develop models of generic products and their in vitro release testing by using microdialysis. These are the, these are 2 formulations that we have developed, and this is the standard formulation, the RLD, that is marketed formulation, and we'll be testing their in vitro release using microdialysis and USP4 methods. Here it is. We have tested the in vitro release testing at every 2 minutes, right? And this is the combination, the Durezol, the standard one, as well as our test product, and this is a non-equivalent product. So we can differentiate the drug release profile of the marketed product versus equivalent formulation and also the equivalent formulation. What we have shown is that the marketed product and the product that we have prepared are showing the similar kind of release. This is the sampling was taken every 2 minutes. Similarly, when we have done the sampling every 5 minutes, as you can see, the Durezol and F1, that is our formulation, these are all supposed to be equivalent, and this is the F3, which is non-equivalent. Its release profile is good. So in nutshell, what we can say is that we can develop a in vitro release testing method that can differentiate formulations that are produced by different manufacturing processes. Okay. So finally, I would like to conclude saying that my research has been funded by different agencies such as NIH, Department of Defense, FDA, and the university grants, as well as National Plant Growers Association. Here are a list of my collaborators who have contributed significantly in some of these, some of the work that I have presented, and some of the projects that are going on, and postdoctoral fellows, as well as the graduate students. Thank you for your attention, and I will be happy to take any questions on this point. Thank you.
Over to Dr. Vijay Kumar to conclude the session.
Yeah. Thank you, Dr. Srinath Palakurti. For us, it's daytime, but for you, it's night, and that too weekend.
Right,
Thank you
right.
for spending your valuable time for this session. And you have very neatly explained, very clean and neat. the survival rate is very less, 11% of ovarian cancer. Yeah, with your efforts of different combinations using ligands with your liposomal therapy, we hope the survival rate will increase. And congratulations for your research. And in fact, you are the first one, I think, for developing release system for ophthalmic products in generic products. Okay, you are the first one to first research group for developing a method for the release system for ophthalmic products. Congratulations once again. Audience, if they are having any questions, you can raise their questions.
Thank you so much. Thank you for giving the opportunity. I appreciate it.
Over to Ganesh.
Hello, is it, I'm audible?
Yes,
Yes,
sir,
yes.
you're audible now.
Yeah. Thank you very much, Dr. Srinath Palakurti. Wonderful information. In fact, I would like to convey my sincere thanks to you, because whatever the work which is carrying out in your laboratory, it is enormous, and definitely our students and faculty might have benefited from this, and whatever the projects which are going on, and especially the nanoparticular drug delivery systems. And in fact, it is a great work, we can say that. And I hope in near future also you will be continuing with us. We would like to know whatever the work which is going on. In fact, it is very much useful to our faculty as well as then students also. What a wonderful, really, we loved it. In fact, whatever the work we are carrying out, formulation. In fact, I would like to request in near future for joining whatever the projects which are going on, especially the topicals as well as then long-acting injectables. In fact, our pharmaceutics department is especially working on the topical formulations and long-acting injectables, nanoparticle drug delivery system. So that's why we would love to share with you this in near future. Thank you very much on behalf of Sriman Suresh Dada Jain College of Pharmacy. And now I would like to request our technical team to display the certificate of appreciation of today's speaker, Dr. Srinath Palakurti, as well as the chairperson, Dr. Vijay Kumar. Please, our technical team. So Dr. Srinath, kindly accept the e-certificate on behalf of Sriman Suresh Dada College of Pharmacy. It is a small token of amount on behalf of our college. Kindly accept this certificate of appreciation.
Thank you so much. I appreciate that.
Yeah, I will be sending with this one. And yeah, Dr. Vijay Kumar.
Yeah.
So, Dr. Vijay, please accept the certificate of appreciation on behalf of Sriman Suresh Dada College of Pharmacy.
Thank you, Mr. Ganesh. I'm happy to associate with you on this occasion.
No,
and,
people like you, especially technocrats, we would like to invite, as well as our students want to be always with you. Okay, so wonderful session. So on behalf of our college, I would like to put my sincere thanks to Dr. Srinath Palakurti. As Dr. Vijay Kumar said, it might be 11:30 in Texas. Is it, Srinath? Is it 11:30 or 12 o'clock?
Yes.
Yeah, in spite of your busy schedules, and as I said, today is Sunday morning for us, but it is a Saturday night for you. And So definitely we are very much thankful to you. And at the same time, I would like to put my friendly thanks to Dr. Vijay Kumar, who is my classmate. Sir, thank you very much for sharing this session.
Yeah. Ganesh, it's a
Yeah,
privilege.
thank you very much, sir. And thank you all the delegates. Thank you very much. And now I'm handing over to Dr. Chatpalivar, sir, to continue for further session.
Dr. Ganesh,
Yes, sir.
I would like to request Palakutti sir
Yes, sir.
to guide us on some projects, what you are conducting or what you are carrying out in your institution. Those are
Okay.
possible here in India in our institution. So if you can
Okay,
highlight
fine.
what kind of facility you have created there for those projects, and we can have a small interactive session on this platform only.
Okay.
What are the requirements of your research? Those can be created here in our college.
Okay.
So, are you asking me to talk about it now, or do you want to have a separate interactive session at a later time?
No, we can discuss now also. There is no problem.
Well, definitely, there should not be these are the basic industry-oriented projects. And so, unless I know the analytical instrumentation that you have at your college, I may not be able to talk a lot. I'm guessing that, I have seen some labs as you have shown in your video, but if you can tell me what kind of instrumentation, analytical instrumentation that you have, then we can go from there. But generally, all analytical characterization instrumentation, depending on the type of the product that you are interested in, physical-chemical characterization, as well as drug release instrumentation that you have, and if you can make a list of it, then probably definitely I can tell you what else you definitely need, and to develop or to help the industry to develop the generic formulations.
Yeah, so, Srikant, I would highlight myself as well as then my pharmaceutics faculty are working on various projects, especially the nanoparticular drug delivery systems as well as the topical drug delivery systems. And my faculty is very much instrumental in developing, especially the ophthalmic drug delivery system as well as the nasal drug delivery systems. And we have a small laboratory, a machine room in which we have the spray dryer, Franz diffusion cell, as well as fluidized bed processor and compression machines, as well as then coating pans are also there in the small where we are working, and we are working academics
to add on.
Yeah. And we are working on the academic point of view, as well as we are helping nearby college students as well as then faculty, as well as then some industrial projects are also going on, especially on fluidized bed processor and spray dryers. And analytical point of view, as I said, we are having a 8-station Franz diffusion cell for the in vitro drug release system for the topical drug delivery system. We are having the Franz diffusion cell in our laboratory. So this is an analytical point of view. We are having the UV spectrophotometers as well as then HPLCs.
Yeah, so HPLC is a must, and since you have the HPLC and you have the Franz diffusion cell, and probably I'm sure that you must be having the USB2, and if you can add maybe USB4, I'm not sure. Or I think the best way of enriching the instrumental facility is that to work with the industry, and if they can support this instrumentation part of it, then perhaps we can have a USB4, and we can have the microdialysis setup, and and we can go from there.
Okay.
And you know, these are the main, you know, the in vitro drug release testing apparatus. Generally, not having the apparatus is one part of it. The other dimension is basically knowing what are the factors that we need to optimize. Most of the industries, what they do is they have the contract with the GMP facilities. They will be doing all these studies, but what they do not know is whether what kind of instrumentation, what kind of factors that they need to look into, and how to analyze the data. And if the data is consistent with the expectations, how to justify that it is consistent. And there will be, you know, most of the times what happens is they come to me with the FDA's comments on their pre-ANDA, and they say, well, you know, they will send 2 or 3 pages of questions saying that, you know, the FDA is saying this is not complying with what is expected, and why the data is coming like this, why this kind of factors have been tested, how are you validating this procedure, so on and so forth. So those need basically the technical input on the data that they are generating. So they spend a lot of money in generating the data, but at the end of the day, that is not what the FDA is looking for. FDA is looking for validation of what you are doing, validation of the protocols that you are using. So definitely we can help them. And of course, you know, in the process, we can establish our laboratories also with the advanced technologies and advanced instrumentation. So if we can have a meeting with the industry supporters, probably who can support your labs or our labs with some kind of funding, then perhaps, you know, we can help them with coming out with the protocols for, you know, generating the data for subject of pre-NDA.
Okay.
Looking at our facility in our institution, our laboratories are equipped as Dr. Basarkar has mentioned. I would like to ask what kind of other instruments are required for developing the strength of the lab in your area?
Well, I'm sure that, you know, most of the labs who are working in pharmaceutics will be having a zeta sizer, right? A particle size analyzer with a zeta potential. And either you can use a transdiffusion cell or you can use a async chamber for diffusion studies. If you have the USP4, that will be good for most of the in vitro drug release testing. USP4 would be good, especially for the long-acting products. Yeah, sometimes you know, pharmacokinetic studies are important, but it all depends on how you are approaching. Most of my industrial work is about bio-waiver. Okay, so generating the data to to avoid in vivo studies and in vivo data, right? So most of the time we would not be doing pharmacokinetic studies, but it is not easy. A lot of things will be coming into the picture in terms of justification, and for providing the justification, we need to do a lot of other studies too. So HPLC and sometimes you might need LCMS. It might be a big ask, you know, asking for LCMS, but what happens is when FDA is not sure about something, they will try to ask questions such as, for example, have you seen any metabolites there? Well, to see the metabolites are there, generally in a product characterization, analyzing the metabolite concentration would be very, very difficult because it is not in vivo samples, it is the in vitro samples. So you would not see any impurity or degradation products there, right? So to analyze the degradation products, definitely you need LCMS. And they are relating that data to the in vivo metabolite, right? So if you have LCMS, it would be a plus, but at least you should have, the definitely a UPLC or HPLC with relevant detectors. The reason I'm saying detectors because most of the times mass balance of different constituents of your formulation is a big task. Okay, when you are doing the in vitro release testing. In the donor compartment and in the acceptor compartment, you are supposed to show that your excipients are not diffusing, and if they are diffusing, they are not causing any, any interference with the detection of your active principle API. So those kind of things. So as it becomes more complex, then you need more complex instrumentation. But I think these are the common things, and we are specialized in microdialysis most of the times. There are only 2 labs here in the US, and we are the first one to show that microdialysis can be used for topical purposes. So microdialysis is something which is more time-consuming process, but gives a lot of data relevant to the biorelevant in vitro release testing. So I think that's it, and probably you would be having a lyophilizer, you would be having a microfluidizer, right? If not, those things would be good, a microfluidizer and a lyophilizer, and other instrumentation such as the DSC, FTIR, XRD, right? So probably you're all aware of these kind of things. So
Yeah.
even if you have access to these facilities, that will be great.
That's great. Because what I think is a group of researchers, if coming together and they are working on one particular line, and but if they concentrate on one particular area, a significant useful or industry useful kind of research can be done. And person like you sitting in the US, you must have gone through a certain path. That's why I was eager to know in this particular area, if we want to carry out our research, what type of facilities are required. Anyways, thank you very much, sir, for your time.
Thank you.
You have given almost it's midnight there, so I won't bother you too much.
I appreciate that.
Thank you.
Generally, yeah, most of the consultations with the Indian companies, it is in the evenings and the weekends. So sometimes, I'm used to this, but
Okay.
thank you. Thank you for the invitation.
Over to you, Ganesh.
Okay, thank you very much, Dr. Srinath, being with us. And we are very happy that you have kindly accepted our invitation, and a lot of benefits for our faculty as well as students. Definitely, we will look forward to work with you in near future also. Please guide us, our faculty as well as guide, because what is the research which is going on in US is always in front of India. So we will be very much happy to learn from you. Thank you very much. So we will keep in touch. Okay, thank you.
Thank you for the conference organizers and Dr. Basarkar, Dr. Vijay Kumar, my seniors at the Kakti University, and also all other organizing committee members and Dr. Kasani, right? And other faculty members. And thank you for your attention. I appreciate, and I enjoyed the opportunity. And definitely, whatever is possible from my side, I will be happy to contribute.
Thank you.
Okay. Thank you, Srinath, and good night. Bye-bye. Okay. Thank you, Vijay, being with us. Back to Dr. Chatpallivar, sir.
Thank you, Dr. Basaka. Thank you, Dr. Upasani, sir. Now we are heading towards a session, most awaited session by Dr. Saranjit Singh, sir. Recently, he has been retired from his services, but still being a scientist and being a guru in the field, he definitely thrives in teaching us, preaching us different aspects of pharmacy. He will be soon joining us
Yes,
to deliver his lecture.
Dr.
Okay.
Chakravarti, you tell him his, tell us about his association with SNJB PharmaCon.
Yes, sir. Definitely. He will definitely be speaking about us, sir. And he will be mentioning the sole purpose behind being associated with us and the purpose of encouraging research. Yes, we do have other eminent personality from the field of pharmacy, a very renowned and a very well-known personality. I hope he is in the class.
Yes.
Yes, Dr. Udupa, sir.
Dr. Udupa, sir.
Other personality, eminent personality. Good morning, sir.
Good morning. Please unmute
He
me.
will be. Yes, sir. You are unmuted.
You are unmuted.
Can hear you.
And put on your video, sir.
Video. Yeah, start video. I'm putting, but you have to unmute.
Yes,
Yeah,
you are audible. You are unmuted.
Start my video. Yeah.
Okay.
Okay.
Yes, sir.
Yeah.
Good
Yeah.
morning,
Yes.
sir.
Yes.
It's quite foggy here.
Good morning. Good morning. Yeah.
Sir, I
Yep.
seek your kind permission to introduce you for the attendees.
Yes.
The younger generation must know your hard work and your achievements, sir. It is quite an illustrious curriculum vitae I am reading. I feel blessed to have a chance to interact and to read, to introduce you to the attendees. Dr. Udupa, a person known for his great leadership qualities. You have been a professor and principal at Manipal College of Pharmaceutical Sciences and known as a renowned educationalist and eminent scientist in the field of pharmaceutical technology. Also known for his noble academician in the pharmacy profession. You are known as a great administrator who practices simplicity, honesty, humanity, and have a very deep concern for every individual. May he be a friend, a staff member, or student. Under your dynamic leadership, sir, the UG and PG courses of pharmacy and pharmaceutical technology had made significant contributions. Here are a few of your contributions to the scientific field. You have conducted 75 workshops, received 51 research grants from several funding agencies in country. You have flown away 16 times for delivering invited lectures abroad. Under your guidance, 95 students have been benefited for their MPharm dissertation and 28 students have been awarded their PhDs. To your credit, you have earned 7 patents and you have successfully made 27 industrial collaborations. You have presented in national and international conferences for the not less than 350 times you have presented your research papers. That is a towering achievement, sir. The research papers to your credits are 425. 425 is a big number, and with the staggering H index of 49, it itself is a very big achievement. To your credit, you have 5 books and you have authored 100 review articles. I suppose these review articles are not forced review articles. These are invited review articles. There is quite a big difference. As always said, sincere hard work is always reciprocated with a lot of honor and award. Here is a glimpse of few of the awards bestowed upon you, sir. Dr. P.C. Dandia Endowment Trust Research Award in Pharmaceutical Sciences in the year 1994-96. Best Research Paper Award in many pharmaceutical conferences. Best Principal Award for the year 2001 by APTI India. Pharmaceutical Scientist of the Year in the year 2008 from Indian Association of Pharmaceutical Sciences and Technologies. IPA Fellowship Award in the year 2009. Honorary Fellowship Award conferred by the Association of Biotechnology and Pharmacy in 2007. Sir, during your tenure, you have served many respectable positions as Secretary of Government Council, College of Pharmaceutical Sciences at Manipal. You have been an advisory member, editor to many journals. Few of them are Indian Journal of Pharmaceutical Sciences, Pharma Today, Indian Drugs, Pharma Link, and many more. You have been an executive committee, a member of executive committee and board of management at Manipal University, Manipal, Rajiv Gandhi University of Health Sciences, Bangalore, and Mahe. You have chaired the monitoring committee of many respective authorities like TIFAC, CORE in NDDS. You have been an advisory board member of TIFAC CORE in pharmacogenomics at Manipal University. You have been an examiner at all levels in several universities of repute. You are also a member of Pharmacy Council of India and NBA AICT New Delhi. With these illustrious curriculum vitae, I welcome you to join us at SNJB PharmaCon 21. Welcome, sir.
Thank you. Thank you. Thank you.
Moreover, I would like to say a few words for Dr. Udupa. Whenever I visited Manipal, I used to observe him. He is such a down-to-earth person. He respects each and everyone. Whomsoever comes to him, he is always ready to help. And moreover, he is having a credential of helping all the associations and organizations without any personal gain. Whenever any association, say for example, it may be APTI, wherever they feel that now location is not favorable, something, and if they go to Udupa sir for help, immediately he will agree to organize APTI, organize any national level conference at his institution. He is having, Manipal institution is having excellent infrastructure and facilities. So he is always ready to share those facilities, and I am very thankful to you, Udupa sir, for this gesture. And fortunately, and most awaited personality of our conference, Dr. Saranjit Singh, has joined our group, and now I will hand over it to Dr. Chatpalliwar again.
Good morning, sir. Welcome to SNJB PharmaCon 21. I would request Dr. Udupa to introduce our protagonist, Dr. Saranjit Singh, sir.
Yeah. Thank you for great. Thank you very much for great introduction. So, without wasting much time, though I am a simple, humble, and among you all colleagues, so you have elevated me and given me this great opportunity to interact and to learn a lot of things, because daily we learn a lot of things. And Dr. Saranjit is a Dayan and is a leader in pharmacy profession and research. So without wasting much time, now let us listen Dr. Saranjit Singh. So in brief, I would like to introduce Dr. Saranjit Singh. Dr. Saranjit Singh is ex-dean and ex-professor and head of the Department of Pharmaceutical Analysis of
Hey.
NIPER, National Institute of Pharmaceutical Education and Research, Mohali, Punjab. He is gold medalist and alumnus, Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. I have a long association with the last more than 40 years, 4 decades, both in NIPER, NIPER Punjab, Mohali, as well as Punjab University, because every year, because a lot of our students, faculty, we had a lot of exchange, lot of collaboration, and with Dr. S.K. Kulkarni and a lot of our people, we had a lot of association. So I was in touch with Dr. Singh, and Dr. Saranjit Singh is in education research for last more than 40 years. He has published more than 250 very quality, high quality research papers, journal articles, review articles, and book chapters. He has one patent and one edited book on drug stability to his credit. Before his superannuation from NIPER, his team executed more than 100 industry-sponsored projects. He is regularly invited to hold full-day training sessions for pharmaceutical industry in India and abroad. He has delivered more than 550 invited lectures and spoken at the forums of American Association of Pharmaceutical Scientists, AAPS, United States Pharmacopoeia, USP, Drug Information Association, DIA, Indian Pharmaceutical Association, Indian Drug Manufacturers Association, SSX, etc. He has guided more than 150 master's degree students for their projects and 15 doctoral students. He is a member of expert advisory panel on international pharmacopoeia and pharmaceutical preparation, and also has been a temporary advisor to the World Health Organization expert committee on specifications for pharmaceutical preparation. He is contributor editor for Trends in Analytical Chemistry, TRAC, and editorial board member of several leading journals, including Journal of Pharmaceutical and Biomedical Analysis. Actually, this is a very short, brief biodata resume of Dr. Saranjit Singh. He requires much more details, several, like a book can be written, and eminent pharmaceutical scientists and technologists in that we are editing a book. He is helping me, and from India, more than 40 to 50 such scientists we have identified, and we are that project, and through the help of IDMA, we are going to publish, and within another 2-3 months, we are going to release that book. So with this short introduction, I welcome Dr. Saranjit Singh, and I request him to give his learned, experienced talk, which much awaited. All of you are eagerly waiting, and he will enrich with us his expertise, and he will tell about all the particular regulatory requirements, analytical techniques, etc., whatever experience he has got. So welcome to Dr. Saranjit Singh. Thank you.
Sir, please unmute yourself.
Thank you very much, Dr. Duppa, for a very nice introduction, and my Sat Sri Akal, Namaste, Namaskar to everybody. Dr. Duppa is my esteemed senior, and I have admired him for the research he did at Manipal. I think everybody knows him, and I am privileged that he is chairing this session today. Dr. Pasni, thanks for inviting me for this SNJB PharmaCon 2021. I think I've been associated for the last few years, since it started 5 years, and I have been given an esteemed honor always by the Institute. And thanks to Vivekananda and whole your team for making it possible. Although I would have loved still to come physically to Chandwad, I always like traveling from Nasik to Chandwad on road with Vivekanand. So I'm missing this year.
Thank you, sir. You are always welcome, sir.
I had always a chance to click a few photographs of my interest on the way. I think Vivekanand remembers that a truck was going
We
which
do
was
remember.
carrying the medicines, and I wanted to really have a photograph of that, and that was a given chance, which I'm not getting now.
You're always welcome, sir.
Okay, so nice to have so much big audience today, and I have put the title of my talk today. Now, is it my slides are visible?
Yes, sir, very much.
Yes, sir.
Okay, so greetings to all in attendance and a very good morning. Now, the topic which I have chosen today is related again to more towards the how industry is developing, what changes are happening in industry. Let me give a brief about my interest in International Conference on Harmonization activity. Since my PhD days in 1980s, since my boss, Professor S.K. Baveja, had an interest in stability testing, and he, if you look into his publications, early publications in 1980s, he had published a lot on the degradation chemistry of metronidazole. So during those days, when I started my PhD in 1979, 80, early, and towards that period when I finished my master's in 1979, from that movement, I joined under him, and he assigned me the project, which was on stability kinetics of one of the CDRI drug, new drug developed by CDRI, under the name of bucridine, which happened to be a local anesthetic. So, as per my way of doing things, I got a vial from CDRI Lucknow, having that drug inside. And when it reached me, I found that it was a yellowish powder, maybe 5-10 grams. And I was not very satisfied working with something which was yellow in color. I still remember that I found out from CDRI Lucknow, called somebody. The boss only handed me that this is a vial and you start working on it and do the stability kinetics. Now, I was not very satisfied with the quality of material that was sent, and this is what synthetic laboratories does even in CDRI, that they will synthesize a compound and push for clinical trials or stability testing, which was again a requirement that if you have to push a product in the market, a new drug in the market, you have to carry out all these studies. To cut the story short, I crystallized that compound, and 5-time crystallization I did from pyridine. And as you know how much noxious is to work with pyridine as a solvent, because it was basically crystallized out of pyridine. And believe me, after 5 crystallizations, I got a yield of long needles, which are purely transparent. Now, when I started stability kinetic studies on it and did an initial trial of a few pH, preparing the solution and putting them on stability, I found that the drug in that purest form, when I had very long needles, transparent needles, it proved to be extremely stable. For this audience, I want to say that more is the purity of the compound, you'll find the compound to be very, very stable. Pure compound like gold, which you all buy for your spouses or the spouses who are present here as the ladies, they know that if the pure is highly, when the gold is highly pure, how much life it has. It is there's no end and shelf life for a pure gold. So it is happening with the chemicals also. If they are in a highly pure state, their longevity can be, I will say, infinity. Now, the only way that happened with me was that after boss has given me a problem of stability, studying the stability kinetics, I had to see some decomposition happening. So I had to increase the temperature to 90 degree. And believe me that to get even 5 to 10%, 15% decomposition, I had to have the oil baths. I had to basically put the solutions on heating at 90 degree for almost 1 year to get 5 to 10% decomposition, so that I can really determine some kinetics at some pH. And it was extreme of acidity and alkalinity. So one table in my thesis used to be, means I will have that table after 1 year of conduct of that experiment and the solutions, and in ampoules, I will put the solutions in sealed ampoules and suspend them into oil baths. Now I had a plenty of free time at that moment. And I started working on researching on the whole literature that was available on stability testing at that time. I had plenty because it was 1 year to generate one table. Now, before ICH, International Conference on Harmonization, was born, I could see what developments are taking place at the international level with respect to regulatory. There were initially bilateral meetings between US and Europe, Europe and Japan, and US and Japan, like that, because they wanted that the standards for the technical requirements were varying from region to region, and it was becoming as a bottleneck for industry. Suppose hello.
Yes, sir, you are audible.
I am audible or not?
Yes, sir, you are audible very much.
Okay. Now, so I was basically looking into what is happening on the front of stability testing, and I still have papers of 1970s when the whole concept of 40 degrees, 75% came into picture. Then I could see that this bilateral meeting went on to trilateral meetings. And the reason was harmonization of technical requirements for registrations of pharmaceuticals for human use. Because there was a variation in requirements, like let us say Europe had a standard that you have to do safety testing on 20 rats. And if USA says, no, you have to do on 40 mice, and Japan has something else to be done, it means industry had to do the same experimentation to prove safety for almost, let us say, repeat the studies with the same endpoint with different sets of animals, and it was a triple effort that industry had to do. Now, when it came to quality guidelines, it was very interesting to see that the shelf life of the way the shelf life was calculated was varying from region to region, and that was becoming a bottleneck for export and import of drugs, because these three regions were the major innovators of drugs. And they were the major consumers also, major producers, major consumers, and also major innovators for new drugs. So if you look into the ICH guidelines, Q1, under the head quality, Q1 was a topic which was selected for stability under the means harmonization of stability testing guidelines. Now, question comes over here is, I had been looking into the development of the ICH forum from that inception and started writing small articles with respect to this to sensitize our Indian industry on impeding requirements that are going to be coming on. Now, if you look into today, you go to the website of ICH. Initially, it was meant for implementation in 3 regions. But now the name of ICH has got changed. Earlier, it was International Conference. Now they have made it more formal as an International Council for Harmonization. This change, I think you people might not have realized that this is globalization of the standards they have laid down from 3 regions to globalization. That's why they created their own secretariat in Geneva and created and registered in the name of a council. Earlier, it was only international conferences. They were only having biannual or annual meetings, twice a year in alternate regions. And that meeting was a conference on which they used to discuss what harmonization activity and what topics are to be selected. Now, over the period, I will say towards 1990, they took up then first topic, they took as stability testing, then method validation, then impurities, then pharmacopoeial harmonization, then subsequent, what we say Q5 guidelines, means biotechnology products quality, and then GMP and other things. So this, then Q6 was specification, and then Q7 was GMP. This way, what they did, their effort of harmonization of technical requirements that went over till Q7. Now, what happens next once the harmonization of technical requirements was done? Now, this is the topic of my lecture today. What happened after Q8? So friends, the title I have put over here is the exciting transition from quality by design to continuous manufacturing. Now, maybe perhaps you are not keenly observing what is happening at the ICH forum. That's why I have to bring this lecture today, because this ICH requirements have direct impact on the industry. And you can only stand before industry people if you are yourself aware of the requirements that are emerging on daily basis. So for me, it is a very exciting transition, which has happened from quality by design to continuous manufacturing. And let me say, what is the origin of the title for this talk? This is a constant follow-up that I have been keeping with respect to developments of the ICH quality guidelines over the period. I will say the developments that I am going to talk about, they match even the theme of SNJB PharmaCon 2021, which is entitled Research, Innovation and Technologies, Challenges and Opportunities in Pharmaceutical Sciences. Now, when we say, let us look, spend a minute on the title itself. Research, you cannot develop a technology or innovation until the research is done. And then research can lead to innovation, and innovation can result into technologies. So there's a direct link with the theme that have been set between the research, innovation, and technologies. Within these, what are the challenges and what are the opportunities with respect to pharmaceutical sciences? Now, keep in mind and look into the flow of my lecture, how it meets the theme and match the theme of what has been put off SNJB PharmaCon 2021. Now, what my lecture is going to cover is what are the recent advances where the industry is struggling to implement, and what are the futuristic developments that are going to further become a challenge for the industry. And with that challenge comes an opportunity for you people. So many of you who are students present over here, that gives you, once you are able to understand the challenges, train yourself to meet those challenges, then it is all opportunity for you.
Excuse me, please mute your microphones.
Okay, I start with recent advances. Recent, I'll say, starting from a few years ago, and then coming to what has happened lately and what industry is what is under implementation industry presently, and then I will talk of futuristic. Now, if you look into ICH website, and keenly, I think all of you should be visiting from time to time the ICH guide website, www.ich.org. Because I tell you, the industry is highly regulated. And until unless you go and study what is happening at the forum of US FDA, EMEA, Japanese authorities, and then all other country authorities, and then ICH and WHO, you cannot really understand what is happening at regulatory, what are the challenges for before industry that are emerging on daily basis, and new challenges emerging on daily basis. So in 2003, ICH issued its quality vision. Now this was away from harmonization activity which happened earlier to this, 2003. Earlier emphasis was on harmonization. Then they came out with quality vision. Vision means how we anticipate the quality of pharmaceuticals in future. So with an intention, what was the intention of this quality vision? The intention was to ensure quality products and reliable supply to patients. Now I take any drug, you might be taking any drug. How to assure that it is a quality product and there is a continuous and reliable supply of this quality product to the patient. Now, harmonization was technical requirements to be harmonized. Now, question comes over here is ensuring a reliable supply of quality products to patient all through. It should not happen that some of the patients are dependent upon a certain drug, certain product. They are on that therapy for lifelong, like I am on some of the anti-cholesterol drug, which I am taking for almost now since 1995. Now, I had chosen a brand after checking a comparative evaluation of, because I am a person working in impurity profiling, I have access to all the equipment. So what I did was I took different brands subject to HPLC studies by a pharmacopoeia related substance method, and the one product which had the lowest amount of impurities I selected from the market, and it happened to be the lowest cost also in the market. So I selected that product and started, despite that physician has written something else, some other brand, I went for selection of my brand which had minimum of impurities. Now the question comes over here is this quality VN was that continuous supply and the reliable supply of quality product. Now suppose I am already fixed on one brand, I am taking that brand continuously for years together, and suddenly there is no supply of that brand, and there's a stoppage of supply due to any reason. It means I have lost that quality product, which I was taking for years together, till it is the supply is restored. So how to make this quality product and reliable supply to patients was the ICH issued this quality VN in 2003. Now, what was ICH quality VN further? To develop a harmonized pharmaceutical quality system applicable across the life cycle of the product, emphasizing an integrated approach to quality risk management and science. Now, a lot is built into this. So it says to develop a harmonized pharmaceutical quality system. Now, again, there is a harmonization, of course, that they want all the industry, total industry in the world to have a common pharmaceutical quality system applicable across the life cycle of the product till the product is in the market. It is applicable. That quality system applies on that life cycle, total complete life cycle of the product, starting from, let us say, R&D to marketing and being in market for years together. Emphasizing an integrated approach to quality risk management and science. What they say is that the pharmaceutical quality system has to be based on quality risk management and good science. Now, what is happening today? In our industry, in generic industry, all the products are manufactured by hit and trial. All formulations which are brought to market are by hit and trial. They are not based on good science. The reason is science requires understanding of physicochemical property of each material that goes into the product. Complete, and the influence and the long-term influence of that property of the material on the life cycle of the, during the life cycle of the product, on the pharmaceutical quality of that product. And it says quality risk management. Now I will explain you what exactly is quality risk management in subsequent slides. So this is something they were trying to emphasize, that whatever product comes into the market has to be based on good science, which is missing in the larger part of the world, except innovative industry in the Western world. And a built-in quality risk management. Now the question is, there should not be any risk to the quality of the product throughout its life cycle. Such management has to be done by building a pharmaceutical quality system within a organization in industry. Now you can stop me and ask any question if something is not very clear to you anytime. So to enable the realization of this vision, ICH issued guidelines concept paper QA to Q12 between 2005 to 2014. Now already, because it is they were issued between 2000 to 2014, 2005 to 2014, they are already under implementation. And of course, in industry like generic industry is trying to struggle to establish facilities, have manpower which can really comply to QA to Q12 guidelines.
Excuse me, the technical team is required to check the noises. We are receiving quite a disturbances. Please.
Mr. Praveen,
I will be muting him. I will be muting him. Our technical.
So if I have written over here for your convenience because the slides may be made available to you in future also. These guidelines are currently in focus and brought a sea change in the working of industry, particularly with respect to implementation of QBD and risk management concepts into product life cycle. Now this was a phase that people who are expert in QBD were the most sought after people in industry. They used to go, I know of my colleague, Professor Bhinder Singh Bhoop from Punjab University, who I requested him to, because he was very good in statistics, quality by design requires design of experiments. I asked him to really develop this field, and he was one time the sought after person in industry to talk on QBD concepts and design of experiments. And of course, the risk management. Now they have industry struggling to have people trained in risk management so that this can be implemented. So question comes over here is regulatory come out with the concepts. Until unless we have a constant follow-up, we cannot prepare our students to stand and really have a confidence to work in industry. Then industry has to retrain them into their requirements. Now, this is a gap which I see between academia and industry. So at NIPER, one thing which we implemented was that the teaching should be all regulatory oriented. And with that emphasis that we brought into NIPER, our students were valued students in that manner. So this all institutions, if although I know you are bound by PCI regulations and all those things, but Still, there is a lot of scope to the teachers when he stands to talk to the class. And if he is teaching with respect to what are the regulatory requirements, why conceptually explains why I'm teaching you this and what is the importance of this to industry, then that person can really know before joining the industry what are the challenges I am going to face and what are the challenges industry is facing. Now, look into this ICH guideline, International Conference on Harmonization of Technical Requirements for Registration of Pharmaceutical Use. Now, the name is International Council for Harmonization. They had come out with this Pharmaceutical Development Q8 guideline, which went into 2 revisions on August 2009. Now, it introduced and outlined the principle of quality by design, the guideline Q8. Our guideline Q9 was on quality risk management, and it was introduced on 9th November 2005. And this introduced the concept of quality risk management, outlining the principle, process, methodology, and integration of these with industry and regulatory operations. They defined what is quality risk management, how it is to be implemented, and how it can be integrated with industry and regulatory operations. That Q10, which was brought on 4th June 2008, Pharmaceutical Quality System, where they said that when you are doing pharmaceutical development, going for technology transfer for scale up, you do commercial manufacturing, and then until there is a product discontinuation, there has to be a quality system developed at all levels, starting from R&D to technology transfer to commercial manufacturing till the product life cycle. So there has to be a proper GMP. There should be management which will be made responsible and define their responsibilities. Then PQS element means product quality system, pharmaceutical quality system, process performance and product quality monitoring system, corrective action, preventive action system, change management system, management review system has to be implemented in industry, and new teams have to be created for this purpose, so that there is a knowledge management and also quality risk management. So these PQS elements, that is pharmaceutical quality system, will enable knowledge management, that is science-based approach, and then quality risk management, so that wrong doesn't happen subsequently at any stage of life cycle of the product. Then came the Q11, development and manufacture of drug substances, chemical entities, and biotechnology, biological entities. So now, having this quality system, how to ensure that your production is going in a quality manner. So this guideline provides clarifications on the principle and concept described in ICH guidelines on pharmaceutical development, quality risk management, and pharmaceutical quality system as they pertain to development and manufacture of drug substance. So how these all three are to be implemented further. So guideline Q11 was brought into. As when it comes to the development and manufacture of drug substance, whether it's a small molecule or a large biotechnological product. It's a general concept that had been given. Now, then there was a quality Q12 concept paper, which was brought out, final concept paper Q12, where the concept paper is issued before the effort is taken to develop as a guideline. So concept paper is what we really want to include in the guideline. So type of harmonization action proposed, statements of the perceived problem. What is the problem which you are like to solve, and what kind of harmonization it will lead to? So Q12 was technical and regulatory consideration of pharmaceutical product life cycle management. So now they are talking of, earlier they talked of quality system development, and now they are talking of product life cycle management. So whole through the life cycle, the quality should remain, not only in production, but when post-production, when the product is in the market, throughout the life cycle, you have a assurance of quality. Now, basically, Q12 focuses on gaps in implementation of Q8, Q11 guidelines, and also talks about post-approval operation flexibility. What was a gap? That this was more related to R&D and production. And then if it is beyond that production and post-approval, once you have got the registration, so how to have some, because post-approval changes are bound to happen. You cannot have a product, what you were manufacturing 20 years ago. Maybe if Crocin is manufactured by a company, there will be always some post-approval changes that are happening. So how much of the operational flexibility is to be allowed to industry for making those changes? So this is what is a concept paper is being brought out, and the guideline is being worked out to allow for operational flexibility. So now let me give you a brief on the concept of quality by design, regulatory flexibility, and quality risk management, so that you can understand how total industry is changing itself under the domain requirement from ICH and other regulatory bodies. First is quality by design. Now, this is applicable to both pharmaceutical development and production. So it says this is a, I will say, significant shift in the paradigm, total thought process, the way industry used to work. It is that what we had been doing until now we are doing in industry, that we do quality by testing. We establish quality by testing. We have produced a product, send it to the laboratory for testing. We take out of the batch a few samples of tablets or capsule or injections, send it to the laboratory. If they are found to be okay, then we release the batch in the market. So we do quality, which is controlled by inspection and testing of finished product. So whole concept that changed was from quality by testing going to quality by design. So you build quality into finished product, no reliance on end testing, then you don't need to do end testing. Now try to think over the whole practice of industry till today is based on end product testing. And here they are saying you build quality and don't test the quality. I published a paper, QBD in education, QBDE. I floated the concept of QBDE, quality by design in education. Now, what we are doing now, we are teaching and training the students, whether in the lab or in a classroom. Midway, earlier it was annual examination, or we now have midterm examinations or semester examinations. As teachers, we are very happy that we prepare a question paper, ask students to sit down, attempt that question paper, collect those answer books, evaluate those answer books, display a sheet on the notice board of the result for each student. And a teacher says, my job is done. We have evaluated the students and the student has got a ranking from, let us say, 90 to 30% or 40% or 50%. Now the question comes over here is, try to understand, this is quality by testing. We are testing the quality of student by subjecting the student to examination. Now we are testing the finished product. Now also, I think if you have seen the news from PCI, now we have another president of Pharmacy Council, Dr. Yol. He had put news item in papers where he has said that we will have an exit exam for B.Pharm students or D.Pharm students. Exit exam means they are again going to have, because the students coming from different institutions, they are going to have another examination to declare them to be worthy of joining industry or the job. So what is happening that we are testing regularly each semester, multiple tests, then we are evaluating them at the exit exam, and then we declare worthy of that they are worthy to join a job. as desired by Pharmacy Council of India. Question comes over here is, if I say I have a class of 60 and all my 60 students should get 95%, then I will be building the quality. I'm not testing the quality. I'm not very happy that my students of the class have scored from 90 to 30% to 90%. My effort would be to build the students in a manner that each student gets 95%, whether he has come from any background. I tell you, I have practiced this in my department. I can say, yes, I could successfully implement it because the number of students in master's class used to be very small. Although it may not be applicable to class of 60, but what extra effort is being done by the institution to build quality, and all students have the same quality and they pass out with the same marks. Do we do that? Now, if I ask you today, suppose I am become a president of PCI, I want all students coming from all institutions. I am not going to have any exit exam. All students should be a national test, should have the same rank. Now you will say how it is possible. We people, you are taking away our freedom. We are very happy with examining the students are putting into 30% to 90%. You are asking every student has to be built to 90%. How it is possible? Think of the situation and challenge before the industry that you are not to test. You have to build quality into finished product, no reliance on end testing. See the challenge to the industry. Same. Every tablet has to have the most optimal quality. And that tablet, till it is in market, till the life cycle is going on, till it is consumed, it has to retain that quality. Think over, please pay emphasis on this. What is industry challenge before them? Challenge before the industry today. Why should not we have the same challenge in academia? Do we train our students to build quality? We are just training a student to test quality. Even we are subjecting students to evaluation of their quality by examinations. So when I say this is a significant shift in paradigm, today we do some batch analysis. They say, no, every tablet has to have a proper quality, not a some analysis of a few samples and testing of a few samples. This is a huge shift in paradigm. So what are elements of QBD? Target the product profile. So question comes over here is, target the product profile, what you want out of a product. Let us say what you want out of a student. Determine critical quality attributes which can improve the quality. And these are minimum standards, specifications. Link raw material attributes and process parameters, CQAs, and perform risk assessment. Develop a design space through what I call design of experiments. Design and implement a control strategy that all products are going to have, all dosage form units are going to have the same quality. Manage product life cycle, including continuous improvement and product process monitoring. This is the workflow for quality by design, which was a new concept brought in. That was a quality vision of ICH. Now, may I ask you one, maybe we can discuss this. Do you feel from where this quality risk management and other things have come into picture, this risk management? Basically from space and aircraft industry. Can you think over a situation where I had to develop an aircraft Boeing, let us say Boeing company says that we are to develop or the other companies, they have to develop an aircraft. They tell you, all you are listening and attending to the seminar of SNJB today, come on, we give you a free ride in an aircraft, which we have not subjected to risk assessment. And pray for it. If the aircraft goes into air and lands successfully, it is your luck. We are under the development of that aircraft and we are going to use you as a pawns, experimental animals, to see whether that aircraft really will be successful in its flight, first flight. Can we do that? Let us say if it is a wide-bodied aircraft which has 300 passengers to carry from one place to another. Can we afford to kill 300, 350 passengers in its first flight? No. And you think over that drugs are used for patients, not a healthy person. Can we take a risk of sending a bad quality? Today, your tablet machine produces 50,000 tablets, 5 lakh tablets per hour. With that rate of production, can we take a risk of bad quality and how many patients will be affected in the market? So all those concepts which were based on the best of the world industry, which were non-pharmaceutical industry, have been brought down into the pharmaceutical manufacturing, taking the risk in equal risk to the patient, because the patient is already under low immunity. You have a headache today, suppose, let us, you are sitting and listening the lecture, but you have a headache. Your morale already is down. You are not able to pay attention to what I am speaking. With that, if I give you a bad quality product, so it's a risk. And there can be a disease, a person is vomiting continuously and the drug is of low quality, it doesn't show its effectiveness, and you get into a very sad situation. So that's why all these concepts up to life cycle management, product profile to life cycle management, that has been brought under the umbrella of QBD, quality by design. Build quality, don't test quality. So I will give a simplistic explanation of QBD. The concept is that it is divided into 3: knowledge space, design space, and control spaces. Build maximum knowledge about what activity you have undertaken. You want to build a tablet of any drug. What are the properties of drug? What are the properties of excipient when they mix with each other? How they behave? Maximum knowledge what you can take about, and what is the individual property of each individual excipient. So once you have a knowledge space, based on the factors that are very critical with respect to critical quality attributes which you have to select out, lay down the critical quality attributes. So what is that in the knowledge space you feel is very critical that can lead to a bad quality? This will be based on the knowledge space. Now, this is a good scientific principle. Within that, you identify the factors which are going to be important. Let us say, I want to achieve this X dissolution. What are the whole parameters or the product issues or the material issues which can lead to whether then one X is combined with Y in the formulation can lead to a disturbance in the normal dissolution profile. List on all those factors which can lead to a bad dissolution profile or unwanted dissolution profile. Look into the relationship. If I mix this with this, I mix this with this, what is going to change, going to happen? Run design of experiments within the design space. And based on this, which are the real true critical factors, they need to be controlled so that there is no variation that can happen in the due course of action. So you focus on controlling those processes, those parameters of raw materials, which can lead to bad quality or bad dissolution. And if you have control spaces over there, then you are more or less sure of producing a good quality, because you have controlled the quality before manufacture of the final packaged product. If there are holes in the knowledge space, you have not really initially established your knowledge fully. And if they fall on the control spaces, these holes fall on the control spaces, you cannot have your quality controlled. Now, let me give you an example. I have been coming to SNJB, okay, for these conferences, regular conferences. All the teachers used to be there in the front row, and the hall used to be packed with students. And if there is no control system, once the lecture is going on, half of the students will be out of the hall. Ye bore kar raha hai yaar, ye to professor bore kar raha hai. The speaker is just boring us. Let us go out. Maybe it was bored up to a certain point, and maybe it was very interesting later. From that, if the student leaves the hall, maybe there was something very, very interesting that was coming in subsequent to that, then the person misses that. So I used to have a control space. I think everybody, Dr. Pasni Vivekananda, all of you remember that I used to say that once I start, the doors need to be closed of the hall.
Yes.
No people can enter but cannot leave, except special request for bio break if somebody needs it. So the question is, you are doing an effort, you are preparing slides, you don't know how many hours one person has to do preparation to come before you people. And if there is no listener and they are not interested listener, my whole effort is wasted. So you have to do control spaces where you feel it can badly affect the quality. So once you have control on your quality buildup, then it means the final product has to have that good quality. So how we have knowledge base? Huge. It can be prior knowledge based on experience. That's why you hire an experienced person in the company. Pharmaceutical development studies, mechanism of action of drugs, structure function relationship, technology transfer activity, process validation studies, manufacturing experience, innovation, continual improvement, change management activities, stability reports, product quality reviews, annual product reviews, complaint reports, adverse event reports, deviation reports, recall information, technical investigation and or CAPA reports, supplier and contractors, product history and or manufacturing history, ongoing manufacturing process information, trends of what is happening in the batch to batch. All this gives you knowledge of some kind. Now I tell you 100% that in our BPharm syllabus, maybe 20-30% of these things are covered. Rest, our students don't know what is happening in the industry. Where we are teaching them, there is a product quality review, annual product reviews. Where are the complaint reports? Where are adverse event reports, deviation reports, recall information? What do we have these all things in the syllabus? So we are sending a student with a 30% knowledge to the industry. We are not building enough knowledge space to prepare students for industry or any other job which we want the student to take after BPharm. So what is design space? The multi-dimensional combination interaction of input variables, they are material attributes and process parameters that have been demonstrated to provide assurance of quality. Design space is proposed by the applicant and is subject to regulatory assessment and approval. So what you do is you identify the factors which can lead to bad quality product. So it's a multi-dimensional combination and interaction of input variables, material attributes and process parameters that can have been demonstrated to provide assurance of quality. So you are applying design experiments that you can demonstrate to provide assurance of quality. So you have to propose how you're going to do is because you know what are the factors that are going to influence. You will know the materials which you are going to handle, which you are going to put into your product. And then you send to regulatory assessment and approval that this is what I am proposing, design or experiment. And please guide me if I'm okay, so that I do all the effort and then tomorrow regulatory says, no, you should have done like this. You should have built the design space like this. So maybe I can give you an example here. Process variables and quality attributes for the fluid bed drying operation. Now I'm talking only of fluid bed drying operation. There can be hundreds of operations in the whole process of manufacture of a tablet. Maybe 10, 5, 10, 15, 20, or whatever it is. In a more complex product, there will be more number. Now for just fluidized bed drying operation, a successful fluidized bed drying operation, as a knowledge space, we list of what are the process variables. They can be drying parameters or input material attributes. What is the water content? How much time? That will decide the time of the fluidized bed drying operation. Granule particle size distribution. Then inlet air temperature, inlet air humidity, air flow rate, fill level in the whole container where you are doing the drying. Filter soak cycle, heating rate, cooling rate. So all these things are the drying parameters which are going to, and the input material attributes which are going to affect the quality of a successful fluid bed drying operation. What are the quality attributes you are looking forward to? For a dried granule, what is going to be the final particle size distribution, including fines? What is going to be the water content? Degradation, whether any degradation happens during the drying operation due to high temperature that is maintained during this thing. And what is the final output you are asking from a tablet is the disintegration, dissolution, weight uniformity, and content uniformity. These are the quality attribute of granule in the intermediate stage, and this is for the final tablet. Now, if you control all these things and see if I'm changing the air, that air temperature, and there is a change in water content, how they are correlated to each other. So I have to do some design or experiment to correlate these parameters and these parameters among others, so that, and then what I have fixed as a specification for particle size distribution, so that my all functionality from tablet is achieved. So this is knowledge space that I build up. What are the parameters that can affect the quality of one operation? It may be 10 operations. It may be a milling. It may be a granulation. It may be punching. So all steps that are involved, mixing, will have a separate set of variables. And that is what is a part of the knowledge space. Now let us say effect of inlet temperature, airflow on degradation and generation of fines, which are the two important parameters they have taken: particle size distribution and degradation product generation. Now, inlet temperature as increases, it leads to more of the degradation, and the airflow has no much influence on the degradation, and it decreases. So increase in inlet, but temperature is a major effect. Fines are generated more with the airflow and not with the increase in temperature. Now, question comes over here, what inlet temperature airflow I have to set that there are a minimum generation of fine and the minimum of degradation. So what I have to do is I have to correlate them with each other, and I put inlet temperature and degradation and fines together. And this is inlet temperature with the airflow. I find out a region. This is prohibited region, red ones. I come with the yellow region where I can do some experimentation, and I will get a good quality of my granules, and I will get a minimum of the degradation if I work in the yellow region. So what I have done is I have looked into intercorrelation of the inlet temperature and airflow in a manner to get a limit of degradation and limit of fines. So I found a region where if I operate within this region, I will be able to meet the quality. So red doesn't meet the quality requirements based on my study in the previous slide, this one. There's a red portion over here, yellow over here, yellow over here, this. So combine the two parameters, it comes out of this region. So this is what is multi-dimensional combination and interaction of input variable, that is material attribute and the process parameters. This is what is design space. So here, if I am working with this, I am having assurance of quality. I don't need to test the final product because I am working in the design space where I know my product cannot go wrong. So like we were doing, this can be extended, qualified design can be extended to any of the, I will say, operations. Our target was, like this is one of our papers that we publish, that we are doing stress testing. Now we have the temperature and acid stressor. So normality of acid versus temperature. We wanted a pure degradation product, DP3. So drug was converted to DP3. I wanted pure DP3. So I manipulated the temperature and acid in a manner that it gave me a region where I could get a pure DP3 100%. So through the study and implementation, design or experimentation, I was able to very easily get to a condition, otherwise the drug was remaining in that solution. So an optimum like 0.2 acid and temperature of around 60-70 degrees, 70 degree temperature and 0.2% acid. This was 0.1% acid and temperature up to 80 degrees. But if I increase the temperature acidity to 0.2 normal, I get a very fast condition where I could get a free DP3 and I could sell this as a standard. So it can be applicable to your formulation development.
Excuse me, sir.
Hanji, yes.
Your voice was lost for a moment.
Okay. Now it's okay. You could
Yeah,
understand the previous
yeah.
slide. My
Yes,
previous
yes.
slide. This previous slide discussion was
Over. Was audible.
okay, done. Now see over here how we build up the control space. Now we have dispensing, blending, sifting, roller compaction, tablet compression, and pan coating. Now one way is that I do the product which is coming out of pan and do the testing. The other way is I raw material and API dispensing I do based on the specification of the product. I do NIR monitoring, blend uniformity when I'm doing the blending operation. I do laser diffraction particle size analysis when the roller compaction is going on. And then I have NIR spectroscopy at line identity, assay, API to excipient ratio, all done during the tablet compression. And now when the finished product testing, I'm assured because I've done the testing all through, which I don't do now. Currently, we don't do it. But if we are doing all these testings on the process of manufacture, then your product is bound to be good quality. That's why you see why we have now midterm examinations and assignments in our systems. That students should be, and we people are not giving a serious thought on this, that a student in the first assignment has got a very varied marks. Now, what I should additionally do that all the students come closer to each other in their marks, whether at intermediate stage 1, intermediate stage 2, or intermediate stage 3, so that all the students get the same marks. So this is what is the total new concept that we should have all the, and there's no sample taken over here. It is online monitoring with the new tools, which we call as process analytical technology tools. So what is the process analysis? Lab-based, there's a human sampling. You go to the, suppose the blending operation is going on, you go to the tank, you take the sample out, send to the lab. There's a queue time for instrument. There is a sample preparation involved. There's a slow response. So what process analytical technology says, at line, I've taken the slide from a USP presenter because this I found was a very interesting to explain at line and online concept and explain the concept of process analytical technologies. There's a human sampling at line, but the machine is very near to the tank. It is not in the separate laboratory where I have to send the samples where all the samples are converging. This machine is dedicated, like UV HPLC is dedicatedly for this blending operation. You have human sampling, sample preparation, but rapid response because the machine is dedicated. Online is that machine is directly attached over to the tank. Sample interface directly to instrument, immediate response. There's no human sampling involved over here. The machine itself is tucked to that equipment. Process analytical technologies, PAT technologies, which is now non-destructive testing and visualization. Lot of techniques have come into picture. FTNIR, ATR, FT-Raman, terahertz pulse spectroscopy, quadrupole resonance spectroscopy, frequency modulation spectroscopy, X-ray diffraction, optical coherence tomography, time of flight secondary ion mass spectrometry. I have a full lecture. And we used to discuss in NIPER, now that I have superannuated, but in my second semester of, we used to discuss all these techniques in detail because these are the futuristic techniques. Our students should know what is coming for their future. Now, let me give you a glimpse of FTNI spectroscopy for control space management. It's just one technique I'm talking, not all techniques. Dedicated NIR analyzer for real-time pharmaceutical blending. Earlier, this was the size of the window that we could put in the tank and insert on the wall of the tank. Now they are coming with very small. One is that throws the light and gets the light thrown back to reflective phenomena. The system can monitor components such as moisture to warn when these components are trending outside the acceptable range. How it is done? That let us say this is what is powder mixing happening. Blending is happening. So you put multiple NIR probes. Now what we do is we put a sampler, take a sample from the bottom. So there is a sample collector tube. It takes a sample from the bottom, then middle size and the upper portion. Then we send it to the lab. No, nothing like that. We have optical fiber multiplexer, which is the FTNIR spectrometer. And you have multiple probes which go and the tank is there. We can say 12 holes are created in the tank and we put the NIR systems. And it records when the, let us say the drug has been added for blending and into the excipient mix, when the whole of the drug at all places, all these instruments or the probes, optical fiber probe, they say the drug is equally reached at all places, the instrument automatically, the blending operation automatically stops. So now it's a real-time stoppage, not a range that blending for 10 to 20 minutes. Anything can go wrong between 10 to 20 minutes. Here, when the drug red has got mixed equally to the rest of the excipient mix, automatically the machine will stop. So there's no reason that there is going to be a bad quality, that some lot has some part of this matrix has a less amount of drug, other part has a higher amount of drug. It doesn't happen. So this is a radical change happening, technological change happening, along with the regulatory paradigm. That is a shift in paradigm that is happening. That technology is coming to the rescue to implement the whole of the concept of QBT and quality risk management. Now, this also means we are doing quality risk management. The risk of uneven distribution of drug in the whole of the mass of the excipients reduces if we have a multiple place monitoring going on within the blending. So there are XDS, near infrared technology, the force process analytics, micro bundle multiplexer provides the next generation of process analytics for real-time process analysis. Non-destructive, accurate measurements are performed directly in the process line, blender, dryer, or reactor. Now, this can be a probe type that can be inserted into the powder, and it will give you the exact readings. Now, I will tell you, all those who are present today, go to Indian Pharmaceutical Congresses whenever there's a chance out of the COVID era. Whenever you go to that, there are a lot of instruments on sale and display, which are the latest spectroscopic tools for process analytical technology requirements. Try to go to spectroscopy stalls and look into what is available. FT-Raman, FTNIR, FTIR, ATR, FTIR, and all those technologies that are being displayed there. You should take specific interest to know about this. This is fully integrated online pharmaceutical tablet characterization pad tool. So it incorporates near infrared transmission spectrum, providing non-destructive simultaneous quantitation of multiple components such as actives and moisture, and physical tablet test of weight, thickness, diameter, and hardness. Just you have to have a conveyor belt fixed to this. Every tablet will be tested for multiple component and active moisture and physical test, weight, thickness, diameter, hardness. Not that you are sampling tablets, but every tablet is counted, means evaluated for quality. This is the paradigm change happening for the sake of patient as a fallout of ICH requirements. Now, here I want to give you the concept of regulatory flexibility. Very interesting concept. Now, I have shown you the design space, previous slides. I am going a little back. This one, this is a design space. Okay. Now, regulatory flexibility, what flexibility are they? They don't say that, no, you have higher degradation products or higher fines. No. You can be flexible from within this yellow space of airflow and temperature. Temperature you can vary from, let us say this is 10 degree to 20 degree to 30 degree. And airflow can be, let us say 20 ml per minute, 40 ml, 60, 80 ml per minute. So you can make a change within this, not going in the red region, but within the yellow, staying within the yellow region, you can make some change. This is what we call as a post-approval changes. So design spaces provide the applicant is subject to regulatory assessment and approval. So this is for post-approval change. So once you have got an approval from manufacturer, then you can really go ahead and do this. So QPD and allowance for regulatory flexibility. Per definition, quality attributes of finished product are achieved when operating within the design space. As a consequence, any process modification within design space should be acceptable without any further regulatory approval. Once you propose a design space, regulatory approval is granted to you to work within that design space. Now you can bring any innovation within that design space. Now, till now, if we go to industry, there is a very strong SOP, STP mechanism. SOPs are there. They say change control mechanism. Why have fixed the, let us say, range of operation of blender from 15 to 20 minutes? You cannot work at 40 minutes or 21 minutes. And like I, as my student who is taught by Dr. Saranjit Singh in Naipur, and this student has a very good idea. I can improve the functioning of industry if I tell them that through my knowledge that if you're doing this, this bright idea can help you a lot. The industry says no. We have been manufacturing the product for last 20 years. We cannot in 21st year implement your idea. So what happens is innovation is killed. Your idea is not taken forward. So what we are doing to change control mechanism, that no change, all control should be there. This is regulatory flexibility. Within the design space, you can bring any innovation. So as a concept, any process modification with innovative idea within design space should be acceptable without any further regulatory approval. This is the regulatory is giving you flexibility that you can bring any innovation to your operation within the design space. It's not flexibility in a high level of degradation products. No. So people sometimes don't understand the whole concept of regulatory flexibility. So here design space is this one. If we go to move from A to B, inside design space, no prior approval necessary. B to C, if I make a change, no prior approval necessary. But if I go out of design space, C to D, I need approval necessary. So within design space, you are free to do any innovation. So they are encouraging innovation within the design space. Now, just giving you something ideas about quality risk management concept. Now, what it requires is quality risk management, identification and control of all those elements that can lead to a low quality product. So try to look into all those elements given through QBD, of course, through QBD, Q8 guideline, Q9, Q10. So Q8 to Q10, we have come reached till now. Identification control of all those elements that can lead to a low quality product. Now, in that respect, quality risk management is a preemptive exercise. Beforehand, you should be knowing what are the possible risks. This is what aircraft industry does. I think you people maybe are not aware. When you have to understand, if you have traveled by air, sometimes there is a turbulence. They will say, get into your seat belts and don't get out from your seats because we are facing turbulence. Fine. Whenever I am, sometimes the turbulence is very huge that you suddenly have the aircraft comes down. So sitting in the aircraft, you feel like that how the aircraft doesn't break away with such a turbulence. Okay, how it remains stable and there's no risk on life. Maybe very huge turbulence can lead to some injuries. There are reported injuries due to turbulence, but you don't lose your life because the aircraft has broken away due to turbulence. Do you know what they do? How they manage the risk to the persons inside, to the passengers inside? They have created air turbines in which they suspend a prototype of aircraft which they are developing to look into the aerodynamics. How the airflow will happen when the aircraft is cutting the air, how the airflow will happen, that is called aerodynamics. So they really develop the aircraft design based on aerodynamics, which they verify through a air turbine tunnel in which they suspend an aircraft prototype. Now what Boeing has done, imagine a situation. They have created a air turbine tunnel where actual aircraft can be put inside to further go for quality risk management. Earlier it was prototype. Now 350-seater mega aircraft can be put into a tunnel, suspended in a tunnel, and then you think of how much pumps will be there, which will be really pushing the air to such a big aircraft through testing whether that aircraft can be made more safer in the case of any turbulence, severe turbulence. They cannot take a risk of losing the lives of 350 passengers and do experimentation with 350 passengers seated inside the aircraft. So it is all a preemptive exercise. Before an aircraft flies, everything has to be perfect. In other way, quality risk management is an advancement over the and above RC and Kappa, which is root cause analysis and corrective and preventive action. They say what industry at this moment is doing, it is limited to RC and Kappa. Root cause analysis, if something wrong has happened, quality is not coming proper in the finished product testing, do the root cause analysis, take corrective action and then preventive action. But quality risk management is a totally different concept. It's directly focused on preventive action instead of root cause analysis. Why something wrong should happen? And then you do root cause analysis. What is the reason for the out of 10 results of out of specification result? Why not you have focused on preventive action, that it doesn't happen at all? So what they have given is the whole chart of risk assessment, risk identification, risk analysis, risk evaluation, risk control, risk reduction, risk acceptance, how much of the risk we can accept. You try your best risk reduction, but still there will be something which is out of your control, how much risk can be accepted. Then output result of the quality risk management process, you review that risk and review events that have happened over the period and then decide on. In between, you need to have risk communication conveyed to all your employees and risk management tools you have to be implementing. In risk evaluation, say it is all risk reduction is not acceptable, it goes back to risk analysis. So if review events that if it is not good, whether you can accept that risk or not in the review events. So all this is what ICH guideline Q9 gives in detail. So they have given definitions because they are very related terms: risk assessment, identification, analysis, evaluation, control, reduction, acceptance, communication. So they have given definitions to all. I'm not going to detail of this at this time. You have to sit down and now just one example of relevance of quality risk management. There was a plant, I was told this story that there was ascorbic acid production going in a plant. Okay. And on one day morning, every day the sample was taken, sent to the lab for testing. There was no QBT implementation. It was a routine ascorbic acid production. Sample was sent in the nighttime. The production goes on the shop floor. Morning, the samples are collected and sent to the lab. The analyst does the analysis, reports the result is okay, and then further processing is done of that. Packaging is done. What happened that one day, just not see the slides, let me explain you what happened. One day, when the analysis was done by the analyst in the morning, or he or she reported the result that ascorbic acid is 0. Suddenly the panic was created all through in the industry. Where ascorbic acid has gone. So it may be 5,000 liter batch was there in the blender and the production was going on, and suddenly that 0 ascorbic acid. Now this is the problem you face in industry at times. Now in this situation, what happens is always the question mark comes to the analyst. Now this is what I do teach my students, that there will be a tug of war between production people and the analyst people, or the formulation people or the analysts, because whole of the analysis is done by the pharmaceutical analysis people in industry. No one else can do the analysis. So you are going to face the tug of war. Whenever you feel that you are reporting a bad result, the firing is going to come on you, that you are wrong. Your analysis, you don't know how to do the analysis. Same thing happened. Previous 100 batches, there was 100% ascorbic acid or 99% of specification, whatever was laid down, it was meeting. Suddenly one day you say it's a 0 ascorbic acid. How it can happen? So always a doubt comes on the analytical result. And maybe this analyst was smart enough. The people from quality assurance ran to him or her. He or she said, okay, fine. You wait, but bring me a sample of the last production, yesterday's product, which I declared as 99%. And also a reference standard. And today's sample. Let me do the HPLC run before your eyes of all the three. He or she was very smart. When the analysis was done, yesterday's sample was again the same result was produced. Reference standard was exact as it was wanted to be done, as it should be. And today's sample was 0%. So this smart analyst proved that something wrong has not happened with the analysis he or she has done. It was the analysis, it was something that has gone in the production. Because night shift, there was some production error. Production had gone and morning the sample was collected. and delivered to the lab. Or sample was collected at night time, delivered to the lab in the morning in quality control. My question comes over here is the whole manager of on shop floor was invited because he has done the night shift. He has gone home. He was immediately asked to come and that ascorbic acid is 0. Now, every time when the shop floor production is going on, they prepare a batch manufacturing record, BMR, we call it as BMR. So BMR was called, everything was same as the previous day. All values of control of temperature, mixing speed, everything was same. No deviation was recorded. So where then ascorbic acid has gone? 5,000 liter tank. And people can lose jobs, I tell you, if such a loss happens to the company management. So nothing could be found out, anything wrong. All the shop floor people were called. Anything change that you have done? No, sir. Everything is recorded in BMR. Everything was perfectly done as was done in the previous batches. We are trained people. We are on the job for so many years. So what we call as a root cause analysis was coming to a dead end. 2-3 days went into this without quality assurance people being after all the production people and everything, that nothing is succeeding. Again, it was dead end situation. Suddenly, the same team which was doing production, one of the members comes to the manager's room, who was in a very bad state of situation because the management was pushing the person find out what wrong has happened. Says, I now remember something which may have some correlation, I don't know. They said, what? He said, sir, the probe which are there in the mixing tank, I could see that temperature was maintained at 25 or 30 degrees, whatever was the set temperature. But the controller was okay, but display started giving some fluctuation. There is always a temperature controller and temperature display probe. And temperature display probe was giving the problem. So I went to the store and found out there was another probe lying over there. I brought it and installed into the tank and everything was fine. So BMR says that the temperature was maintained at 25 or 30, whatever was the specified temperature. Now this manager says, bring both probes to me. When he compared, what was found that the initial probe that was installed in the tank was of platinum. And what this person found in the store was of copper. Now, if you are teaching through physical pharmacy by Martin, in the chapter on kinetics, it says copper can catalyze oxidation of ascorbic acid to dehydroascorbic acid when present as 10 raised to -5 molar concentration by 10,000 times. So what happened was that the total ascorbic acid in the tank, by the time the whole process finished, the ascorbic acid was oxidized to dehydroascorbic acid, and the method of analysis was focused and specific to ascorbic acid and not to dehydroascorbic acid. So there was 0 ascorbic acid because 10 raised to -5 molar concentration can lead to acceleration, catalysis of oxidation by 10,000 times. Now the question comes over here is, because the risk of contact of copper with ascorbic acid was not emphasized in any of the documents, so the change of probe was not controlled. Now, the problem resulted not only in the loss of batch productivity, income, and profits, rather there was additional loss on account of time and energy spent on investigations. So question is, when we say quality risk management, it is risk communication. So there was some lapse in the communication of the risks, that the copper should not be there in any place. Any copper device or tool should not be there in that production area where the manufacturing of ascorbic acid is going on. There was a lapse in risk communication. Now, if I go to the literature, believe me, in the first report that had come out, ascorbic acid oxidation by copper was a paper published in 1930s. This is what is called as a QBD knowledge space. That somehow that knowledge of that ascorbic acid can be catalyzed, oxidation can be catalyzed by copper, was not the person in charge in manufacturing ascorbic acid didn't have the knowledge about it. It was not conveyed to the shop floor people, and this loss all happened. This is risk. When you are putting your everything into risk, either you win or lose. You win otherwise if you have a QBD implementation. If you put yourself to risk due to lack of knowledge, you are bound to lose. And this is why they are talking of fishbone analysis. That incorrect test result can come through which reasons you really work out and have a knowledge of everything. Ishikawa diagram you build up. Identify the potential hazards, whether it is sample related, operator related, laboratory environment related, reagents related, or measuring system related. And this can happen, this is the test result, this is for the analyst, but it can happen in processes also or otherwise. And then they say, your risk ranking system, low, broadly acceptable risk, no further investigation needed. Medium risk is acceptable for the investigation, may be needed in order to reduce the risk. Risk is unacceptable for the investigation, needed to reduce the risk. So accordingly, today the focus on risk evaluation and mitigation strategy, which is entirely different concept from RCA, root cause analysis and kappa. Nothing, it should not allow to happen. Then you are doing root cause analysis. Risk, evaluate the risk and mitigate the risk. So very interesting was ICH-Q9 and XR2. Quality risk management as part of laboratory control and stability study, which captured my attention because of mention of stability study and laboratory control, because I have to teach this. There's only this much part in the whole Q9, ICH guideline Q9, which is applicable to laboratory control and stability studies. So I had to teach this to my students. So this was only 11.7 was the paragraph listed over here. What it talks of auto specification result, which I am listed in the previous slide. This one, incorrect test result. So it says auto specification result is incorrect test result. To identify potential root causes and corrective action during the investigation of auto specification result. And retest period expiration stability studies to evaluate the adequacy of storage and testing intermediate excipient and starting material. This is what is written in the guideline. Now the question is, here also they are talking of root causes and corrective actions. So when I ask my industry friends, what does it mean out of this auto specification? They say simple auto specification has happened, do the root analysis, root cause analysis, and take the corrective action during the investigation of auto specification result. I was not convinced. That is not risk management. So now the question comes over here is, if I say root cause and corrective action during the investigation, it say identify root cause and corrective action, it is root cause analysis and corrective action, preventive action, RC and kappa. But the whole thing changes by the one single word in the guideline, that is potential. Pre-hand, look into what can be the wrong thing what can be a root cause for creation of a out of specification result. Potential preemptive action you have to take. Identify potential root causes. This is coming out of knowledge space. Once you are able to build up knowledge space, then only you can identify potential root causes. See, one word in the whole guideline, word potential, total industry concept changes the whole concept in the industry. One word in the guideline. And we people read it, ignoring what is the word meaning of word potential over here. This is where person like me have to jump in and have to stand before industry to explain then that one word in the guideline changes and it creates spending of crores of rupees, a total conceptual change in the industry. One word in the guideline. I think you will agree with me. Yes, sir. This is what we need to teach the students of what is happening, difference happening in the industry today. So, done with what is the current challenges. The futuristic ICH vision is focused on continuous manufacturing that is likely to be under wide implementation by 2030. So, comes the news in 2015, CPHI, we had the meeting just now finished at Noida. CPHI experts predict continuous manufacturing globally for all pharma products within 10 years. It was at October 1, 2015 news. Means up to 2025, whole of the manufacturing will shift to continuous manufacturing globally. It may not happen, the pace with which it is being done, it may not happen by 2030, maybe something will happen because they are talking about global and they are targeted, they predicted that it will happen in 10 years. It may not happen. And we had a news in 2015 supplement of pharmaceutical technology. PAT paves way for continuous manufacturing. Same September 2015. QBD PAT-based strategies provide the cornerstone to move towards continuous manufacturing, pharmaceutical and biopharmaceutical production. Today we have a batch production, batch manufacturing. They are talking of continuous manufacturing, means non-stop manufacturing. Today we prepared a batch, clean whole of the operators, equipment, everything, surfaces, environment, we clean the room. Here we are talking of a continuous non-stop manufacturing for companies which have global sales, high production sales, which are blockbuster drug, worldwide sales. So this is a pharmacetechnology.com. So this is all technology orientation thing we are talking about, challenges and opportunities now. So continuous manufacturing has a strong impact on drug quality by Lawrence Yu, PhD from US FDA. It is, if we used a time machine to transport a pharmaceutical scientist from the 1960s into the current pharmaceutical production plant of today, it might be surprising to learn that they would already be very familiar with most of the processing and production techniques being used. That is because not much has changed in pharmaceutical production over the last 50 or so years. For decades, most drugs have been manufactured using what is known as batch technology, process whereby the ultimate finished product has been made after many stops and starts in a series of steps. Unfortunately, each break in the process causes inefficiency and delay, as well as increased possibility of defects and error. Today, new and exciting technology, continuous manufacturing, enables much faster production and more reliable products through an uninterrupted process. How much faster is continuous manufacturing? In some cases, manufacturing that takes a month by batch technology might only take a day using continuous manufacturing techniques. That is what is going to happen in future. And this was also April 12, 2016, where the world industry was already starting the process of moving towards continuous manufacturing. And US FDA also realized the importance of it. So traditional manufacturing via batch processes. You have API, there is a wall. The API factory is different. Formulation manufacturing is the different factory. So this applies to all the manufacturers who are doing tableting or any other dosage form development, and the API is sent by truck or by air or whatever it is. So you have chemical intermediates going to API, multiple disconnected batch steps, and multiple disconnected batch steps to produce API to tablets. So this is what has been happening in the traditional manufacturing in the past, disconnected process steps. Now, QBD environment since 2010 is a lean manufacturing QBD environment where process steps and the impact is understood, knowledge space. We know if I do this, what is going to be impact on the next step? If I make a change over here, what is going to affect the next step? And this step, how it is going to further affect. Some properties change over here, how it is going to affect the final product. This is known. Process steps and their impact understood. What is continuous manufacturing beyond 2015? It is seamlessly integrated and well-characterized processes. So seamless integration and well-characterized processes. So what is pharmaceutical manufacturing via continuous processes? Chemical intermediates prior to API formation, API is formed in situ, and then tablets are out. I think this I have discussed earlier in my previous lecture on inspired career, this portion of continuous processes. Now, this is Janssen Presta continuous manufacturing line. Already you don't see any person over here. Now, this is highly dependent, technology dependent, high tech. There's no person seen over here. Lot of fluid mechanics, lot of solid fluid or liquid fluid flow mechanics involved. So you have to have the tablet punching machine, and tablets are going to come out from the API which has been fractured in line. So this is now what has it come down to. One compartment, one or two compartments. Mini factory churns out drug cheaply and at a furious pace. A small fully automated assembly line can produce 4,800 tablets an hour and could make drugs more widely available. So this is what all contained. There are no persons manning the floor shop. So this is what has been developed with the MIT. Novartis and MIT team developed the same type of assembly you can see over here, container you can see over here. This is from a Korean company. This is what was developed with MIT by Novartis. So this is the all chain of events in a one room, closed place. No human beings. And you can see how much technology is involved over here. So the drug plant of the future, manufacturing catches up and hooks up with research informatics. So one person is sitting on the chair and running whole of the plant. This is the future of pharma industry. So that all those errors that a person has gone out, brought a controller or something which is a, let us say, display probe and makes a change, that's all going out of picture. So whole of the operations which are happening in a company are on your screen, and you have control system built in over here. So the futuristic ICH vision is reflected by Q13 and Q14 guidelines under development. So Q3 concept, Q13 concept paper, final concept paper, which was brought out in dated 14 November 2018. So continuous manufacturing of drug substance and drug products. Now it is again an ICH topic. So what are all issues connected with continuous manufacturing? Are CM related, continuous manufacturing related definition and regulatory concepts, key scientific approaches for CM related regulatory expectations. So if the industry adopts and moves forward towards continuous manufacturing, which has been done like Janssen Pharma is doing, Novartis has done with the MIT Manchester Institute of Technology. So what are the issues? What regulatory will accept out of it? Whether regulatory acceptance is going to be or not. And they have come out with the guideline on 27 July 2021 already. continuous manufacturing of drug substance and drug products, Q13. This was, see how fast they are going. In 18, they developed this concept paper, and now already they have brought out the guideline. Now, how many of we are keeping our students aware of development at the monthly basis or yearly basis? So Q14, analytical procedure development and revision of Q2 R1 analytical method validation, again in 2018. What is required? Because you are going to use a lot of NIR, Raman, NMR, MS, which are PAT tools. The scope of revision will include validation principles that cover analytical use of spectroscopy or spectrometry data, NIR, Raman, which are newer techniques, which require multivariate statistical analysis. So now their method validation guideline is going to be revised soon. Q2 guideline is going to be revised soon. So question comes over here is that you have to really follow up what is happening. So to conclude, my friends, these are exciting developments, exciting future ways for those who gain expertise in high-tech areas of QBD, QRM, modern PAT tools, and continuous manufacturing. Now, think over, this is exactly fitting the theme of this conference: research, innovation, and technology, challenges, and what we call as the scope, opportunity. So if you are working in these high-tech areas of QBD, QRM, modern PAT tools, and continuous manufacturing, there is an exciting future due to these exciting developments which will await you. And if we are able to tell our students all this, I think they are going to be sought after persons by industry today. So in NIPER, Dr. Vibha Puri, our MS and PhD NIPER, worked with MIT Novartis team and established a part of, as a postdoc, she was continuous pharmaceutical manufacturing. She set up these facility, which I've shown you. She was part of these facility. This one, which I showed you previously, sorry. This one. So she was part of the establishment of this. Our Shivang Chaudhary from NIPER Ahmedabad has adopted QBD as a QBD expert, your partner in excellence, all-in-one solution for quality by design. He is MS Pharmaceutics PhD and from NIPER Ahmedabad. So for me, everybody, they are part of NIPER, and he's training all the industry team. I have another student, Amrit Podal, who is in university in Austria. They are also specializing in all that, understanding the material properties, so that continuous manufacturing requires knowledge of material properties, excipient material properties, drug properties. So they are working into, and they created a center which specializes in this. So the question is, this person is again, there is a belt system in QBD and lean manufacturing. You have a black belt, white belt, or whatnot. So he is already gone very ahead. And this is what can happen to you also. What you understand, what is futuristic, emergence of futuristic, that you can really expertise, gain that expertise and become an expert. Now he's giving a training to all the scientists, students, technologists, all over the industry on the QBD expert system, which the software he has developed, and he has been awarded even by European Medicine Agency. So Alembic recently put an advertisement in journals, India's first fully automated continuous process plant for high volume commercial API manufacturing, built on modern concepts of flow chemistry, PAT tools, and EBMR concept. So they require MTech, BTech, chemical engineering with 15 to 20 year experience in Gujarat. So the question comes over here is, if these type of high-end technologies, they are going to come, why not pharmacy students take a lead and be a part of it? Because you understand the drug material properties better than anybody else. They are engineers, but material understanding is in your forte. So I believe that my lecture, today's presentation, will stimulate you people, any one of you, really get into these fields, which are emerging fields. So my thanks, be blessed. So these are my current emails, and Jai Hind. Thank you so much.
Thank you, Dr.
So,
Saranjit Singh.
I am done.
Thank you, sir.
Yeah,
Thank
so it was really a great last more than 2 hours. We had a marathon, and to the theme, this theme innovation and all, whatever the great theme what the management for this conference have set up. Befitting great marathon, excellent lecture, his whole experience in 2 hours. He has tried with some very good examples, very good stimulating lecture. It was like a storytelling and very simple way. All of us, because sometimes in any lecture it goes above the head and will not take away message. But here 100% take away message is there, and we will be very much pleased to come back to Dr. Saranjit Singh to learn more and more, and he is still quite active. He is maybe some service, they have government rules, regulation, they have told he is retired, but he is not tired. So he is with us, and he is will be with us, and enriching with the further announcement and mentoring all the students, young minds, and see that the same trend of good work, NIPER in other places, even other places, will get good output from his great learning and experience. So and actually I need not sum up, and because already he has so nicely told, and we have a lot of takeaway message, and we are grateful, thankful, and our great hats up to Dr. Saranjit Singh, and in this stability, QBD and all, of course, Dr. Bhupinder Bhup is also there. He has also, because his QBD means we remember always Dr. Bhupinder Bhup, but both are from there only, originally from Punjab University, but NIPER completed his tenure, and we actually whole India, and we are very proud, the whole world, in the pharma, scientific world, the technology, particularly stability, regulatory means it is Dr. Saranjit Singh, nobody else in India, because all the industries and all the educational institution is Dr. Saranjit Singh, a great expert for government of India, pharmacopoeia, all the everywhere is there for quality assurance, for drug control department and all. It is for all harmonization means all. It is Dr. Saranjit Singh. So our great support and encouragement, and we are very grateful to, and we learned a lot of things today, because many of us, even what is stability, what is analysis, even ABCD we did not know, but he gave basics, and also in such a simple way he made us at least a small masters, and even we are stimulated to do a lot of things are to be done, and Dr. Saranjit Singh is there to give us the path and direction. So once again, on my behalf and on organizing committee behalf, we greatly thank, and we are very grateful to Dr. Saranjit Singh. Now I will hand over to the organizing committee.
Thank you, Dr. Lupa. So nice of you. Thank you.
Thank you. Thank you, Dr. Saranjit Singh.
Thank you.
Thank you, sir. I may confess that we surely require Dr. Saranjit Singh sir every year, because through his lectures he reminds us every time to stick to the quality. Quality cannot be maintained or cannot be achieved only by preaching it, but by following our books, our guidance which we get from successful people like Dr. Saranjit Singh sir. He has clearly related quality, whether it may be drug product, whether it may be a formulation, or whether it may be a student coming out of a pharmacy college. We require quality to be potential threat to be identified and be treated it at the starting stage to the end stage, not after the production stage. Thank you, sir. We will be indebted to you and we will always remember this. Now I may request my technical team to display. Although we are very small, we are in no authority to certify your expertise, but still, although Dr. C.D. Upasni has already made a statement that from next year, Dr. Saranjit Singh, sir, he will be certifying all the certificates and all the efforts for the resource persons. Still, if you can accept, sir, our humble request from all the organizers, just to accept this e-certificate prepared by us for your efforts, sir.
Thank you so much. Thank you. I humbly request that I will do the job, whatever you are assigning me, and I accept this certificate with all humbleness.
Okay. Thank you, sir.
Thank you, sir. Thank you, sir. I may also request our technical team to display the certificate for our chairperson of this session, Dr. Udupa.
Thank you, sir. Thank you. Thank you.
Kindly accept, sir.
Yeah, thank you. Thank you.
I may now hand over to the coordinator, Dr. Atish Mundada. Thank you, sir.
Thank you, Dr. Chatpaliwar. It was really a brainstorming session being delivered by Dr. Saranjit Singh, sir. And it goes along with his name, the way he is going to deliver. It is always admirable. And for us, it is like a moment of pride that every year, Dr. Saranjit Singh, sir, will be there. Being a brand ambassador of SNJB PharmaCon, we are really fortunate to have you with us, sir, every year. And every year, our strong is getting strengthened and strengthened. And we will continue this for years together.
Thank you, Dr.
Thank you very much for sparing your valuable time. And we are really ashamed of ourselves seeing your energy at this age, what we should be doing. And you are a role model for n number of pharmacists throughout the world. We'll try our level best to follow whatever you said today. Quality really needs to be built into the product. We cannot just expect the quality. And we'll try to design the quality in each and every student to whom we teach, to whom we nurture, and to whom we ensure when they are getting into the field that they will be the quality product and they will be taking our name ahead into the field. Thank you, sir, for being there and for blessing us every year. At the same time, I'm really thankful to Dr. Udupa, sir, for sparing his valuable time and for chairing the session.
Thank you.
We are coming to the end of technical session of day 3. Here we'll take a break for a lunch, and in the noon session we are going to have last 10 presentations which are going to be presented by PG and PhD students for presentation competition.
Dr. Advait,
Yes, sir.
Advait Gujarati wants to share his experience.
Okay, sir.
Advait, kindly.
Advait, over to you.
Yes, sir. Thank you so much, sir, for giving this opportunity, sir. Actually, sir, from last 2 years, I'm in very less touch with the academic department, and currently, sir, I'm working with Alembic Pharmaceuticals only, sir, as a marketing professional in particularly ophthalmic division. So, as Saranjit sir was telling at the beginning of this session, related to quality products, and if the particular that active pharmaceutical ingredient is of utmost quality, so its expiry will be at infinity. That particular molecule will never expire. So I just wanted to point out that one thing that still say in market, as I'm working with ophthalmic division, there are many products in market, sir. Particularly, I'll tell regarding the moxifloxacin brand, sir, with very cheap quality, sir. And there is one brand in I'll just not take the brand names or company names of that particular brands, but there is one very good brand, sir, in market named Vigamoxer of Alcon, which is a subsidiary of Novartis. So I just wanted to share that one of the doctors shared that experience. So when he's using that Vigamox in particular patients after phacoemulsification cataract surgery, so he is getting very good results. But similarly, so when he was using initially some other brands of that cheap quality, which are so very well-known and renowned companies of India, I'll not take the names, but he is facing some issues in the quality, like the patients are having hyperemia conditions, watering, irritation, and that conditions in eyes. So just wanted to share that still, sir, that quality products in the market are not coming from very renowned companies also. And if means just wanted to ask that also, that what actions also we can take against such products, or like just wanted to ask you that only.
You're asking me?
Yes, sir. And this, yes, to you, sir. And it's very, sir, always. Yes, sir. Please continue.
It is a role of the regulator to monitor such thing, and doctors, like we have a very strong pharmacovigilance program these days, and
Yes.
it is being conducted under the banner of Indian Pharmacopoeia Commission. They are collecting the data from doctors, and doctors should really fill up some pharmacovigilance format is there in which they can report to the regulatory agency, and the pharmacovigilance division will report to the regulatory agency of these bad quality products, or they can directly write to DCGI about it. Now, we people will not come to know very easily of the bad quality products unless we analysts test in our labs. But this is an efficacy issue, efficacy and safety issue, not only quality issue at a time. So what ICH has done and what I have propagated through my presentation today was that this is quality vision of ICH of 2005, that 2003, that we need to set up systems that every product piece that comes out from the factory anywhere in the world is of right quality for the patient. So this technological change that I have discussed today is only targeted to achieve the problem you have raised. So
right, sir.
So that is due to lack of knowledge of factors that can lead to bad quality product in industry. So you are in Alembic, allow me to give a lecture in Alembic of a similar kind to sensitize to all of your team, total team. What is the direction of change that is happening? I already have started continuous manufacturing, but every staff member, why it is being done, that is very necessary. So that total, what we say, awareness is created. It's very, very important. So once awareness is created, people will start taking action. Till that, we as patients will continue to suffer. So that is the objective of ICH Quality Vision of 2003. Thank you so much.
Right, sir. And thank you so much, sir. And it's a very pleasure, and it's always interesting to listen to you, sir. And thank you so much, sir.
Most welcome.
Thank you, Advait.
I am happy to share that many persons, those who are working in industry, they are also attending this session.
Great.
So we have limitation in our Zoom platform, but they are on YouTube platform.
Okay.
That's a great thing. Thank you very much, sir.
Thank you. Okay. So nice of you.
We are highly obliged by your presence, sir. Keep showering your blessings every time.
I'm missing your auditorium.
Yes, sir. We are also missing your presence in Chandwad. And today, let me tell you, the atmosphere in the morning was awesome. You would have actually loved to be here in Chandwad today. And we are also missing you and your presence.
Probably
next year, we will be having you in our campus.
Discuss the theme before you put a theme for the next SNJB PharmaCon with me so that I can prepare some lecture on the theme.
Yes, sir.
Okay, sir. No problem. For sure, we'll do that.
Great.
Thank you, sir.
We are really thankful to our protagonist, Dr. Saranjit Singh, sir, for being there with us. Here, we'll take a lunch break and sharp 2 p.m. we'll start with our oral presentation competition. Thank you, one and all.
Thank you, sir.
Thank you. Thanks a lot.
Thank you, Udupa sir.
Thank you.
Thank you, Udupa, sir. Thank you,
Have a
sir.
great day.
Yeah.
Thank you. Thank you.
Look forward to visit Chandwad.
Yes, sir.
Thank you, sir.
Call you.
We'll plan, sir.
Yeah, yes.
Next year you will have to come.
Yeah, definitely. Yes.
Thank you, sir, Ranjit Singh, sir.
Okay, bye-bye.
Thank you. Bye.
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्रातल्या नाशिक जिल्ह्यातलं चांदवड इथलं SNJB कॉलेज.
MBA,
इंजिनिअरिंग,
फार्मसी,
डिप्लोमा इन फार्मसी,
होमिओपॅथी मेडिकल,
पॉलिटेक्निक,
ड्रेस डिझायनिंग अँड मॅन्युफॅक्चरिंग,
इंटिरियर डिझाईन अँड डेकोरेशन,
आर्ट्स, कॉमर्स अँड सायन्स,
B.Ed,
व्होकेशनल कोर्सेस या सर्व शिक्षण शाखांचं सर्वोत्तम शिक्षण देणारी
मुला मुलींसाठी स्वतंत्र वसतिगृह असलेली
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्रातल्या नाशिक जिल्ह्यातलं चांदवड इथलं SNJB कॉलेज.
MBA,
इंजिनिअरिंग,
फार्मसी,
डिप्लोमा इन फार्मसी,
होमिओपॅथी मेडिकल,
पॉलिटेक्निक,
ड्रेस डिझायनिंग अँड मॅन्युफॅक्चरिंग,
इंटिरियर डिझाईन अँड डेकोरेशन,
आर्ट्स, कॉमर्स अँड सायन्स,
B.Ed,
व्होकेशनल कोर्सेस या सर्व शिक्षण शाखांचं सर्वोत्तम शिक्षण देणारी
मुला मुलींसाठी स्वतंत्र वसतिगृह असलेली
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्र
Good afternoon. Good afternoon, everybody. We are about to start the scientific competition of day 3 for SNJB PharmaCon. I welcome you once again for this competition. I am just waiting for confirmation from today's guest invitees, Dr. Sridhar Pai from Manipal, College of Pharmacy, Manipal.
Yeah, good afternoon.
Good afternoon, sir.
Good
How are
afternoon,
you, sir?
Good afternoon. Good afternoon.
Greetings to
Dr.
you,
Pai,
sir.
how are you?
I'm good, sir. Very good. How are you, sir?
Fine, I'm good.
Thank you.
Thank you for joining us.
It's my pleasure.
And moreover, thanks to you for sparing your half of the day for us.
Definitely, sir.
In the morning session, we had the professor and principal, Dr. Udupa, with us.
Okay.
He is still there.
He's in Dharwad now.
He is in Dharwad now.
Yes.
Okay. Yeah, the other expert, other judge is Dr. Kushwant Yadav. Dr. Patil has he joined? Dr. Kushwant Yadav? Yeah, I can see him.
Yes, sir, I'm there, sir.
Welcome, doctor.
Thank you so much, sir. Good afternoon, everyone.
Good afternoon.
Good afternoon, Upasni, sir.
Good afternoon, Kushwantji. How are you?
I'm good, sir.
Good afternoon.
Okay.
Thank you so much. I'm pretty happy to be here. Thank you, Sanjay, sir, for the invite.
We too are happy to see you here.
We are glad to receive these two scientists, eminent scientists from the field of pharmacy, serving pharmaceutical institutes of repute in the country. I now request my fellow colleague, Mr. Manoj Kumar Mahajan, to introduce the guests, the judges of today's scientific competition to the delegates and participants. Over to you, Mr. Mahajan.
Thank you, sir. Today, we have two eminent personalities from the field of pharmacy as evaluators for the scientific session. First one is Dr. Sridhar Pai, who is currently working as professor in Department of Pharmacology, Manipal College of Pharmaceutical Sciences, Manipal Academy of Higher Education, Manipal. He has completed his B.Pharm in 1994 from National College of Pharmacy, Shimoga, M.Pharm in 1997 from Manipal College of Pharmaceutical Sciences, Mangalore, and received his PhD from QMP University in 2002. He has been working in academics for the last 24 years. He presently holds the head and professor of pharmacology in Department of Pharmacology, Manipal College of Pharmaceutical Sciences. He has received a full resident scholarship award to the Residential School on Medicinal Chemistry in 2008. Chemistry and Biology and Drug Discovery by Drew University, Madison, USA. He has also received 4 times Good Teacher Award for the years 2007, 2009, 2013, and in 2017, and the Teacher of the Year Award in 2014 for excellence in teaching by Manipal College of Pharmaceutical Sciences. Dr. Pai has visited several internationally reputed research laboratories and universities in several countries, such as Australia, Singapore, USA, UK, France, Dubai, Malaysia, Indonesia, and the list is still going on. His area of research interests are cancer biology, computer-aided drug discovery, and metabolic disorders. He has research collaboration with molecular neurotherapy and imaging laboratory at Massachusetts General Hospital and affiliated to Harvard Medical School, Boston, USA. He has guided 48 MPharm students and eight PhD candidates and currently guiding 2 MPharm and 7 PhD candidates for research projects sponsored by various government and private funding agencies. Dr. Pai has published more than 107 articles in national and international journals with high impact factor and contributed to the chapters in books. The H index is about 19, and he has also one patent to his credit. He is also the quality management representative for the Manipal College of Pharmaceutical Sciences, and also a team leader for the NAC action team, the NRF ranking team, and Institute of Eminence team for Manipal Academy of Higher Education. With this brief bio, I'd like to welcome Dr. Sridhar Pai for this scientific session. Welcome, sir.
Thank you. Thank you very much. Thank you.
Another evaluator that we got for today's scientific presentation session is Dr. Kushwant Yadav, who has completed his MPharm from Roland Institute of Pharmaceutical Sciences, Behrampur University in July 2002. He pursued his PhD from the reputed Maharaja Sahajrao University of Vadodara in 2010. He has several research projects which were funded by different eminent agencies like SciTech Center, Mumbai. He has also received a postdoctoral research grant from the University of Oklahoma Health Sciences Center, USA. Also, he is the principal investigator for the AICTE RPS scheme under formulation development and evaluation of hydrogel-based formulations, and he has till today faced grants of more than 40 lakhs. Key achievements of Dr. Kushwant Singh includes the Young Scientist Award, the Young Achiever Award, and he is also serving as the editorial board member for several reputed journals, including the Daru Journal of Pharmaceutical Sciences, the Drug Delivery Letters, Neuroscience and Nanotechnology Asia Bentham. He is currently working as an associate professor at SVKM's NIMS Mumbai. With this brief bio, I'd like to welcome Dr. Kushwant Yadav for the scientific presentation session of SNJB PharmaCon 2020.
Thank you so much, sir. It's my pleasure.
Over to you, Dr. Sir.
Thank you, sir. Thank you, Mr. Manoj. I welcome both the judges, and I would like to give a short preamble to this scientific competition. This is the third day. We have in total 30 presentations, out of which 20 presentations were scheduled yesterday and 15 presentations had been made till yesterday. Today we have to judge for 10 presentations. In all, we have received 60 participants, 60 abstracts, out of which we have scrutinized 30 abstracts, shortlisted 30 abstracts for presentations based on the rationale of the experimentation, rationale of the project, explanation of the project, experimentation, and the results which were submitted to us through abstracts. This year, we had totally put a no for submission of review articles because we have to respect science and research. In order to increase the standard, maintain the standard, we have to say no to the review articles, and we have to entertain only quality abstracts. So out of those, we will be going for 10 abstracts, remaining 10 presentations today. I will read out the names. These are from all 4 fields. The first presentation will be made by Ms. Sheetal Gosavi. Her presentation is coded PH9. Every presentation has been coded and the mark sheet is available with the evaluators. The title of Her presentation is synthesis and characterization of stavudine-loaded mesoporous silica nanoparticles. The second presentation in line is entitled Formulation, Development and Evaluation of Pharmacosomes of Terminalia catapa, presented by Ms. Anjali Saharan. The third presentation is from the field of pharmaceutical chemistry, that is bioanalytical method development and validation of azenilidipine by RP-HPLC. This will be presented by Ms. Anuja Bhosle. Fourth presentation is from pharmaceutical chemistry 2, development and validation of simple RP-HPLC analytical method for chlorhexidine acetate. This will be presented by Mr. Mayur Tekade. Next presentation will be made by Mr. Mehul Chorawala from the field of pharmacology, entitled Cell Wall Contents of Probiotics, Lactobacillus acidophilus ameliorate lipopolysaccharide induced murine colitis by inhibiting immunoinflammation and oxidative stress. Next in line is Ms. Lipi Joshi. Her topic is entitled Development of Novel Animal Model of Hyperuricemia by Chronic Administration of Uric Acid in High Fructose Fed and Oxonate Treated Rats. Next from the field of ecology is Agamantine attenuates behavioral and biochemical alteration in rats exposed to ethanol during adolescence. This presentation will be made by Mr. Mayur Kale. Eighth presentation today is effect of ferulic acid in fructose-induced hyperuricemia and associated complications by Ms. Palak Shah. Next two presentations, that is 9th and 10th for today, is from the field of pharmacognosy. First one is entitled Comparative Analysis of Anti-Glycating Potential of Natural Products by Mr. Ahmad Ali. And the last presentation today will be Role of Embelin isolated from Embylliarabis in biofabrication of selenium nanoparticles and its study in breast cancer nanomedicine. This will be made by Ms. Rutika Jagtap. I appeal all the 10 participants today to be ready with their slides. When they will be called and they will be allowed, they should display their presentation, their slides, and start their presentation. Should I call upon the first candidate today, Ms. Sheetal Gosavi?
Yes, sir. Yes, sir, I am audible.
Yeah, you are audible. Will you please share your slide?
Yes, sir.
I wish you all the best for your presentation, Ms. Sheetal.
Thank you very much, sir. Is the screen visible, sir?
Yes, sir. Dear judges, should we start?
Yes, please.
Okay. First of all, I want to say thanks to the organizer for giving me such an opportunity to presenting my work. So I am Sheetal Vaban Gosavi, presenting here the synthesis and characterization of stavudine-loaded mesoporous silica nanoparticle under the guidance of Patil sir. Okay, so these are the content of my research work, introduction and objective plan of work, experimental result, and the discussion. Okay. So the mesoporous silica nanoparticles, it is a versatile drug delivery system that can be used for the loading different gas molecules into it. For example, anti-cancer agent or antibiotics, antiviral drug, protein, or the peptide. Now, simply mesoporous silica nanoparticle. If I want to explain it in simple word, I can say the mesoporous silica nanoparticle, it is like a honeycomb. So the mesoporous silica nanoparticle are prepared. This is the blank, and then the drug is loaded into it because it is a honeycomb-like structure and it has a pore. If we can say the honeycomb without honey, that we can say this is the blank mesoporous silica nanoparticle, and honeycomb with the filled with honey, that we can say the drug loaded mesoporous silica nanoparticle. It is versatile because of its properties like high surface area. It has a tunable pore volume and the pore diameter. It has rigid and stable framework. It has thermal, mechanical, chemical stability, and it is a biocompatible high loading capacities here. So the aim of my work was synthesis and characterization of taurin loaded mesoporous silica nanoparticles. The objective was to prepare mesoporous silica nanoparticles to load taurin into the blank mesoporous silica nanoparticle and to study the in vitro release study, then to improve the patient compliance. So with the help of literature survey, we have selected drug and excipient, reformation study, synthesis is done, and as per the material and method, the stavudine, tetraethyl orthosilicate, polaxomer, N-hexane, dihydrogen phosphate, these material are selected. Then one simple method is selected, that is the Stober process, and we can say the Sol-Gel process. So with the help of this process, we have formed the mesoporous silica nanoparticle. First of all, 2 molar HCl and water is mixed together. Then polaxomer is added. Then it's stirred for 15 minutes. Then add tetraethyl orthosilicate silicate dropwise under the continuous stirring for 2 hours at the room temperature. Then white precipitate was obtained, that filter, wash with water, dried at room temperature for overnight, and then it is calcinated. So the calcination time is then selected. We have selected the calcination time with the help of muffle furnace by placing that material into the muffle furnace for particular time. Then we observe the free flowing white powder, that is our optimized batch. Then drug is loaded into that particular blank mesoporous silica nanoparticle into 2 steps. One is initial loading and the solvent evaporation. Initially, solvent is dissolved in N-hexane, then that MSN and N-hexane solution is formed. Then the mixture is sonicated for 10 minutes. That's why the silica nanoparticle are evenly distributed, and the mixture is stirred for 2 hours at room temperature. Then solvent evaporation is done. Then the resulting material were donated as the mesoporous silica nanoparticle of stavudine. After that, we have done the characterization of that particular mesoporous silica nanoparticle. As per the characterization, we have get some results. So first, we have studied the nitrogen absorption, desorption, isotherm of blank mesoporous silica nanoparticle. So as per the N2 absorption, desorption, the results show there is the 4th type of graph. As per that graph, we have say that is the mesoporous material. Blank MSN is, as per the N2 absorption and desorption, we have observed the surface area, pore size, and pore volume that particular mesoporous silica nanoparticle. We have observed the surface area of blank MSN is more as compared to the drug-loaded MSN. It may be due to the blank pores, and after the loading drug, its surface area may be decreased. Then the pore size of blank MS is observed is 2.161, and drug-loaded MSN was found 3.439. It is may observe the pore size is less as compared to the drug-loaded MSN. As per the FTI spectra, there are the three spectra. A indicate the blank emissions, then poloxamer. B indicate the drug, that is the stavudine, and C indicates the drug-loaded mesoporous silica nanoparticle. So as per the graph, we observe that there is no interaction in between the drug and the excipient. As per the DSC thermograph of stavudine and the stavudine-loaded mesoporous silica nanoparticle, it is observed that the stavudine melting point is observed at near about 171 degrees Celsius, and the drug loaded melting peak is observed at 168 degrees Celsius. So as per this graph, we observe the presence of drug, and here the slightly broad endotherm is shifted to the lower side temperature. It is observed that stavudine successfully loaded, or the material is loaded into that blank mesoporous silica nanoparticle. Then we have characterized that mesoporous silica nanoparticle by X-ray diffraction. As per the X-ray diffraction, there is observed the broad peak that indicates the material is amorphous in nature. Material is crystalline. The phase transmission is here, and the material is become amorphous in nature. Then the particle size is determined by the zeta sizer. As per the observation, we observed that the Z average diameter is found is 171.5 nanometer. Then pore diameter index is found to be 0.40T2, and the intercept is found to be 0.937. So as per the study, we have gained the particle size. Then after that, we have studied that mesoporous silica nanoparticle by scanning electron microscopy. Both the blank mesoporous silica nanoparticle and stavudine loaded mesoporous silica nanoparticle are studied. As per the SEM, we have observed that the formed mesoporous silica nanoparticles are spherical in nature. So the blank emitters are less aggregated as compared to the drug-loaded mesoporous silica nanoparticle. This point is observed. Then we have studied the drug release or in vitro release study. We have made the 8 batches. Then we have observed the release rate from 1 hour to the 10 hours. So as per our study, we have optimized the batch 5, because at the batch 5, at the hour of starting or the first hour, there is a release of near about 14.30% of the drug is released. Then after 2, 3, 4, 5 up to the 10 hour, at the last hour, the drug releases 78.30%. As per this dissolution study, we observed that the drug is releases sustainable in nature. So after that, we have studied the percent yield, entrapment efficiency, and drug loading of that stavudine containing mesoporous silica nanoparticle. So the percent yield also we have determined, then entrapment efficiency determined, drug loading capacity also determined. Here also we have studied the 8 batches, but we have obtained the F5 batch as the optimized batch, because percentage yield is found most as compared to other. Then entrapment efficiency also is more, and drug loading capacity also more. So determination of optimized batch is F5. Entrapment efficiency is 85.56. Drug loading is 61.11 and percent CDR found is 78.13. So the prepared mesoporous silica nanoparticle was prepared by the process is Stobber's process, or we can say the Sol-Gel process. This process is very simple and cost-effective process. Then, after the characterization and evaluation study, we have observed that we have made the batches as per the variation of that particular surfactant. As per the surfactant, we have observed that the F5 batch, it is optimized because the surfactant ratio was 1 gram and it showed the most of the entrapment capacity, drug loading capacity, and the sustained release mechanism. So the first of all, I want to say that our aim was to produce the sustainable drug delivery system because we have selected the stavudine drug. Stavudine, which is the antiviral drug and which have the most of the dose dependent side effects. Now, the dose of that particular drug is given. After that, the dose dependent side effects are seen. But if we produce the sustainable drug delivery system, for example, the patient have to take the dose of stavudine within a day, it is a 2 times. There is the most of the chances of the side effect. But if we have produced the sustainable drug delivery system, then we want to take only 1 time that particular dose, and at that particular dose up to the whole day, it will release slowly, and there may be the less chances of the side effect. So this was the aim of my work, and we successfully loaded that mesoporous silica nanoparticle into that particular stavudine is loaded into that particular mesoporous silica nanoparticle, and as per the in vitro drug release, we observe there is a sustainable drug delivery system. Thank you very much.
Thank you, Ms. Shital.
Thank you, sir.
Now, may I invite Dr. Pai to have his questions?
Yes. Okay. Thank you.
Yes, sir.
Hello. Yeah. Can
Yes,
you hear
sir.
me?
Yes, sir.
Yes.
You're audible.
Yes, sir.
Okay. Thank you. Thank you, Shital, for the nice presentations. I have one query. Yeah.
Yes,
Can you hear me, Shital?
Yes, sir. Yes, sir. I can hear you.
Yes. Why we have specifically selected stavudine in this formulation? Is there any specific reason?
Sir, as per the literature survey, I was studying that particular point. The mesoporous silica nanoparticle particularly used for the poorly soluble drug or the drug which have the most of the dosing. If there is a most of the dose, that time there is a side effect of that particular dose. Then when I was studying or I was literating that particular, I was surveying, that time I observed that there is a very difficulty to patient to take that of lots of dose of that any particular drug. So as per the literature survey, it is observed that there should be the patient compliance, and to overcome the side effect or to decrease the side effect of that particular disease. So I selected the stavudine for my work.
Hello.
Hello. Am I audible, sir?
Yeah,
Yes.
you were
Yes.
audible. Yes, yeah.
Yeah. Thank you.
Okay.
Thank you.
I think Dr. Sridhar is satisfied. May I now invite Mr. Khushwant Yadav?
Yes, sir. Yeah, Ms. Sheetal, I have a couple of queries in your work that you have stated. Like, first of all, in your objectives, you have listed you aim to work towards patient compliance.
Yes, sir.
I mean, if you can take me to your slide, where you have put your
Yes,
objectives.
sir. Yes, sir.
So I would want to know how are you going to check the patient compliance?
Sir, my work now, I study through, but when we have studied the in vivo, that time, one minute. The future of work of this study is main, that is the in vivo study. And as per the US FDA, in 2011, they have approved for the silica nanoparticle for the clinical trial. So because of that, my objective was to improve the patient compliance.
That's what I'm asking. Even if I understand you have not finished your work, but by any chance, even if you would have finished your work, will you be able to check your objectives? So ideally, objectives once you plan, they should be achievable. Can you check your the study of planning to do the patient compliance? Are you going to check your formulation in the human beings?
Now I'm not checking, but in future I will try, sir.
In future means, is that within the scope of your PhD?
Yes, sir.
No.
I hope you are understanding like what you're saying. Usually in PhDs, we would go for preclinical studies, which is in animals. And it's so difficult to get permission because how will you get the permission for clinical trials?
Okay, sir. I got, sir. I got.
So please do not put them, whenever you state your objectives should be achievable. That is one. Okay. Next, when you go down, when you have shown your XRD, in the XRD, to me, both of them look quite similar, and you have stated that it is getting converted from crystalline to amorphous. How did you say that? Can you show your, the slide, both of them? Yeah, so if you see them, I am still seeing strong crystalline peaks in both of them. How did you think that one of them is amorphous? They are almost same.
Quite apparent from IR spectra also.
True. So, I mean, please repeat this study. Maybe there's a problem in the study. Next issue is in your zeta potential. When you're showing your size, in your size, you're showing there are two peaks actually. Did you not think that ideally you should have got a single peak, because that is also impacting your PDI?
Okay, sir.
Can you go to that? I think it's next. Size distribution. So what according to you should be the PDI? Yeah. So if you look here, you got a very clear two peaks.
Yes,
do you
sir.
think this kind of two peaks is acceptable? Generally, no.
Yes, sir. No.
Then when you go down further in your DISO studies, I don't know, did you just do one single study for each batch, or did you do it in triplicate?
Sir, it is done in single batch, but
In triplicate? Can you show that table where you have shown those 8 batches? Yeah. So when you look, yeah. The previous one. Previous.
Yes, sir. Yes.
The table where you have shown 8 batches of dissolution data. Yeah. So here what I want to ask is, did you do a single study? A single times you have done one diso for one batch, or in duplicate or triplicate?
Single study, sir.
Single study. So I would want to tell you is, whenever you perform these kinds of studies, please perform them in triplicate. And after that, use plus minus standard deviation, because how, I don't know, you have thought that one of those batches is optimum, because to me all of them almost look the same. I don't know for how for you 68% drug release is different from 72. Even if you would apply the statistical significant difference test, you will find that they're almost same.
Next time I will consider that point, sir.
But even if you look at these, like with these results, out of 8, I think 4 of them are almost same. And then one last question, like before we end, you aim to make them sustained release. How do you feel that this data of 10 hours is eligible to show that this is able to sustain the release?
Sir, because of at the first hour it is a less of the drug is releases, and after 10 hours it release as compared to first.
What is your route of administration?
It requires 10. Oral, sir.
So in oral, did you compare the diso data with the conventional tablet that is available?
No, sir.
Why didn't you do that? That is important, na?
Yes, sir.
And even if you would have done that, you would have seen that. So what are you expecting in a conventional tablet? How long will it release?
Okay.
So ideally, your aim, if you're planning to make it a sustained release, I think that is not still achieved. You still need to work.
Yes, sir.
That's it from my side. Thank you so much.
Yeah, Ms. Shital, you need to convey this message and you need to make changes according to the points raised by the respected judges. I want to ask you one question. Ms. Shital, do you believe the statement that excipients, whatever excipients you are using, it has no interaction with the drug?
Yes, sir, because of the FTIR, we have studied FTIR.
No, I'm not talking only about FTIR, but it should be, take it as a basic thing. Isn't there any interaction, physical or chemical, happening between any of the excipients and the drug candidate?
I think no interaction.
If there is no interaction, then every excipient, every chemical will be an inert compound.
Because, sir, IMSN is most of the chemically inert and physically and chemically stable.
No, I'm not talking about toxicity. I'm talking about the interaction between the drug molecule and the excipients. There has to be some interaction. Then only the excipient will be acting as an excipient for carrying the drug into the body and releasing it. Are you getting my point?
Yes, sir, I am getting.
And that is the interaction which you have to study. You are not supposed to show that there is no interaction, but you have to show the interaction which occurs.
Okay, sir.
Okay. Thank you. Dear judges, we will be taking the next presentation. I may mention here that I have checked the list and I have called many a times for Miss Anjali Saharan. I think she is not able to establish a connection with us. So with your kind permission, I will be calling next candidate. Her presentation is encoded PC3. She is Miss Anuja Bhosle. She is available in the room and she has been asked to display her slide.
Yes, sir. Thank you, sir. Sir, is it visible? Yes, sir.
Yeah, it is now visible.
Okay.
Yeah,
Thank
it is
you, sir.
visible. Yes, yes.
Okay. Good afternoon, one and all. Today I'm here to deliver.
Make it a presentation mode.
Yeah.
Yes, sir. One. Good afternoon, one and all. Today I am here to deliver my presentation on the topic bioanalytical method development and validation of azilnidipine by RPHPLC. Here I have selected the drug that is azilnidipine. That is an antihypertensive drug which is used for the treatment of hypertension. And here I have optimized the method for RPHPLC. So due to the slide constraint, I have developed means I have shown the in the 8th slide the whole experimental work that I have done. So here first the flow of the work, it involves the selection of suitable solvent for the analytical solution preparation. So for that I have selected the methanol as a solvent for the preparation because the drug is soluble in methanol. After that, next one is to select the appropriate wavelength for the analysis. So here from this UV, you can see that here 256 nanometer, it is a maximum wavelength where the drug shows the maximum absorbance. So here 256 nanometer is selected as a wavelength for the analysis. Next one is, as it is a bioanalytical method development. So first I have optimized the one method on HPLC, and after that I have done the extraction of the drug from the blood plasma, and after that again I have performed the whole method along with the validation according to the bioanalytical validation guideline. So here first I have selected the methanol water as a mobile phase in the ratio of 70:30. So here and the flow rate is 1 ml per minute, but the AZP was not eluted here. So this is my first trial 1. Next one, after that I have changed the mobile phase, that is astronitrile water I have took. So here again the 1 ml per minute is the flow rate, and retention time was more. So here if I see in the trial 2, so here the retention time is more. So this is not accepted. We should, if you want to get the economical results, so I should reduce the retention time. So again I have done the trial 3. Sorry. So here astronitrile water 70:30. So here the 2 ml per minute is the flow rate is optimized, and here the 7.2 is the retention time. But here some peak distortion, peak shape is not good here in trial 3. Next one is trial 4. So here astronitrile buffer in 90:10 I have tried. The again flow rate is 2 ml per minute. Here I got the retention time which can be acceptable, but tailing is observed in trial 4. After that, next one, trial 5, I have changed the flow rate, and here the good chromatography is observed. So here first I have tried methanol water, after that ACN water. After that I have used the buffer. Okay, so after using the buffer I got the means peak distortion is minimized, and I have got the RT at 4.8. So this is about my trial 1, trial 2, trial 3, trial 4, and trial 5. After that, next one, I have done the extraction after getting the sufficient retention time after optimizing the method. I have done the extraction. So here first I have prepared the 1000 ppm of solution of AZP. From that I have spike 5 microliter, 10 microliter, 15, 20, 25, and 30 microliter of the that is a stock solution into the 1 ml of plasma. After that, I have mixed it vortex for 2 minutes. After that, I have added the extraction solvent. So here I have used the protein precipitation method. So I have added the 0.5% formic acid as a extracting solvent. And after that, extracting solvent addition, I have again vortex for 1 minute. Again centrifuge for 3 minutes for 4000 rpm, and again supernatant is collected and it is injected into the HPLC system. So here after that I have performed the validation parameters for the bioanalytical method development. So for that I have used the CDR guidelines. So in that the standard that is depend on the first chromatography, means chromatogram. If at 100 means for the 25 ppm I have tried for the trial. So if it gives a 100 height, so for the 2 to 3 height, so 5 ppm is considered, depend on the that is 2 to 3 height. After injection of the 5 ppm, I got the 2 to 3 height and good chromatogram there. So it is considered as a LLOQ, depend on the visual observation. As according to the guideline, your LQC, MQC, HQC, it should be selected for the further validation parameter. So in guideline, they have given the how to select the M and HQC. So if LLOQ is 5 ppm, your LQC, it should be 3 times of your LLOQ. So 15 ppm I have considered as a LQC. At the mid range, 20 ppm is considered as a MQC, and HQC is 25 ppm. So this is the area. And next one is the, again, I have performed the selectivity. So for the selectivity, the criteria is that there should be no interference of the plasma at the RT of your drug. So here the RT is around 2.5. So here there is a no interference of the plasma. So blank plasma is also analyzed along with the calibration curve, means QC samples. Next one is this is the calibration curve for my extracted AZP. Here I have considered the 6 that is non-zero standards for the method, that is 5 to 30 ppm. Next one is the accuracy and precision. So for the accuracy and precision, I have injected the 6 sets of the LLOQ, LQC, MQC, and HQC, because as per the guideline, the LLOQ, LQC, MQC, and HQC, this should be used for the validation parameters. So from that, according to the guideline, the precision, that is percent CV, it should be plus minus 15% of your that is percent CV, it should not be more than the 50%. So here the 5.7, 0.55, sorry, 9.22, 6.38, and 3.04. These are the percent CV. Next one, for the accuracy, the percent nominal concentration, it should not be plus minus 15% for your LQC, MQC, and HQC. It should be again within the limit according to the guideline. For LLOQ, it should not be more than plus minus 20% of nominal concentration. It also comes 5.7. So here I can say that my method is accurate and precise one. Next one, system suitability parameters are also done. So here the retention time is 2.5. After that, asymmetry 1.09, and theoretical plates, these are 5,290. Next one is the most crucial criteria in bioanalytical method is the percent recovery. So here the percent recovery, as I have done the in vitro analysis. So here the percent recovery, it should be that is it should be more than 70%. It is not need to be 100%, but it should be more than 70%. So here I got the 92% recovery. Next one is the sensitivity. So sensitivity of the method, it should be checked. That is a lower concentration. It should be injected 6 times in your system. And again here your percent CV, it should not be more than the 15%. So here 5.46% is the that is a percent CV. So here again we can say that our method is sensitive. Next one is a carryover. Carryover, it should be performed because means there should not be any drug carryover in your next injection, because while we are doing the analysis, there should be the chances that the one drop of the solution, it can remain on the injection system. So again, it can be carried forward for the next. So here the first, the blank is injected. After that, the upper limit of quantification, means last standard, it is get injected, and again LLOQ. So in this way, again blank. So after that, at the retention time, there should be no interference. So means your drug is not carry over in the system. Next one is the stability. So your stability should be assessed for the bioanalytical method. So your benchtop stability, after that freeze thaw stability, auto sampler stability, and process extract stability, it should be performed. So it is performed on the LQC and HQC. So here benchtop stability, it is performed, that is sample, it is placed on the benchtop, that is a spike plasma. It is kept for the benchtop for 4, 6, and 24 hours. And after that, the stability checked. Again, here your percent CV, it should not be more than the that is 15%. Next one is the after benchtop stability, auto sampler stability is performed. So here Again, auto sampler means here extract, it is placed in the auto sampler for 4 hours, and after extract stability is performed, that is process extracted sample stability at room temperature. It is kept again for the 6 hours and analyzed. Again, your percent CV, it should not be more than 15%. Freeze thaw stability, it is performed, that is your spike plasma. It is kept for it is done 3 freeze and thaw cycles, means it is kept for 24 hours in the freeze and again thaw, again kept for 24 hours in freeze thaw. In this way, the 3 cycles we have conducted, and after that the plasma, that is your sample, is analyzed. So again, there should be no again percent CV, it should not be more than the 15%. Again, a long-term stability, it is carried out, that is 10, 20, and 13 days. That is, but what is the difference between this benchtop and long-term stability? Your long-term stability, it is conducted at your sample, it is stored at intended storage temperature, but your benchtop stability, it should be performed at the room temperature. Next one is the stock solution stability. That is your aqueous stock, not your extract. That is aqueous stock. It is kept for the 24 hours. After that, it is spiked into the plasma, and immediately it is processed, means it is extracted and it is analyzed. So this is all about regarding the stability study that I have performed for the under this bioanalytical method. So here, that is the conclusion, is that the developed HPLC in human plasma was selective. After that, sensitive. Selective means there is a no interference due to the that is a blank plasma in that system. Next one, it is sensitive because that is a percent CV. It is within the limit of the validation guideline. It is economical because the retention time that I got after the method development is a 2.4 minutes, and runtime I have took, that is a 10 minute. Hence that is your mobile phase, and again time it is get reduced here. Next one, the use of protein precipitation. Again, there are 3 methods by which we can extract the plasma. There is protein precipitation, liquid extraction, and solid phase extraction. So protein precipitation, it is the most economical method that I have preferred, and where I got the result. Next one, here the method, it is as we have done the stability study, the method is suitable for the handling various plasma levels of the drug. And again, it is validated by the CDR guideline. So thank you.
Thank you, Ms. Anuja.
Thank you, sir.
May I now invite Dr. Pai to have some queries?
Okay, thank you, Anuja. Can you hear me, Anuja?
Yes, sir. Yes, sir. I can hear you.
Yes. Thank you very much. Can you tell me why you have selected this particular candidate, and what is the outcome?
Yes, sir. Please, can you repeat your question?
Why you have selected this specific candidate molecule? You have selected this particular molecules. What is the specific reason, and what is the outcome, final outcome in one sentence?
Sir, this candidate means this drug molecule I have selected because there is no specific means method is available for the which is economical for the bioanalytical method development on this candidate or on this drug molecule. And outcome is that I have optimized the method which is sensitive, economical, and simple for the determination of drug in plasma, and it can be used for the pharmacokinetic study in the animals.
Okay, what is the present method is available?
Yes, sir.
What is the present method is available? Is there any other method available?
Yes, sir. The HPLC methods are available on the plain drug, and again LCMS methods are available. But here I have done the method on the RP-HPLC, means which is economical than our LCMS method.
Okay, thank you.
Thank you, sir. Thank you very much.
Thank you, sir. May I now invite Dr. Yadav?
Yeah, yes, sir.
Dr. Yadav.
Yeah. I think it was a very pretty good piece of work, and happy with the kind of data that you have got. Only my one small concern is I could not see much of plus minus SD data put in most of the things, and the units were missing at some places. ensure
Actually,
that
sir,
that is done?
in the bioanalytical, there is a data is more, but I have constrained into the 8th slide. And here the percent CV, the concern is regarding the percent nominal concentration and percent CV in case of bioanalytical. So I have presented that only. That is what is the percent CV and what is the percent nominal concentration as per the guideline which is required. So I have put it that only there, sir.
Yeah, that's fine. I've seen it. Like what I'm
Yes,
saying is
sir.
in most of the data, wherever you have done it 3 times or 4 times, you could have used plus minus standard deviation. That is missing.
Proper
Okay.
statistics missing.
Actually, I have means from that standard deviation, I have calculated the percent CV. So I have not put
Of
there.
course,
So I
I
have
can see
directly put
that.
it. Yes, yes, yes,
I
sir.
could see
Yes, sir.
that.
Okay,
So
sir.
I could understand that you have done those studies. Only thing
Yes,
it's not being shown here.
They're not
Okay, sir.
calculated.
Okay, sir. I will add it
Yeah,
afterward.
that's it. That's it. Okay,
Thank you,
thank
sir.
you.
Thank you.
Okay, Ms. Anuja, I have few queries.
Yes, sir.
Can I know the PK of this drug?
PK of this drug is around 6.
Around
6.
Okay. So I would be interested in knowing the pH of the mobile phase, the buffer which you have used in the
Yes, sir. I have used the 3.5
that was
buffer.
not mentioned here.
Yes, sir. I have not mentioned there. So here astronitrile water pH adjusted to pH 3.5. 3.5.
So do you know the use of buffers? Why buffers are used to in the composition of mobile phases?
So here during the method developments, I come to know that here there is some change after multiple injection. There is some change in the means reproduce. The method is that is RT is not reproducible. So
No, Miss Anuja.
I have added the buffer there in the mobile phase, sir.
No, why only buffer? Why not any other chemical?
Because what happens, sir? Means in the PK when pH in case of acidic drug, the pH of the mobile phase it should be plus means -1 or -2 of the PK of drug. So I have first studied the yes. So I have first.
So if you know this, you shouldn't have used the first 3 mobile phase compositions for trial even, because you have used the first 3 compositions without the use of buffer.
Yes,
Even
sir.
though you were aware of these facts that the mobile phase should have a pH -2 than the PK.
Sir, but due to the buffer, there may be the chances. Means, what is our first aim?
No,
Not
that
to
is
use
the
the
literature.
buffer.
That is
Yes,
the
sir,
literature.
not to use the buffer. So first, I have tried without buffer, but at the moment I want to use the buffer. I have done according to PK. Means this is all about the trial and error, and it is a research work. So
No,
for the
research is
method development, there is no guidelines. So I have done according to
Miss
my
Anuja,
knowledge.
no, Miss
Yes, sir.
Anuja, trial and error doesn't go into research. You have to stick to the literature. You have to search from the literature because it is very clear. You
Yes,
have
sir.
used a few experiments without any logic. The other
Okay,
fact which I have observed is there is a large tailing factor in your method. And do you feel that this selection of that chromatogram will be suitable for bioanalysis? Because the tailing factor which you are displaying is less, but the chromatogram displayed is showing a large tailing factor.
Okay, so in trial 5, sir, the tailing factor is less.
Yeah, that's what is displayed in the table, but from the chromatogram it can be read that the tailing factor is too much. That peak cannot be selected for a standard quantification.
Okay, sir.
Okay, so these things are to be taken care of while working with HPLC. All the best,
Thank you,
Mr.
sir.
Anuja. I wish you all the best.
Thank you, sir.
May
Thank
I
you very
call
much.
upon another contestant, Mr. Mayur Tekade. He is available.
Yes, sir. Yes.
Yeah. Will you please put your slide?
Yes. Yes, sir. Is it visible?
Yeah, it is. Please enlarge it.
Yes, sir. So, good afternoon, all of you. Today I'm presenting the seminar on development and validation of simple RPHPLC analytical method for chlorhexidine acetate under guidance of Dr. P.M. Patil madam. Next, that is introduction of my drug, that is chlorhexidine. It has a wide spectrum of bactericidal and antiviral activity of its common ingredient in various formulations, ranging from skin disinfectant in healthcare products to anti-plaque agent in dentistry. So the chlorhexidine is a chemical antiseptic. It is effective on both gram-positive and as well as gram-negative bacteria that have been bactericidal or bacteriostatic, that both have been mechanism of action. It under membrane disruption of that main mechanism of action, not ATPase, that is inactivation of a mechanism of action in that chlorhexidine. In that present work, the objective is to design and validate a simple RPHPLC procedure for the assay of chlorhexidine acetate. That I have been getting the chemical and reagent that is pharmaceutical grade chlorhexidine. That I have been getting chlorhexidine acetate was gifted from Avance Cure Life Science, and marketed formulation chlorhexidine acetate was purchased from Inforbia healthcare pharmacy at Mumbai, and methanol is used as SPLC grade, and acetonitrile were purchased from Ajanta and Pranav. Then chromatographic system and conditions which are used, that is SPLC system, Youngkin, that is SK gradient system, UV detector also uses. That column is used as 4.6 into 250 mm of C18 grace column. I have used the mobile phase, that is acetonitrile is to water in the ratio of 70:30 volume by volume. Then flow rate is 1 ml per minute, and injection volume is 20 micro per liter. Then temperature is ambient, and that wavelength is detected at 270 nanometer. That is optimization of chromatographic condition. Before that, there is a spectra of standard chromatograph of chlorhexidine, time versus voltage, that found on 3.13 minutes. That is different mobile phase were tested with the view of achieve simple, rapid and economical separation between drug and degradation product. That is optimal phase composition was found in to be acetonitrile and water of HPLC grade, that is 70:30. That runtime was 1 ml per minute. That before analysis, the mobile phase gas for 5 to 10 minutes in sonicator. Then chromatography studies were performed at ambient temperature. The optimized flow rate was 1 ml per minute at detection of wavelength, that is 272 nanometer. Here are the method validation. That is, first is linearity. It proposed method was evaluated according to ICH guideline. That is, chlorhexidine acetate showed linearity in the concentration 10 to 50 microgram per ml. That R square is found to be that is 0.9987%. Then the regression equation is obtained was y is equals to 91.329x plus 135.78, where y is the peak area and x is the concentration of chlorhexidine acetate that is used in microgram per ml. That equation was used to determine the amount of chlorhexidine acetate present in the stability sample. Then we can determine the detection of a limit and quantification. That determination of LOD and LOQ was based on the standard deviation of the responses and slope of constructed calibration curves, that is noise ratio is 3 as described by ICH guideline Q2R1. This is analytical guideline. That LOD and LOQ values were found, that is 0.2895 microgram per ml, and LOQ is found as 0.8773 microgram per ml. Then accuracy is determined, was measured by spiking standard drug in a predetermined tablet solution at concentration level of 80, 100 and 120%. Then calculating the percent recovery of active ingredient. In the present study, the mean recovery of target concentration was set to 100 plus minus 2% of acceptance. After that, we can determine the precision, that is intraday and interday precision. The method was expressed as CV percent was measured using triplicate determination of quality control sample of 10 microgram per ml of API on 3 different occasions on same day, that is intraday, and 3 different days, that is interday precision. That the precision percent CV determined as each concentration level was required not to exceed 2% at level on different occasion and different days. Here is the robustness. That method was evaluated as per the ICH Q2 to determine the constancy of the result when variable inherent to method analysis are varied deliberately, and it depicts the reliability of the analytical procedure. That tested was performed in order to obtain information about critical parameters affecting response, that is flow wavelength and mobile phase. Here is my conclusion, that it is a simple, isocratic, and rapid, accurate, precise HPLC method has been developed for the determination of chlorhexidine acetate in bulk and tablet formulation. That main advantage of this method over the USP method are that is simplest mobile phase and optimum flow rate and wavelength is isocratic mode. That develop the method suitable for high throughput analysis, quality control samples of API and formulations in the pharmaceutical industry. Here are the references. Thank you, sir.
Thank you, Mr. Tekade. May I now invite Dr. Pai for having some examination.
Hello, Dr. Mai.
Yes, sir.
Hello, can you hear me, Mayur?
Yes, sir.
Yes, sir. Yes, yes.
Yes. Yeah, Mayur, can you tell me what is the advantage of this particular methods over the conventional method?
Yes, sir. It is a simple, accurate, and we can use as a very precise or sensitive method. That's why
No,
we
what is the
choose that.
what is the present methodology available for this? Is there any?
Yes, sir. Present methodology are LCMS, MS methods are available on for impurity profiling of that specific substance.
Okay. So overall, what is the advantage of this?
Mean to say, what is the advantage that your method provides over the existing methods?
Yes.
Existing methods are we can determine the isocratic, simple, rapid, sensitive methods.
Okay. Anything else, Dr. Pai?
Thank you.
Thank you, sir.
Dr. Yadav?
Yes, sir. Mr. Mayur, when you look at this drug, this is quite an old drug and it is already there in so many pharmaceutical industries. And if you go through the literature also, you will find there are so many HPLC, RP-HPLC methods already available. Why did you want to develop one more method?
Yes, sir. I'm doing MPharm, but I'm side by side for that determination of that HPLC method. So I choosing that drug and its availability as also.
But
so that's why I choosing that drug.
But the HPLC and RP-HPLC methods are already available.
This
Yes,
reflects
sir.
the dearth of literature survey.
Yes, sir. Yes. Sir.
Okay, that's it.
Literature
That's
is
it from
not
my
deep.
side.
Here again, you have missed the pH of a PK of the drug. The importance of pH and importance of buffer was not apparent from your presentation. I wish you all the best.
Yes.
I hope that these all points would be helping you. You will be discussing with your guide and making necessary changes in your research work.
Yes,
May I
sir.
call
Yes,
upon. Okay, thank you.
Thank
all
you,
the
sir.
best, Mr. Mayur.
Yes, sir. Yes, sir.
May I now call upon Mr. Mehul Chorawala?
Yes, sir. Am I
If you
audible?
are- yeah, you are audible. If you are ready, you can put your slides on presentation.
Yes, sir. Okay, is it visible, sir? Is it
Not
visible?
yet.
Yes.
Not yet.
Yeah.
6 seconds lapse.
Is it visible, sir?
No, not yet.
Okay. Just a minute, sir. I think my laptop is getting hang.
Mr. Mayur.
Hello. Hello, sir. Sir, my laptop is getting hang. So
Yeah.
could you please start
Should-
with another
Yeah.
presentation?
That's what I was about to
Yeah.
ask.
Sure, sir.
Okay, thank you.
Okay, sir.
Next in line is, respected judges of the contest, we may call upon Mr. Mehul Chorawala at the end of today's presentations. There is some technical hitch with his laptop. We may ask him later on. In the meantime, Ms. Lipi Joshi is available in the classroom. Is she ready? Hello, Miss Lipi Joshi. She is visible in the class. Hello. She is not responding.
Hello.
Hello. Yeah, will you please stop your screen sharing?
Sir, actually, my laptop is getting hanged and nothing can be do.
Yeah,
actually.
I will.
Yeah, sir, you can.
I request my technical team to allow
Yeah.
Ms. Lipi Joshi to share her screen.
Sure. Thank you, sir.
The members of the technical team, would be nice if you could please change the host. Please make Lipi Joshi the host of the session, co-host of the session. Hello. Yes, Miss Lipi Joshi, are you available? Has it been sorted out? She is responding to me in the chat room. I request the technical team to allow her to speak, to put her microphone to on, unmute her microphone.
We have done, sir.
Okay. Miss Lipi Joshi, there is some problem. It seems that there is some problem from your microphone, from your end. So for the time being, it will be better if we shift to another candidate, Mr. Mayur Kale.
Hello, sir. Good afternoon. Am I audible?
Yeah, you are audible. Are you ready with your presentation?
Yes, sir.
Yeah. Will you please put your slide?
Yes, sir.
Respected judges, Mr. Mayur Kale's presentation is encoded P-call 9.
Yes, sir. Is it visible?
No, not yet.
Sir, now?
Not yet.
Hello?
Yes, we are waiting, Mr. Mayur. Yes, it has now started. Yeah,
Yes, sir.
finally. Yes.
Okay. So
Best
good
of luck.
afternoon, all of you. I am Mayur Bhimroje Kare, research scholar Pharmacology Department, Srimati Shantabai, SNJB SSJD College of Pharmacy, Neminagar, Sandwad. One of my protocol of PhD today I'm going to present here. The topic is agmatin attenuates the behavioral and biochemical alteration in the rat exposed to the ethanol during the adolescence. The code is PCOL9. Moving toward the introduction, the adolescent brain is in dynamic state of remolding. So as compared to an adult, when we expose ethanol to an adolescent animal, it more likely to exhibit a cognitive deficit, which include the learning and memory dysfunctioning. So when it comes to alcohol, it's showing a harmful effect depending upon its time of exposure, as the adolescent brain is more vulnerable to this condition. So this chronic ethanol administration, it alters the expression of the NMDA receptor and causes the complications. Also, amygdalocortical circuitry may be particularly vulnerable to the ethanol insult during the maturation. So postnatal day 28 to postnatal day 58, which is considered as an adolescent age in the experimental animal, in the rats, and that period, early adolescent, that is the postnatal day 28 onward, it is a vulnerable period where ethanol causes the insult during the maturation, and that will show the complication during a late life of that particular organism. So when my drug is agmatin. This agmatin is a novel neurotransmitter. It is synthesized from the allergenin. It is an endogenous biogenic amine. It is a pleiotropic molecule and shows its action by binding to multiple receptors like imidazole. Hello, sir, am I audible?
Yes, you were inaudible for some seconds.
Okay.
You are now.
Okay. So shall I continue?
Yes.
Yes. So my drug is agmatin, which is a novel neurotransmitter. It is an endogenous biogenic amine, which is synthesized from the L-arginine, and it is widely available in the chromatin cells of GIT. And in this, when we were talking about the CNS, it is widely located in the hippocampus. So this agmatin, it is an endogenous neuroprotective molecule, which is having a significant potential for the drug development. This agmatin is having NMDA antagonism. It attenuates the glutamate-induced neurotoxicity. It improves, previous study shows that it improves the cognitive function. It prevents the neurodegeneration, and it enhances the hippocampal neurogenesis. So in view of this, in view of the prominent role of the agmatin in the neurodevelopmental programming, the present study was designed to examine its ability to abrogate behavioral and biochemical deficit induced by the ethanol exposure in the adolescence rat. So methodology, male Sprague Dawley rat postnatal day 25 were assigned to a different treatment group n equals to 8 and housed 4 per kgs in a plastic cage under the control condition and were provided ad libitum access to the food and water. Animal were handled twice daily for 3 consecutive days, that is from postnatal day 25 to postnatal day 27 before the dosing commenced on the postnatal day 28. Animal were randomly assigned to receive the ethanol or it will receive the saline. Okay, so animal received a total 12 chronic intermittent ethanol, that is 5 gram per kg of ethanol, 35% volume by volume in a normal saline. So animal received a total 12 chronic intermittent ethanol treatment over a period of 21 days, that is from the postnatal day 28 to postnatal day 49. Dosing occurred on Monday, Tuesday, Thursday and Friday of each week, and animal were then allowed to wash out until they reach to the postnatal day 70. Postnatal day 50 to postnatal day 70 is considered as a washout period, and from postnatal day 71 to 81, behavioral assessment was carried out. Following behavioral assessment, animal were euthanized and biochemical estimation was performed. So the basic criteria for selecting the animal for postnatal day 28. The reason is from the postnatal day 28 to 50 was chosen based on an extensive literature which demonstrating that rat undergo the physiological and behavioral development similar to the adolescence in the human from this postnatal day 28 to 48, as this period postnatal day 28 to 38 is early adolescence and 38 to 48 is a mid adolescence. So the behavioral and cognitive impairment evaluation is performed from the postnatal day 71 to 81. Before that, during the washout period, agmatine sulfate 20, 40, and 80 mg per kg, which is dissolved in the saline and administered by the IP route once in a day during this washout period. The the behavioral and the cognitive evaluation was performed from the postnatal day 71. On postnatal day 71, exploratory behavior of the rat was measured using the whole board apparatus. This test whole board apparatus is widely used to assess the motivational drive, specifically neophobia, which contains 16 similar square compartment. And during the test session, the animal was placed in the center of the platform, and the number of hole poking was measured during a time of 10 minutes. On postnatal day 72, first we perform 72 to postnatal. For the next 4 days, we perform the recognition memory. So the novel object recognition test is performed on postnatal day 72. The novel object recognition test is used. It is a non-forced recognition memory test. So it contains a day 1 and day 2, which is a habituation period, where the rat were first transported to the preparing area and placed in the operative space for the 10 minutes. Then the rats were put into the apparatus for the analysis for the 10 minutes to acclimatize the or for we can say manipulation of the environment. Day 3 is considered as a training day, that is the postnatal day 74. As compared to or similar to the day 1 and day 2, rat were first transported to the preparing area. Then following 2 identical objects were placed in the plexiglass boxes as indicated for a rat to explore for the 10 minutes. And on the testing day, that is on the day 4, all the processes were performed to similar that of the day 3, except the one old object is randomly replaced with the new different object. And the rat were evaluated for the exploration time on the novel object and time spent with the familiar object. Furthermore, to assess the what we can say short-term retrieval memory, this is assessed in the passive avoidance test. So the passive avoidance test was performed on the postnatal day 76 to assess the retrieval of a short-term memory. So initially in the training period, the animal is performed in apparatus for the 5 minutes. The gluteal door is then closed and the animal were placed in the dark compartment. After 10 seconds, the door will be open and an electrical food shock will be delivered for the 2 seconds, and the food shock is delivered until the animal reaches the safe area, that is the light compartment. So we perform this activity repetitively until it meets the criteria, and the criteria is for 9 avoidance in the 10 consecutive.
Sorry for interruption, Mayur.
Yes, sir.
Have you changed the slide or are you going on the same slide?
Yes, sir. I am going on the same slide.
Okay.
Yes.
Okay. Continue.
Yes. So last point, which is on my slide, that is on postnatal day 78 to 81, we will perform a fear conditioning and fear extinction. So fear conditioning is performed on the postnatal day 78 and 79, and on postnatal day 82 to 81, we will perform the fear extinction. In the fear conditioning, we will couple a 10 condition and unconditioned stimulus. Conditioned stimulus is a specific intensity sound followed by a shock, whereas in the extinction, we will not give a conditioned stimulus. There is only a conditioned stimulus, not a unconditional stimulus. Immediately after the behavioral assessment, rat were euthanized, and then we will perform the biochemical estimation. So after following this methodology, we get the result. First one is the exploratory activity, as shown in the result. In the animal which is which were given ethanol during the early adolescence from the postnatal day 28 to postnatal day 49, there is significant decrease in the number of nose poking, showing that there is a decrease in the exploratory activity or neophobia in the animal which are exposed to the ethanol during the adolescence. The dose dependent effect of agmatine is seen in the animal, where agmatine 40 mg per kg and agmatine 80 mg per kg significantly increases the number of nose poke, whereas agmatine 20 mg per kg was not found significant. When we are going toward the recognition memory, the result shows that in a ethanol exposed animal, the exploration time on the familiar object is increased, whereas the exploration time on the novel object get decreased, showing what we can say recognition memory get hampered. My drug agmatine, specifically for 80 mg per kg, significantly reduces the time spent with the familiar object and time spent with the novel object. To assess the short-term memory, as we have discussed, we perform the passive avoidance test. So we calculate the escape latency. In the ethanol exposed animal, there is a significant increase in the escape latency in this animal, which is significantly decreased by the agmatine 40 and agmatine 80 mg per kg, whereas agmatine 20 mg per kg found to be insignificant. Now the following this learning and memory, we shifted toward the fear conditioning and fear extinction. So in the fear conditioning, we given a condition and unconditioned stimulus, and the freezing time is recorded during ITI, that is an inter-trial interval. So as in case of fear conditioning, there is a significant decrease in the freezing time in ethanol exposed animal during the adolescence, which is normalized by agmatine 40 and 80 mg per kg. Whereas in the case of fear extinction, there is a significant increase in the freezing time in the ethanol exposed animal during the adolescence, which is normalized again by agmatine 40 and 80 mg per kg. Following fear conditioning, we have performed the biochemical estimation where BDNF level we have checked, as BDNF is important neurotropic factor for the cognition and it is widely present in the hippocampus and the cortex. So the BDNF level is significantly decreased in the animal exposed to the ethanol, whereas it is normalized by the agmatine 80 mg per kg, whereas agmatine 40 and agmatine 20 and 40 found to be insignificant. So from my result, though we have not yet explored involvement of the receptor by which molecular mechanism or by which receptor it is showing action, but our hypothesis show that when we expose the ethanol the animal, that will lead to alteration in the glutamate level, which will act on the NMDA receptor and causes the neurodegeneration, behavioral abnormalities, impaired learning and memory, and alcohol-induced complication. My drug, that is agmatine, which is a pleiotropic molecule, it is an endogenous biogenic amine. It is it having a potent action against the neurodegeneration. It reveals the behavioral complication which is caused because of the ethanol exposure, and it normalizes the impaired learning and memory. The possible mechanism may be NMDA antagonism, reduction in the glutamate efflux, and the neuroprotection. But still further exploration is needed to check the receptoral environment and the molecular mechanism of the agmatine. So the conclusion of our study shows that agmatine could open a new pharmacological perspective for the cognitive and excuse me, and neurological complications associated with the ethanol, which is exposed during the early adolescence. So these are the reference. Thank you very much, all of you.
Thank you, Mr. Mayur. May I invite Dr. Paif to examine
Yes.
Mr. Mayur?
Thank you, Mayur, for the nice presentation.
Thank you, sir.
Mayur, yeah.
Yes, sir.
Now, what is the significance of NF levels?
Sir, BDNF is a brain-derived neurotropic factor which is important for the neurogenesis. Also, it several studies shows that its level get decreased during what we can say impairment or neurodegeneration. So BDNF is a hallmark where the cognition is impaired. So if there is a decrease in the level of BDNF, it shows that our cognition or the learning and memory ability get impaired.
Okay. Now I have one more question. Why
Yes,
you have selected that agmatine as 20 to 80 mg? Is there
Sir,
any specific
previous study which are performed in our lab that shows these behavioral abnormalities which are related with the CNS. Also, we have published paper on fear conditioning, extinction, withdrawal induced memory, withdrawal induced anxiety, and also the spatial learning task performed the MWM. Agmatine 40 and 80 mg show the significant effect. So depending upon the previous data, which is already what we can say available in our lab, we use this dose that is from 40 and 80 mg.
40 and
80 mg. Actually, 40 and 80 mg show significant effect on the various behavioral parameters like anxiety, depression, memory, fear conditioning extinction, withdrawal induced memory, alcohol sensitization, alcohol tolerance. So we use one sub-maximal dose 20 mg per kg, whether it is useful in 20 mg or kg or not, or we will directly shift toward 40 or 80 mg per
20
kg.
is the minimum, no, 20 is the not maximum. 20
yes, it
is
is minimum dose, yes, sir. So we use a sub-maximal dose, sir, sub-maximal.
Maximal dose. Okay,
Yeah,
okay.
yes, sir.
Now, can you tell me which is the statistics you have applied for this particular?
Yes, sir. One way ANOVA and two way ANOVA for following multiple comparison test.
Multiple comparison test.
Yes,
Okay,
sir.
but because you're not mentioned about that one, because
Sorry,
asterisks
sir.
also.
Yeah,
What is the significance level? What is the p value?
Star is star hash and dollar. We have used different notation. 0.05 is compared as a significant value, sir.
Yeah, that is missing actually. That's why I asked.
Legend is missing.
So final last questions.
Yes,
and what is your outcome of these particular things? What you want to say? What is
Yes,
the
sir, this
outcome?
agmatin molecule is already endogenously present in our body. It is abundantly present in the hippocampus. Several studies show that in many what we can say complications related with the CNS, there is a decrease in the level of agmatin. Also, exposure of ethanol during the early adolescence causes the behavioral complication as well as biochemical alteration in the late life, which will leads to anxious behavior, depressive behavior, or impairment of the learning and memory. Agmatin, which is endogenous, which is already present endogenous, if it level get decreased because of this condition, the exogenous administration of agmatin is useful for the treatment of what we can say alcohol and related complications.
Okay, thank you, Mr. Mayor.
Yes, sir.
All the
Mayor,
best.
what was the route of administration?
Sir, ethanol is given by per oral route, by oral gavage, whereas agmatin is given by IP route, intraperitoneal.
Haven't you thought of stereotaxic rule?
Yes, sir. Actually, we are as the amygdala is more preferable for fear conditioning and fear extinction. So we have planned next study as the site-specific administration of the agmatin in the ICV, intrahippocampal and intraagmatin. Intra-amygdala, sorry.
What is the chemical nature of agmatin?
Sir, agmatin, it is a precursor, L-arginine, it is, yes, sir, it is a biogenic amine.
It is a protein?
Yes, sir, it is amine, yes, sir.
Okay, so isn't there any threat to the chemical nature of agmatin before it enters the BBM, before it enters the brain?
No, sir. Already pharmacokinetic studies shows that its T-half get increased when it's entered into the CSF. Actually, its T-half is 8 to 10 minutes, but the fraction of the agmatin entered in the blood-brain barrier in the CSF, the T-half get increased to 3 to 6 hours.
Before entering the CSF?
Its T-half is peripheral agmatin T-half is 8 to 10 minutes.
Means it will be degraded if you give it IP.
Oral may be more what we can say complicated as compared to IP or SC.
Yeah.
At least fraction of drug entered into a brain in 8 to 10 minutes.
Okay. Oh. May I have, I'm sorry for intervening in between. May I have questions from Dr. Yadav?
Yes, sir. Like, first of all, let me congratulate Mayur. I think it was a very
sir, you are not audible.
Yeah, we have lost Dr. Kushwant Yadav. I think his voice is breaking.
The work is still in progress.
Yeah, yes, sir. The work is still in progress.
Yes. I could feel that. What I wanted to ask is, how did you decide the dose of ethanol?
Yes, sir.
Like, wasn't it a bit more?
No, sir. Depending upon previous literature, the literature of Poke et al. 2016, Salari et al. 2014, and one more paper, I don't remember it. So we have reviewed 3 different papers and validated the protocol in our laboratory, and then we will select this dose.
Okay.
So already there are established published literature which are showing that this dose is not what we can say shows mortality, and it shows behavioral and the cognitive impairment in the adulthood.
Fine. Like, another thing I wanted to know is this this molecule that you are like working on, and how will you confirm that agmatin is not present endogenously?
Okay. Agmatin
is not present, you said, because if it
yes, sir, it is present endogenously. Agmatin is present endogenously.
How will, in what cases its level might get decreased, and if that happens, how will one come to know?
Yes. Actually, sir, this molecule was established by the scientist Albert Kosher in 1910, and it is left undisturbed for the 100 years. In after 100 years, it is seen in what we can say person who are having a suicidal tendency. There is a decrease in the level of hippocampal agmatin, brain agmatin level. Sorry, specifically. So from that, this molecule get evolved. And now this molecule is also used as a nutraceutical agmatin. Also, we have performed brain agmatin level in different pathological conditions. In case of depression, in case of anxiety, in case of cognitive impairment, also in the case of FASD. Our previous protocol was performed on the FASD. It was already published paper, Kale et al. 2020. So in case of FASD, there is also decrease in the brain agmatin level. So the studies which performed in our lab from last 11 years, that showed there in various neurological complications and the cognitive impairment, neurobehavioral, neuroaffective, neuropsychiatric complication, there is a decrease in the endogenous level of agmatin.
Yeah,
Yeah.
I think Dr. Kushant was interested in knowing the symptoms of decrease in agmatin level.
Yes.
The
Actually,
detection of decrease in agmatin level.
It is seen that in neurological complication, there is a decrease in the level of complication. There are not what we can say prominent sign that there will be condition like anhedonia or apathy or other condition. But when we checked in that what we can say specific abnormality condition, there is a decrease in the level of agmatin.
Okay. There are already some works which have shown that it has got effects on the stress.
Yes, sir. Stress-related food intake, repetitive routine stress, and cognitive impairment also.
Okay, so you have gone through all of those papers also.
Yes, sir. I'm also gone through that paper also.
Thank you, Mayur. It was a very nice piece of work.
Yes.
That's it from my side.
All the best.
Yes.
All the best, Mayur.
Thank you, sir.
Okay. Let us now call upon Miss Palak Shah, the next contestant.
Hello, Chatpalliwar sir.
Yes.
She has some laptop problem, so she had
Yeah.
sent the PPT to me. I
Okay.
have forwarded to Aher, Dr.
Okay.
Aher. So he
Yes.
will going to run that PPT and she will going to present, right?
Okay.
Okay.
Hello. Yeah, the slide is available. Presentation is visible.
Hello.
Yes.
Yes.
Yes, Miss Palak, you are online.
Am I audible, sir?
Yes, you are audible. You may start your presentation.
Thank you, sir. Very good afternoon to all of you. My presentation title is The Effect of Ferulic Acid in Fructose-Induced Hyperuricemia and Associated Complications. My code is PCOL10. Please change the slide. Hello.
Yes, you are quite audible.
Sir, change the slide. Yes. This is my study rationale. Major complications which are associated with the hyperuricemia are gout, urate nephropathy, hypertension, and kidney failure. As it is shown, the dietary effects of fructose. The fructose causes hyperuricemia, insulin resistance, diabetes mellitus, hypertriglyceridemia, NAFLD, obesity, accelerated aging, endothelial dysfunction, hypertension, and IBS. Fructose itself can induce the renal dysfunctions by inducing hyperuricemia and by induction of the local inflammation. Next. This is the proposed mechanism of the fructose-induced production of uric acid. As during the phosphorylation process of fructose, while conversion of fructose to fructose 1-phosphate by fructokinase, there will be the transfer of phosphate group, and ADP will be converted to adenosine diphosphate, which will be eventually converted to adenosine monophosphate. And adenosine and guanosine and inosines are the bases, purine bases, which are eventually converted to hypoxanthine, followed by xanthine and uric acid by the enzyme xanthine oxidase. Now, fructose is only the sugar that causes the rapid rise in the uric acid. Fructose infusion causes the rapid rise in plasma and urinary uric acid. Next, please. As we want to see we wanted to check the ferulic acid effects in fructose-induced hyperuricemia. Ferulic acid is a polyphenolic acid, 4-hydroxy 3-methoxycinnamic acid. It is obtained from the rice bran, wheat grain, orange, apple, and fruits. The fruits, the reported activities of the ferulic acid are the xanthine oxidase inhibition. It is antioxidant and anti-diabetic effect. It has also anti-hyperlipidemic, anti-inflammatory, and anti-cancer activity. So in this present study, we tried to check that whether the ferulic acid is effective in high fructose induced hyperuricemia and associated complications or not. Next, please. These are the material and methods. The project was approved by the CPCSCA and the Institutional Animal Ethics Committee. Project number is this, K/per 2012-283. Animals which were used were the male sprague dolly rats of body weight 150 to 180 grams. The number of animals utilized were 24, 6 animals per group. The materials were ferulic acid, allopurinol, and xanthine, and their sources are given: Cisco Research Laboratory Private Limited, India. Diagnostic kits were from Beacon and Span Diagnostic, India. High fat diet was the reference is given. High fat diet was designed and using high fructose almost 60% per 100 gram, 60 gram per 100 gram of normal diet, which was replaced from the cornstarch. Cornstarch was replaced by the high fructose diet for 8 weeks. Ferulic acid and allopurinol, which were acting as a taste and standard compounds, were administered from day 29 to 56. These are the study groups. There was 4 study group: normal control, disease control, standard control, and taste group. Control diet was provided to normal control, and all other animals received the high fructose diet. The standard group received allopurinol 15 mg per kg, and taste group received ferulic acid 100 mg per kg. Both standard and taste were administered orally once in a day. Next, please. The following parameters were assessed. Physical parameters which were measured were food intake 24-hour and body weight on day 0, 28, and 56. Biochemical parameters: uric acid, creatinine, glucose, total cholesterol, HDL, LDL, triglyceride, and nitrate were measured on day 0, 28, and 56. Insulin test tolerance test was carried out on day 55. Liver weight was on day 57. The animals were sacrificed, liver were isolated, and they were weighed. From the liver homogenate, the xanthine oxidase inhibition, antioxidant parameters like melonaldehyde and reduced glutathione were measured. The data is expressed as a mean plus minus a standard error of mean, ACM, and one-way ANOVA was followed by post hoc test for the multiple comparison group, and p-value less than 0.05 was considered as a statistically significant. Next, please. And here are the results. So the results are showing the effect of allopurinol and ferulic acid on serum uric acid, serum creatinine, and the lipid profile. As we saw that we can see this that on day 0 and day 28, there was a significant rise in serum uric acid as compared to normal control in high fructose diet. So high fructose diet induced the increase in the serum uric acid, serum creatinine levels were increased. Then as compared to the HFD, that shows that the significant difference was found in the allopurinol and ferulic acid group on day 56. That means the allopurinol and ferulic acid decreased the serum uric acid and creatinine significantly from the high fructose diet disease or hyperuricemic group. Now the results of our serum cholesterol, total cholesterol were increased significantly in high fat diet, and they were also increased in the allopurinol and ferulic acid on day 28, but on the day 56 there was a decrease in the total cholesterol was found in the allopurinol and ferulic acid group. HDL, which is a good cholesterol, it was a significantly decrease in HFD group as well as allopurinol and ferulic acid group in on day 28. But from day 29 to day 56, because of the administration of allopurinol and ferulic acid, there was an increase in the HDL levels. Next, please. Same way, the low density lipoprotein, serum LDL and serum triglyceride levels were significantly decreased on day 56 in the allopurinol and ferulic acid treated group as compared to the high fructose diet group, in which the LDL and triglyceride groups were increased, the levels were increased. Regarding, if you talk about the serum glucose levels and insulin tolerance test, serum glucose levels were significantly increased because the fructose causes the hyperglycemia, and it impairs the insulin, we can say, tolerance, or on the day 28 and day 56, high fructose diet significantly increased the serum glucose level, and in allopurinol and ferulic acid group, on day 56, the serum glucose level were significantly decrease in now insulin tolerance test. Yes, in allopurinol and ferulic acid group significantly show the improvement. Next, please. Okay, regarding urine uric acid excretions, if you talk about, then high fructose diet has significantly increased the urine uric acids because of the hyperuricemia, and allopurinol significantly decreased by inhibition of the synthesis of uric acid, and of course the ferulic acid also significantly decreased the urine uric acid on day 56. Now, administration of the high fructose diet causes the we can say increase in the liver weight by altering the liver metabolic processes. So there was a increase in the weight of liver in high fructose diet as compared to normal control animals, and liver weight were decreased, or they were not increased that much in allopurinol and ferulic acid group when on the day 57, when the animals were sacrificed. Now, what is the effect on the antioxidant parameters? Reduced glutathione and liver melanaldehyde. So the reduced glutathione was significantly decreased in a high fat diet group, high fructose diet group, and it was increased in allopurinol and ferulic acid group on day 57 in liver homogeneity. Now, melanaldehyde serves as a parameter for the lipid peroxidation, which was significantly increased in the high fructose diet group, and allopurinol and ferulic acid showed the decreased melanaldehyde level and thus less lipid peroxidation. Next, please. Serum nitrate was also measured to assess the indirect parameter for the endothelial dysfunction. On day 56, the allopurinol and ferulic acid both show increase in the serum nitrate level, while the serum nitrate level were significantly decreased in the high fructose diet group as compared to normal control. Xanthine oxidase inhibition was measured in liver homogenate. The ferulic acid shows the higher percentage of the xanthine oxidase inhibition. So from all these parameters, what we can conclude that ferulic acid decreases the elevated serum uric acid level. It also decreases the urine uric acid levels, the triglyceride levels, LDL, total cholesterol, and triglyceride level. It decreases the creatinine and glucose. It improves the antioxidant parameters. It improves the endothelial function and through the xanthine oxidase inhibition. So the ferulic acid is effective in prevention of fructose-induced hyperuricemia and the metabolic complications which are associated with it, like hypertriglyceridemia, hypercholesteremia, impaired renal function, and the hyperglycemia. So these all were, and of course the antioxidant parameters. Next, please. Here are my references. Next.
Okay, Ms. Palak.
Yes, sir.
May I now invite Dr. Pai to examine Ms. Palak.
Okay, thank you, Ms. Palak, for the nice presentation.
Thank you, sir.
Just I want a few basic questions. Can you tell me the hypothesis of this project?
Yes, sir. Actually, sir, what the hypothesis was that that majority of the food beverages what we are taking, like cold drinks and all these things, they are containing very high amount of fructose. Except if you see the many more candies and all these things are, and the especially the fruit juices are containing very high amount of fructose. So it has been also recommended that fructose is not the juices, too much juices are not recommended because they are containing high amount of fructose. Then we checked that not the glucose and other sucrose, other sugars does not cause the much more complications problems like fructose. So it was that fructose administration was found to impair all these metabolic processes in our body. So from that, we just hypothesized to induce the fructose induced hyperuricemia. Also, we had a previous study in which we tried to check the high fructose induced hyperuricemia, and we had done it. So from that, we tried to check it, and ferulic acid had shown the beneficial effect by xanthine oxidase inhibition. And it also was showing the improvement in the antioxidant parameters. Xanthine oxidase enzyme itself is inducing the oxidative injury by production of the peroxides and other things. So we try to check the beneficial effect of ferulic acid in all the way, that whether it improves the lipid profile, improves the we can say glucose profile, and also the hyperuricemia and renal impairment and endothelial dysfunction all the way. So that's why we try to check the ferulic acid in a high fructose diet induced hyperuricemia.
Okay. That ferulic acid is available, readily available?
Sir, yes, sir, pure compound we obtain it. 98% was the purity we obtain it.
Okay. So one more question. What is the composition of high fructose diet?
Sir, it was containing methionine.
Just tell me the main ingredient, more than enough. Yeah.
It was sir fructose.
Yeah, fructose. Yeah, not fat. Yeah, fructose.
Yeah, 60%, sir. Of 100 gram diet, mein 60%, 60 gram was fructose. And in a control diet, per 100 gram diet, the 60% was cornstarch.
Okay.
The reference is also given for the high fructose diet, sir.
Okay, right. Thank you, Palak. All the best.
Thank you so much, sir.
Thank you, Dr. Pai. May I have some questions from Mr. Yadav?
Yes, sir. Yes, Palak, in your study, you've done a part of this work on ferulic acid. So suppose if this product is launched, you would like to give it as an active moiety, or you would want to give it as a dietary supplement?
Sir, actually, yes, sir. We can definitely go for, sir, add-on therapy. First of all, of course, we have to see that, because all the time we already know that nutraceuticals may be always acting as a supplementary, having a supplementary role. But what I think from my point of view, that or a drug discovery point of view, the structures like ferulic acid can be synthesized in a chemistry lab, and or directly and the compounds which are highly selective for the xanthine oxidase can be synthesized, and then they can be utilized for the, or they can be checked for the anti-hyperuricemic activity, because currently we don't have the so many drugs. Like we have only options like allopurinol, oxypurinol, yes, and febuxostate. No other options are available for the treatment of hyperuricemia. Uricosuric drugs are not that much effective. So for treatment of, and yes, hyperuricemia is associated with so many complications, because it does not, it is a very high risk factor for uncontrolled hypertension also, and renal dysfunction. So currently available drug is only allopurinol and febuxostate. So and allopurinol is having many adverse effects. So we can go for the synthesis of the drugs which are having a similar structure like ferulic acid and more potent as compared to allopurinol. We can go for that also. But just now we can go for add-on therapy and we can check the efficacy.
So what will be the dose of the drug?
Sir, the dose of ferulic acid is 100 mg per kg for animal what we selected, and it was selected from the pilot studies we conducted previously. We had selected 10, 30 and 100 mg per kg dose of the ferulic acid. From that we found that 30 and 100 milligram per kg was effective, but we selected higher dose, 100 mg per kg. And we have also gone through the literature survey that at this dose, it is not causing any kind of carcinogenicity or mutagenicity or other problems. So it was
there are already these health benefits of this ferulic acid are already known. Like for example, antioxidant is already known, and there are so many others also which are already known. So how is your study different from already existing work?
Sir, actually, antioxidant that we already know that polyphenolic compounds are usually having an antioxidant activity, but not all the antioxidants are effective in treatment of hyperuricemia. So that is the main point, and hyperuricemia associated metabolic complications are also very much important. So especially the renal impairment and nephropathy. So that was the additional thing, and hyperlipidemia. So these we try to combine all these things, not only the antioxidant parameters.
True. I think it's a nice piece of work. Thank you so much.
Thank you so much, sir.
Thank you, Ms. Palak.
Thank you, sir.
I wish you all the best for your further journey.
Thank you, sir.
May I now call upon other participant, Mr. Ahmad Ali. Mr. Ahmad Ali, if he is present, he was visible few minutes ago. I think he has lost the connection. We may move up to next presenter, Ms. Rutika Jagtap. Ms. Rutika Jagtap.
Yes, sir. Am I audible?
Yeah, you are audible. Are you ready with your slides?
Yes, sir.
I did read your question in the morning session. I have advised you to send your questions. I would like to help in your case after the presentation.
Yes, sure. Thank you, sir. Is it visible, sir?
Yeah, please change into it presentation mode.
So good afternoon, everyone. Myself, my presentation topic for today is role of embellin isolated from Embellia rice, that is one of the medicinal plants, in biofabrication of selenium nanoparticle and its study in the breast cancer nanomedicine. My guide is Dr. Shubhangi Puranik. So this is what the entire summary of my work. I have not given as such. Am I audible?
You are audible.
I guess.
I appeal to the other member who are available in the room to mute their microphones.
Yes, so as such, I have not given the methodology completely because of the limitations of the slide. So this is what the summary of work which I have put. First of all, the Embellia rice is one of the medicinal plants which I have chosen. This is collected by myself from Koyna region, Maharashtra. Then the extraction was done, and the more promising results can be by the literature we could get that the embellin is the major constituent. So I have isolated the embellin from Embellia rice, and then synthesis of selenium nanoparticle was my work. and cytotoxic activity at MCF7 breast cancer cell line was performed. Along with this, as this is only one topic of my PhD, so we are working with some other cell lines also. Here for today's work, I have presented with the MCF7 breast cancer cell line and in silico work that is related to ER alpha, that estrogen receptor alpha we have carried out. Now, why or what is the aim of my present study is actually related to the breast cancer. As we know, the breast cancer is the major cause of the cancer-related death in the woman. No doubt, breast cancer can be seen in 0.5 to 1% of male also, but it is not always with the statistic. The Globocon of WHO, they have proposed the statistic in year 2020, that is the current latest statistic, what is estimated that 2.3 million cases related to the breast cancer. And almost we can see with the 11.7% of the total cancer cases that can be due to the breast cancer. Approximately 6,85,000 women they have shown death only in the 1 year, that is 2020. This treatment, we are having a number of treatments, those are available for breast cancer, involves the surgery. Now, previously the surgery was mastectomy, that was completely removal of the breast, but nowadays we can have the different methods by which the entire breast can be, we can have the breast conservation in the female, because it is related with some emotional issues also, those are related by complete mastectomy. So we can have the lumpectomy nowadays. Radiation therapy, yes, the gamma rays can be utilized for chemotherapy. There are n number of chemotherapies are available for so many different cancers. As we know, there are having the alkylating agents, or we can have some another targeting compound. So chemotherapy can be given over here, and the targeted one, yes, there are monoclonal antibodies which are available. One of the example is the cetuximab, which actually targets with the estrogen receptors. Okay, now there are many hurdles which we can see, that the non-specificity, a high cost involvement, reoccurrence, or the metastasis conditions of the cancer, and the, of course, the side effects which are involved in all the cancer therapies. Because of that, the medicinal plants can be options, or rather than medicinal plants, we can say the phytoconstituents can be applicable for treatment of various diseases. And we know that the paclitaxel is one of the compound which is taken out from the medicinal plant itself, and which is already in use for breast cancer treatment. Now, another one is the camptothecin, which is a natural alkaloid that is also targeting with the topoisomerase 1, that means the targeted therapy. So these natural compounds, as they are already in the use in the market or in the study, we can say. One of the plant is the Embellia ribes, which is popularly known in Maharashtra as a Vidanga or Vaudinga, mostly utilized during the pregnancy or the post-pregnancy. The woman is given water of this Embellia ribes because of its anti-helminth properties, and it is showing so many different roles in the as like anti-helminth, antibacteria, wound healing properties, and many more, as like anti-cancer is reported. So embellin is one of the major constituents which plays a major role in the medicinal activities. Now, another part of my study, we have also done with the LCMS study of different extracts, and there also we could find out not only the embellin, but there are few more compounds which are also involved for the anti-cancerous activity in this plant. Now, selenium nanoparticles, the once we have chosen for synthesis of the selenium nanoparticle, the reason behind is the nanoparticles, they are involved in the drug delivery system. Because of the nano size, as they range in the dimension of 1 to 100 nanometer, those can be endocytosized by the cells. In the morning also, sir have given a explanation related to why nanoparticles can be a good medicine, can be utilized in the medicinal way, that they are having the drug carriers, or they are having with a high stability capacity, high carriers capacity, feasibility of incorporation of both hydrophilic and hydrophobic substances. So we can have the various ways by which the nanoparticles can be administered in the body. Selenium is one of the essential trace minerals which can be incorporated in the proteins to prevent the cellular damage. Now we know that it is having the mechanism in the cell cycle arrest. By the literature survey, I could get this antioxidation and the apoptosis properties, even interruptions of the cell signaling pathways, and it is showing the less toxic to the normal cell proliferation. So I have chosen selenium as a synthesis part. There are for embelin, it is also reported with the silver nanoparticle. And here in my current study, I have taken with the selenium nanoparticle. So this current study is actually based upon the isolation of the embelin and its biofabrication of selenium nanoparticle. In silico study was done with the ER alpha uh, receptor, and we are having one PDB, that's structure I have taken from the PDP, and MCF7 breast cancer, where I have done with my in vitro MTT assay. So before coming towards the result and discussions, I would like to go with a synthesis. First of all, the isolation or the extraction of the embelin. So extraction was done in the N-hexane. As with the literature survey, we could get that it is having the most solubility with the non-polar solvents like hexane. With the solvent extraction, solvent extraction method, we have done it for 10 gram of Embellia ripes fruits with a 50 ml N-hexane and 4 hours Soxhlet apparatus. The residue was collected after distillation, and it was dissolved with dichloromethane and methanol in 1:2 ratio. And after 24 hours, we could get with this crystalline structure. This crystal structure was confirmed that whether they are embelline or not with the help of biochemical test, and it has shown with the positive way in that biochemical test with the PET ether and ammonia solution. It has shown with the purple color formation. Also, now the FTIR analysis have done where it could match with the literature that yes, embelin is present. One more is what NMR study that is remaining, that we will be carrying out. Then the synthesis of selenium nanoparticle. The selenium nanoparticles were synthesized by sodium selenate, and for 5 days analysis, the ranges we have checked that when we could get with the selenium nanoparticles to be synthesized. Then those are, yes, the characterization of the selenium nanoparticle has been done. So the UV spectroscopy, we could get the peak at 374.5 nanometer, which is in the UV range, and we could get the synthesis way. Then the particle size analyzer, we have done with the particle size analysis, and it has seen with a very good result, because previously reported study for the nanoparticle through embelin had shown in the range of 30 to 100 or more than that. Yes, we could get with this. In my study, I could get it in between the 20 to 30 nanometer that the particle size has been seen. Then the scanning electron microscopic study, that it has also given the uh, characterization for the nanoparticle. The X-ray diffraction study have two theta values, and those could the two theta values could give with the explanatory way that the particle size with the calculation with the different calculating methods we could get with a nano size. So here also the confirmatory, the various nano size were also seen by the X-ray diffraction by 45 nanometer, 96 nanometer, as well as I have seen with the 103. So it is in the different ranges that we could get. Now the cytotoxicity study, it is done by the MTT assay, though I have done in the triplicates, but due to maybe some issues, I couldn't get a good prominent result. Now the repeating study will be carried out. So here I have just done whatever the study, see, before 1 month only I have done with this. So I have put over here. So the concentrations were 25, 50, 100 and 200. Why we have chosen with the MCF7 breast cancer cell line? Because my further analysis is depending upon the in silico study. If I have taken with some another cell line where it could be showing with a triple negative breast cancer, which is showing the ER negative, HER2 negative, and the progesterone negative receptor, which cannot be utilized for the in silico analysis, and that's why the MCF7, which is widely utilized, and it is the ER alpha positive cell culture. Those cells, they show the estrogen receptor positivity, and that's why I have chosen with the MCF7. Now, while carrying out with the MTT assay, why we carry out with the MTT assay is what the live cells, or we are saying the viable cells who are having the active cellular oxidoreductase enzyme, those are dependent upon the NADPH, and they reduce the tetrazoleum di MTT to the insoluble formation, which could give with a purple color. Now, whenever the enzyme is going to be converging into the water insoluble formation crystals, then we can get the number of live cells over here. The DMSO is used so that dissolve the insoluble purple formation product into the colored solution. Now, even my nanoparticle, the selenium nanoparticles which we have done, we have dissolved it into the DMSO. Now, the reason behind choosing the DMSO as a vehicle, because most of the study they have shown the rate of absorption of the compounds with the biological tissue, and one of the good paper that is present, I mean published by the Perk et al, they have shown that it is the DMSO, though it shows the, excuse me,
Sorry.
no,
Sorry.
yeah, may I continue?
Yeah, you may continue. Yeah, you may continue.
So DMSO, there are many questionnaires that why chosen with the DMSO, that it may be toxic, but the study is showing that the DMSO is toxic at a very highest level. And here I have used only 100 microliter of the DMSO. And yes, for the control, we have also used DMSO, and to check with results so that we can have the comparison. So carrying out with the MTT, the cells were first cultured in 96 well plate for 24 hours. And once the cells got settled into the 96 well plate, then the nanoparticle, as those are dissolved in the DMSO, they have done the weight 50, 100 and 200 microgram per μg per ml. 10 microliter of this dilutions, they were added into the wells, and wells were also, the control were also set for. After the drug, or we can say the nanoparticles addition, the cells were incubated for 48 hours. After 48 hours, the 10 microliter quantity was loaded in each well, and to prevent it from the photo damage, we have wrapped it into the aluminum foil and incubated for 4 hours for next into the CO2 incubator. And then carefully the supernatant was removed, and 100 microliter of DMSO was dissolved, was added. Once that has done, after 20 minutes, we have taken the readings at 515 nanometer. As per the percentage viability, we can see over here in the control cell, the viability was 100%, and as it is the dose dependent, it is showing the viability that is decreasing. IC50 value was calculated. Now, the major part, what I want to show over here is in silico analysis using the ER alpha receptor. Now, ER alpha receptor, why to choose with the ER alpha only? There are many ways by where the embelline is going to be embedded and the embelline is showing its result. But why I have chosen this is in the breast cancer, the major, the reason behind having the breast cancer in females is what mostly estrogen treatment after the menopause, mostly the to reduce the post-menstrual complications, post-menopausal complications, the estrogen or the hormone therapy is given, and the symptoms whatever provided or the female is suffering with a post-menopause complications. So to relieve her from all those complications, the hormonal therapy or the estrogen therapy is given. Once the estrogen is completely, the therapy is given for a very longer period, then what happens is there are two ways by which the estrogen can damage. So the first of all, it generate the DNA damaging free radicals, and another, it can be having the estrogen, it proliferate the cells in a cancerous region. So the major pathway where we are targeting, or my possible hypothesis that yes, my drug can work about over here is the estrogen is showing one pathway. It binds the estrogen as a ligand, it binds to the estrogen receptors, which are controlled the cell surface. Then the receptor dimerization is possible. The dimerized receptor and this ligand complex, they translocate to the nucleus, where they react and they activate the target gene expression. Once they are activating the target gene expression through the estrogen receptor elements, then what happens is the transcription factor, they are another genes they are going to be proliferating, and the genes they are expressing, and that result into the more and more proliferation of the cell. So there are few drugs which are under study, and also there are like we are having the transumab that is targeting into the HER2 region, HER2 positive breast cancer. Also we are having with the cetuximab which is targeting with the this ER alpha receptor. Here I have chosen that yes, we can carry out this. So possible way is what? It will be selectively binding towards the estrogen receptor so that it can antagonize the effect of estrogen receptor activation. If the embellin binds to the estrogen receptor, then it won't be allowing the estrogen to bind over there, and the next proliferation pathway can be we can hamper with that. So tamoxifen treatment is in the similar way which is given for the ER alpha. Now there are two receptors ER for the estrogen receptor. There are two we can say types, alpha and beta. Alpha is more prominent. 80% of the cells they are going to be expressing with the alpha one. That's why we have chosen. From the PDB we have taken the 4xI3. This is what the code for the receptor for where we have carried out with the binding. Now the docking was done by the Pyrex AutoDock Vena virtual screening software, and the interaction study, as the Pyrex study could not give with the, it could give only with the binding energy, and if we want to get with the more affections or the interactions to be studied, so those are given by the Discovery Studio Visualizer Admit Prediction. Now interaction of the standard drugs, here I have taken, as we have to compare it with some standards, so I have taken with bezodoxepine. That structure is this bezodoxepine with 4xI3 that we have docked with, and it has shown the hydrogen bond interaction. This is what the 3D image and this is what the 2D image. So what we can see over here is we can have with the arginine aspartate, or here the pi interaction, the phenylalanine 404. These interactions could be shown by the bezodoxepine. So bezodoxepine, it is what the already present for the breast cancer, and according to that we have checked its activity also, what binding or what docking it shows. According to that we can compare with the embellin structure. Now why we have to go with standard drugs? Because it can give at least that yes, my drug can be in future, it can be utilized for the further study. So we are carrying out with this. It has shown with a different interaction, and when we see the interactions details of the embellin, now these are what the highlighted one in the red color. Those interactions they match with the bezodoxepine interaction, like in the previous slide. Yeah, so in the previous slide, these interaction, even the phenylalanine interaction, the same I have shown over here. These could be matched with bezodoxepine. That means I'm not saying that 100% interaction is equal with the bezodoxepine, so my drug is too much effective. It shows the binding energy is -7.2 and the bezodoxepine 7.2. There is a difference, but still in future we can have the synthesis, or we can have such a modulator synthesis with embellin, and those can show with the they can show with the anti-estrogen effective effect. So here the docking, here we can see with the different amino acids which are targeting with the embellin, and the ligand and the receptor binding is shown by this slide. So the conclusion is the use of nanotechnology. It offers the possibilities, the exciting possibilities, so that we can target to the tumor-specific molecules. The nanoparticles can concomitant improve the efficacy and decrease the toxicity with the existing novel anti-cancer agent. So there are two research paper which I have got where they have used the embellin with another present this anti-cancer drugs and their effect, how it is showing, whether it is showing the synergistic effect, whether it is decreasing with the toxicity. That paper is with us. Embellin derived selenium nanoparticle could show promising dose dependent results in the MCF7 breast cancer cell line. And in silico approach now opens the new gates like directly targeting towards the in vitro studies or in vitro studies. At least with the in silico study we can get whether it shows the binding or not. Here in this interaction it has shown with the estrogen receptor alpha their binding, and it can have the anti-estrogenic activity of the membrane. So the future aspect, the work which I have to target at the very first year of my PhD, only this much study I could do, and my future aspects are the molecular and cellular pathways involved in the anti-cancer treatment and the apoptosis mechanism. So first, if once we are getting the molecule, yes, it is showing some with the MTT assay or some other assays like SRV, we can get with the result. Then what we have to go with whether it is targeting with the kinases or the caspases or some other enzymes involved in the pathway of anti-cancer, that I have to study. Even the Western blotting for the protein expression will be studied further. And another very important is the anti-metastasis activity that we will be carrying out with the wound scratch assay, which may show whether the apoptosis is done in this way or whether my drug is going to be showing or it will be also affecting about the metastasis cancer. Hemolysis study, as we know, the toxicity of a nanoparticles can be raised a question that when you are using into the nanoparticles, then whether they are going to be toxic or not. So hemolysis, RBC hemolysis, the SHIP RBC hemolysis is the way by which we can say that whether it is toxic to the the RBCs. If it shows the hemolysis, that means we can get a toxicity over here. And the cytogenetics, that means I would like to study over here that whether it will be working upon the chromosome or not. So this is the entire study. Thank you. These are the key references.
Thank you, Ms. Rutika. It was a very exhaustive talk over whatever work you have done. May I invite Dr. Pai to examine first?
Okay. Thank you, Ritika, for the nice presentations. Now I have one basic on 2-3 questions. You have selected N-hexane as a solvent. Yes, right?
That's what my
That
question is.
For
N-hexane.
isolation, yeah. N-hexane, that is for isolation of embellin, I have said.
Okay,
From
you
the
are
plant
isolated
or
yes.
From plant, Embellia ribes, I have taken the extract of the hexane, and from that embellin was isolated, and the further study was embellin loaded nanoparticle, or we can say embellin as a nanoparticle.
You would have taken directly embolin, no? Why you have taken the extract?
That's what the problem is.
You could have taken embolin directly. Yes.
That is sigma aldrich embolin you're talking about? Ready?
Yes,
yes.
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 See, extract means there will be other components. Other components need not be
hundreds
only
of
embolin.
compounds.
Yes, I would like to say over here is what we have done is one study is from the isolation of the embellin, and another study where I have taken the extract, that is ethanolic extract. Then from ethanolic extract, we have synthesized Embellia ribes derived nanoparticle. The LCMS study has been done. In many papers, they are saying that in ethanolic extract, we can get the embellin, but we have repeated the LCMS study twice and we couldn't get embellin. Then I have also got another literature where the embellin in LCMS was not reported. So my study is like embellin and its comparison with the another compounds which we can find out into the ethanolic extract. So yes,
Okay,
you are right that it is not only the embellin or why I couldn't get with the
yes, because in silico you had done the in silico experiment. In the in silico experiment, you have taken embellin as a pure compound, right? Structure of the embolin.
Yes,
You have docked with the embolin with your PDB, that means protein. From the protein data bank, you have selected the ER alpha receptor protein, right?
4xi3.
Exactly. So, see, you have done the reverse order. Normally, we'll go first silico, then afterwards formulation. Afterwards, you establish the activity, then you will go for the formulations. But in your case, you have done the nanoparticle first, then afterwards in silico. Can you justify this?
Sir, for this was the addition for my study.
No, whatever it may be, that's correct only. But it's correct. No, see what will happen. No, see here. Now you are doing the nanoparticles. You already prepared the nanoparticle. You already established the cytotoxic of the nanoparticles. Then afterwards, you are doing in silico. You are saying in silico means you are establishing embolin is having some effect on alpha estrogen receptor alpha receptor, right?
Yes.
Yeah. that I don't know how you're going to justify. Just you make it clear, okay?
Yes, sure.
Yeah, that is the one of the
Sir,
things.
first of all, my objectives, those were set, and I wanted to go with some MTT and related to apoptosis study with the annexin PI pathway. But this was the addition to my study.
No, whatever it may be, addition means you have to modify it, you have to present in such a way that it suits
Yes,
the
sir.
presentation.
Yes, sir.
Because what will happen? No, now it is a reverse pharmacology.
The
Okay.
addition to the study has made a lot of lacuna.
Okay. Okay, I'll improve, sir.
Yeah, because the conclusion third, you are telling about the embolin, only embolin. In conclusion point number 2, you are telling about the nanoparticle emboli. Okay, that is the one of the things. That is not matching. That's what. Okay, that is my observation. Okay, you may be right, but this is my observation. Okay, and one more last question. And how will you ensure that embolin is not effective for the triple negative cancer?
Sorry, I wouldn't get.
How this your main component is not active against the triple negative cancer?
So there
Breast
I will
cancer.
not go with the receptor study. I have to go in the way for the chemotherapy, because for the triple negative cancer, chemotherapy is mostly justified, or it is mostly given because the related to this hormonal study is not given, or
No. I'm talking about, okay. Just find out about the in silico also. Further, you also can do the in silico.
In silico. Okay,
Yes.
okay, surely.
Just find out and do it. Okay, that's all from my side. Thank you.
Surely, thank you, sir.
Okay, thank you, Dr. Pai. Dr. Yadav?
Yes, sir. Yeah, that's a nice piece of work, and I've seen you've done a huge number of things. I only have one query in the MTT essay.
Yes, sir.
In that, why have you not taken the concentrations slightly more wider? And ideally, then how did you finally conclude the IC50? Because in the concentrations that you have taken, you've gone for, I think, 25, 50, and then 100.
Yes, sir.
You have not even reached actually the 50% inhibition. Then how did you come to the fact of IC50 value?
So that actually I have calculated through the way of better, like basic calculations. But yes, I got your questions. Now another way is what I can modify the concentrations. This was with 2-3 literature survey we have done, and the basic study I have carried out with the 25-50. But I can go with 10, 20, 40 in that way.
No, ideally you should have gone for a logarithmic values increase. For example, you could have started with 1, 10, 100, 1,000.
Okay.
Because that will definitely give you the IC50 value. 10, 20, 50, how will you know?
By the literature survey, I mean, I just answered. I have with the embolin, I could get with this literature also. Where
Yeah, we have always stated that understanding literature is very important. Understanding the literature is very important. Yeah, that's what. There's a lacuna in the work that you have shown some slides over of the CADD. What I observed from the slide is the docking of the force of attraction, establishment of force of attraction between the drug which you have shown, between the molecule, your molecule under investigation which you have shown, with the binding with the receptor is quite amazing type of presentation here, because it requires the molecule to approach the distance between two sites over the molecule as well as the amino acid residue should be less than 2 angstrom. It should approach that much, and the minimum distance which you have shown is 3.9, that is nearing up to 4 angstrom per unit. Okay, so there the drug binding with the receptor, that fact cannot be established.
You are talking over here, right?
Yeah, I'm talking about
Yes.
this table.
3.
It should be near about 2.
2 angstrom. Yes.
Yeah, so here also it is missing. The other thing which is mismatching is the statement that it is extracted in n-hexane. If it is extracting in n-hexane, means it is a totally non-polar compound, whereas the structure of embolin, the compound which you are stating, is quite polar. And if it is polar, then only it will interact with the AF2 mode, AF2 part of the receptor ER alpha.
Okay.
AF2 domain, that is the ligand binding domain, which is differing in ER beta, which is the difference between ER alpha and ER beta. You have stated that it is 80% expressed in the cells, but the difference is the specific difference is AF2, and that AF2 is having a lot of amino acid residues which are polar, such as you are shown in the list of binding with BAZ, basiloxifene, ASP351, arginine 394. These are all polar amino acid residues, and the molecule which you have shown is non-polar. So it cannot be digested that this molecule will bind with the amino acid residues in AF2 region of domain of ER alpha. So that makes this presentation a very weakly planned or a very meek presentation. Anyway, I wish you all the best, Ms. Rutika, for your presentation. I may call upon the candidates who have missed their chance because of technical difficulties. Is Mr. Mehul Chorawala available and ready?
Yes, sir. Yes,
Are you
sir.
able to present?
Yes, sir. Yes, sir. So should I present my screen?
Yeah.
Is it visible?
Yes, sir.
Thank you, sir. So, good evening, all of you. And today I'm going to present my research work on the cell wall contents of probiotics, specifically Lactobacillus acidophilus, that inhibited the lipopolysaccharide-induced colitis in the rat by modulating the immunoinflammation and oxidative stress. Hello.
Yes.
Yeah.
You're audible.
Slide is moving or hello? The slide are moving or yeah. Okay. Thank you, sir.
Yes, moving.
Yeah, thank you, sir. So we all know that inflammatory bowel disease is a chronic immunoinflammatory and oxidative stress condition, which is generally arise from uncontrolled and dysregulated immune response to the host enteric microbiota. And it is well established due to the involvement of enteric microbiota, the gut, TLR4 and LPS axis are responsible to cause immunoinflammation and oxidative stress. And generally, currently available therapy like sulfasalazine and steroids are poor responsiveness. Rather, they are only targeting immunoinflammations, but not targeting oxidative stresses. So, need for inexpensive and safe alternative therapeutic strategy. And we all know probiotics are just a health benefit organism which do not produce any hazardous effects. So they Hello. So probiotic.
Hello, please put your microphones to off. You are distracting. Yes, Mr. Mehul, you can continue.
Yeah, thank you, sir. So probiotic meet these criteria, and they are nowadays they are always used as a supportive therapy, and specifically Lactobacillus species. They are supportive therapy for many gastrointestinal disorders. So, however, the direct cell wall component of Lactobacillus acidophilus never be tested, and their activity proved antioxidant, anti-inflammatory, and immunomodulatory, and therefore this might produce a beneficial effect. So my hypothesis is these TLR4 and lipopolysaccharides. So when lipopolysaccharides and TLR4 are bind, it will produce a cellular response and damaging the interstitial mucosa, recruitment of various inflammatory cells like neutrophils and macrophage that may lead to the release of pro-inflammatory cytokines as well as release of reactive oxygen species and other active oxidative stress markers and inhibition of the antioxidants defense system or boosters. So that way provoke the immunoinflammation, oxidative stress, and ultimately amplification of whole vicious cycle will be their continuation, amplification of this whole vicious cycle and damaging to the mucosa, and that will lead to the colitis. Now, my target is to block the TLR4 receptors using cell wall content of a lactobacillus acidophilus. Actually, whole organisms or live bacteria or killed bacteria are generally used as a supportive therapy, but no one has tested the cell wall content of the lactobacillus acidophilus till date. And we are targeting directly to the systemic use of the cell wall content without affecting the local microbiota present in the gastrointestinal tract. So by blocking the TLR4 receptors using the cell wall contents of the lactobacillus, we'll try to block these both pathophysiological mechanism, release of pro-inflammatory cytokines and reactive oxygen species. So treatment with this cell wall content of lactobacillus acidophilus, inhibition of immunoinflammations, inhibition of oxidative stress, and boosting the antioxidant activity might be possible, and that we have proved in our work. So the experimental design for our study is actually we have used a novel model that we have been established in our lab, and that has been already published in antimicrobial protein probiotic and antimicrobial protein journal. We have already published that work. Here we have induced colitis by sensitizing the subcutaneous injection of lipopolysaccharide to rat, and then we have boosted the animal with the half dose of the lipopolysaccharide, and later on from 8th day to 21 day, that is a duration of total 14 days, we have induced the colitis by intrarectal administration of lipopolysaccharide 500 microgram per rat. These all the doses are irrespective of the body weight. We have just injected to the individual rat. Now consequently, when we started the lipopolysaccharide administration, we have also started the treatment with standard control drug like sulfasalazine that we have provided per oral for local purpose. Here the sulfasalazine already established drug for the inflammatory bowel disease, and the dose is already established and given oral route, while our cell wall contents of a probiotics which have different doses regimen. One is obtained from the 10⁶ CFU, one is obtained from 10⁹ CFU, and one is obtained from 10⁻12 CFU. Now in model control, these all standard cell wall and different treatment groups are receiving the lipopolysaccharide for continuously 21 days. Similarly, model control group also received the same, but normal control group didn't receive any lipopolysaccharide. They are just treated as a normal control and injected with the saline subcutaneously. And we have measured different parameters, specifically body weight, food intake, rectal bleeding, stool consistency were measured at alternate day, and colon length, inflammation marker, oxidative stress marker, histopathology done at the end of the study. So I used a one-way ANOVA or two-way ANOVA for normal distributed data, followed by the Bonferroni test, and Krusell-Wallis test, followed by Dunnett test for the non-normally distributed data, and P less than 0.05 was considered as statistic significance. Now come to the result part of our study. Here cell wall contents of lactobacillus acidophilus improve the disease activity and attenuate the macroscopic colon damage in LPS induced colitis. So what happened? We all know that when colitis occur, there were naturally a disease severity is always increased, and that disease severity is measuring the change in a body weight, change in a food intake, and disease activity indexes. So let's start with the food change in a body weight. As we observed, the lipopolysaccharide gradually decrease the body weight up to 22 days when we started the lipopolysaccharides. However, body weight is also decreasing, and simultaneously we have also observed the food intake with this group is also decreasing. Now this will directly correlate with the disease activity index. Now what is disease activity index? Disease activity index is actually the average score of three things: decreasing in a body weight, decreasing in a food intake, as well as it's a stool consistencies and everything we have seen and given the score. So that score that I will show you last, how we have scored the disease activity index that we have tabled From that, we have measured the score, and we can see that disease activity index is increased here in lipopolysaccharide treated rate. However, when we started the treatment with the sulfasalazine, the blue mark or blue asterisk, it can be shown that it will inhibit the gradual reduction in the body weight. And similarly, lactobacillus acidophilus is also highest dose of a lactobacillus acidophilus, that is a cell wall content obtained from the 10^12 CFU. This will 10^9 CFU, and this is a 10^6 CFU. So highest, actually all are producing significant effect, but the highest activity is observed with the 10^12 CFU cell wall content, and therefore the lactobacillus acidophilus specifically at 10^12 CFU dose, it improved the disease severity. Now, having observed the improvement in the disease severity, we next go for the macroscopic characteristics of a colon. So in macroscopy, we have measured different parameters, but here I mentioned only colon length. Actually, due to the some PowerPoint number of a slide restriction, I only presented colon length here. Otherwise, you measure colon length, colon weight to length ratio, and spleen weight. So here only colon length data are there. When animals are administered with the lipopolysaccharides, there is a decreasing in a colon length more than 70%, 60 to 70% fall in a colon length was observed. However, the pre-treatment with the sulfasalazine inhibited the decreasing in a colon weight. Similarly, colon weight to length ratio also getting increased. And when we treated the animal with the cell wall content, the highest dose, that is a lactobacillus acidophilus from the 10^12 CFU was producing highest effect. And when we compare these things, that this is also significant to the model control. However, the sulfasalazine showing prominent effect as compared to the highest dose of a lactobacillus acidophilus cell wall contents. So next we observed that as there is an increasing in a severity of a disease in lipopolysaccharide group, and the rat which are treated with the lactobacillus cell wall, they are showing inhibitory. So we next go for the inflammatory response, because inflammation is an important pathophysiological characteristics for the ulcerative colitis. So to determine whether these improved colitis with sulfasalazine group as well as cell wall of lactobacillus group, we measure the inflammatory parameters like pro-inflammatory cytokines in plasma as well as colon homogenate. So when we it can be easily clear that when lipopolysaccharide is given only to the model control group, they are showing significant rise in all pro-inflammatory cytokines marker like interleukin, TNF alpha, interleukin 1b, and interleukin 6. However, when treated with the sulfasalazine, there is a nearly 54 to 60% down in pro-inflammatory cytokines observed. And similarly, the highest dose of cell wall content nearly showing the similar effect to that of a sulfasalazine. However, the 10^6 CFU was not as effective as 10^12, and it is generally significant. The sulfasalazine as compared to sulfasalazines, it is insignificantly different. No significant difference was observed. And similarly, all these parameters, interleukin 1b and interleukin 1/6, showing similar protections or inhibiting the inflammatory process during the progression of a colitis. So same result is also observed with colon homogenate. Hello. Hello.
Yes. You may continue.
Hello. Should I continue?
Yes.
Okay, thank you, sir. So similar result was observed in a colon homogenate, where we have isolated the colon at the end of a study and make a homogenate, and in that homogenate we measure various parameters like a TNF alpha, interleukin 1b, and interleukin 6. and similar result as we observed in a plasma. That same kind of a result has been observed in our colon homogenate. Now in parallel, so in parallel, we also observed the C-reactive protein. We all know it is an active phase protein that indicate the systemic inflammation. So in parallel, we observed the C-reactive protein, and similar rise in a C-reactive protein was observed specifically in a model control group where they are only injected with the lipopolysaccharides. However, the rat who those received the lipopolysaccharide as well as treatment with the sulfasalazine and sal wall content of lactobacillus acidophilus, they are showing the decreasing in a C-reactive protein level. The level in a C-reactive protein, the decreasing in a C-reactive protein level was gradual as the dose is getting increased with the lactobacillus acidophilus cell wall contents. Now, next we move on. As the inflammation is there, we all know that inflammation mainly because of the recruitment of inflammatory cells. So here we measure the recruitment of inflammatory cells, specifically of neutrophils, because these recruitment of cells, inflammatory cells specifically at the site of injury or site of colitis, are measured as a MPO activity, myeloperoxidase activity, because myeloperoxidase enzyme is generally secreted from the inflammatory cells like neutrophils and macrophage. Neutrophil is a acute phase response cells, while macrophage is a chronic phase response cells. So here we have observed that these myeloperoxidase activity is also increased at the end of a study in model control group, and the treated rat, sulfasalazine treated rat, as well as the treatment with the cell wall content of a cell wall content of a lactobacillus acidophilus, showing protective effect and showing inhibited the recruitment of the inflammatory cell at the site of injury. Now, next we targeted that this lactobacillus acidophilus, as we know that inflammation can modulate the oxidative stress reactions. And we know that as inflammation is getting increased, oxidative stress are getting also increased due to the release of several oxygen species, release of several free radicals and reactive oxygen species. So similar result we have observed. Sorry, actually I want to show you the we have measured the oxidative stress marker. Due to the inflammation, there is a rise in oxidative stress marker. So we measure the malondialdehyde and nitric oxide as a oxidative stress marker, and oppositely, exactly, we have measured reduced glutathione, which is getting decreased because it is an antioxidant. So we have observed that when the rats were treated with the lipopolysaccharide dogs, similar to the inflammation and parallel to inflammation, oxidative stress markers are also increased, while antioxidant defense systems like reduced glutathione are getting decreased. So this indicates that our model is also giving immune inflammation and oxidative stress. Now, same the treated LPS treated animals and LPS injected animals when treated with the sulfasalazines as well as cell wall content of lactobacillus species or acidophilus, specifically we have used acidophilus. So it can be shown that there is a gradual decreasing in a oxidative stress marker in the both parameter, nitric oxide as well as malondialdehyde, and sulfasalazine is most potent in this condition. And on the contrary, the reduced glutathione is getting increased with the sulfasalazine as well as highest dose of it is increasing, but it is no significant difference between these disease control and the cell wall content of obtained from 10⁶ CFU. However, the 10⁹ and 10⁶ showing significant effect as compared to the model control group. This indicate that our lactobacillus species treatment boosted the antioxidant defense systems. It has boosted the antioxidant defense system. Now, later on, we have to take a histological examination. As we know, histology is most important part of any studies when we have no actual proper parameter for the observing the things inside the body. So we have taken the histopathology of a colon, and we can see this is a normal rate which we have injected only saline. So they do not show any disease severity, and all the cells, these are the goblet cells, and that these are the crysts, and the dotted cells, they are the neutrophils. So they do not show any severity. However, the animals which are only treated with the lipopolysaccharide, the integrity of globulate cells, integrity of cyst and neutrophils are also recruited highly. However, the treatment with the sulfasalazine, improve the condition of the colon and restore the globulate cells, make an integrity of the cyst, and there are less number of neutrophils are getting also recruited. However, the cell wall content of a lactobacillus acidophilus obtained from the 10⁶ CFU are not producing any significant effect in the therapy. However, the 10⁹ and 10⁻12 CFU cell wall content producing significant effect, not as equal to sulfasalazine, but it is providing protection against the colitis. So in last, the conclusion, our study is producing the evidence promising protective effect of we are providing protective effect. We didn't check for the post treatment, so it is just a protective treatment for the cell wall content of acidophilus in lipopolysaccharide induced experimental colitis and favorably mediated by immunoinflammation and oxidative stress. However, the further studies are needed to confirm the exact mechanism and signaling network which are occurring through TLR4 and LPS axis. These promising the cell wall component and suitable dose with administration route, different routes of administration can be further studies. Here we have used subcutaneous dose of cell wall contents. So different route can be set up, then different dose can be set up, and we'll check the effect of the cell wall content of probiotic organisms. And as I told you, we have to do the disease activity index. This is my published paper already, and we have to follow these things for the giving a disease activity index, that loss of body weight, stool consistency, and rectal bleeding, which are the 3 parameters. The average score of these 3 parameters can be considered as an indicator of a disease activity index, and these are the score which we have adopted from these Sigmunds and Patel et al. So this is all about my presentation. Thank you, sir.
Okay, Mehul.
Yes,
Mehul,
sir.
can you hear me?
Yeah,
thank you.
Yes, sir.
Yes. Can you tell me in one simple one sentence or few sentence, what is the probable mechanism that you are proposing for this particular?
Sir, actually, we are working on the Western blot right now, and we are targeting the myeloid dependent 88 pathway, which are modulating through the TLR4, and that actually TLR is well reported for inflammation and oxidative stress, but in between pathway, which are myeloid dependent 88 pathway or other pathway are there, that NF-kappa B signaling is there, that we don't know. So we are currently titrating for the pathway using Western blots. So most probably via myeloid dependent pathway, because we get some result in that things.
Thank you. Thank you for nice presentation.
Thank you, sir. Hello.
Yes, Dr. Yadav.
Yeah, I just have one query. I mean, it's very nice work.
Thank you, sir.
Like other than the like already known things about the probiotic, why do you think that there is this need of working on the cell wall contents? Otherwise, also, being probiotic, it works.
Yeah. Actually, sir, generally people are using whole organisms or intact live bacteria or killed bacteria, and they are given by, generally these probiotics are given by oral route, and they are manipulating the local gut microbial flora. And our target is to systemically affect, actually targeting gut microbiota and disturbing that gut microbiota that may be harmful using this cell wall content. So we want to systemically target the TLR4, and that's why we use actual cell wall content which are responsible for the modulatory effect on inflammation and oxidative stress. And here we want to modulate immunoinflammation. So that's why we selected the root subcutaneous.
Fine. I mean, that is, I think, something surely different, novel. Thank you so much.
Thank you so much, sir.
Thank you, sir. And thank
I
you,
have one
Mr.
question.
Yes, sir.
Yes.
Hello.
Hello.
Yes, yes.
Just tell me, what is the significance of estimating myeloperoxidase, and in which part you have performed this myeloperoxidase?
Myeloperoxidase is an enzyme which are generally secreted from the inflammatory cells like neutrophils and macrophages. So when inflammations occur and neutrophils and macrophages are recruited, they generally release the myeloperoxidase as a site of injury, and we have major myeloperoxidase in the colon homogenate.
Okay, I think it also has some role in oxidative stress.
Yeah, myeloperoxidase always, as inflammation is going on, myeloperoxidase is activating the free radical systems. So that's why consequently next parameter was targeted on the oxidative stress.
Okay, thank you.
Okay, thank you, sir.
Thank you, Mr. Mehul.
Thank you so much, sir.
Okay. Miss Lipi Joshi, who has missed her chance when she was called, is now available, and she will be presenting her topic. The judges may be informed that this is the last presentation today, and in fact it is the last presentation for the competition. Other presenter, we have missed one presenter. He has not reported, nor he has been seen in the class. So Miss Lipi Joshi, are you available? Are you ready?
Yes, sir.
Okay. It will be nice if you could please share your slide.
Yes, sir.
Mr. Mehul, please mute your microphone.
Sir, is my screen visible?
Yeah, your screen is visible, but please expand it, please.
Yeah.
It's now.
So good evening, everyone. So today I'm presenting uric acid induced hyperuricemia in high fructose fed and oxonate treated rats, a novel chronic murine model of hyperuricemia. So as we all know, hyperuricemia is a metabolic disorder characterized by serum urate level greater than 6.8 mg per deciliter reflects supersaturation of urate in extravascular fluids. Purpose and rationale. Administration of fructose for prolonged period leads to sustained hyperuricemia along with metabolic complications such as hyperlipidemia, insulin resistance, glucose intolerance, and renal dysfunction. Its metabolism finally leads to formation of uric acid and causes hyperuricemia. Potassium oxonate is the potassium salt of oxonic acid which inhibit uricase enzyme which is responsible for conversion of uric acid into more soluble product allantoin. This leads to accumulation of uric acid in rodents and induce human resemblance clinical condition of hyperuricemia. Furthermore, exogenous administration of uric acid in fructose fed and oxonate treated rats cause elevation of serum uric acid. Chronic treatment of about these agents together leads to increase in serum urate level and simulate clinical condition of hyperuricemia and associated complications. So here's the hypothesis. So here we were administered three main things. First is high fructose diet. So why high fructose diet?
So a worldwide epidemic shows that fructose intake is associated with metabolic syndrome, which is currently defined as having at least 3 to 5 characteristic signs as an impaired fasting glucose, hypertriglyceridemia, low HDL cholesterol, BP elevation, etc. Then second is potassium oxonate. So why potassium oxonate? So uric acid is converted into allantoin by the enzyme uricase, which is more soluble product than uric acid and easily eliminated by renal excretion in rodents. But in human and certain other primate species too, lack of expression of uricase enzyme. Therefore, serum uric acid level are several folds higher in normal humans than in rodents. To make human resemblance hyperuricemic model, uricase enzyme should be inhibited in animal rodents. So we can block the effect of uricase by potassium oxonate. And third is uric acid by intraperitoneal route. So uric acid level after exogenous uric acid administration are generally higher than endogenous uric acid level. So it will cause hyperuricemia. This is the model. So experiment design. The animal were divided into 6 groups. Group 1 and 2 received normal pellet diet and 60% high fructose diet respectively for entire study period. Another group of animal, group 3, fed with normal pellet diet was injected with potassium oxonate. The dose is 250 mg per kg IP for 7 consecutive days. And the group 4, 5, and 6 fed with 40% of high fructose diet were injected with potassium oxonate. Dose is 25 mg per kg intraperitoneally daily for 56 days. Once clinical condition of hyperuricemia was achieved, approximately 2 to 3.5 mg per deciliter on the day of 28. So from the next day, on next day, day 29, different doses of uric acid 1, 5 or 25 mg per kg by oral route were injected to 40% high fructose diet fed and potassium oxonate treated rats for another 28 days. So various parameters such as serum uric acid, urine uric acid, serum nitrate, xanthine oxidase activity, and uricase activity were measured at interval of 14 days till the suitable hyperuricemia and its complications were achieved. The effect of high fructose diet, potassium oxonate, and uric acid was evaluated by calculating significant changes in following parameters. So first is percentage change in body weight. So graph 1 indicates that on the beginning of the study, the body weight of the animals were in a range of 240 to 270 grams. The body weight of animals was to be increased during the experimental period, but it did not give statistical difference. Feeding high fructose diet 40% and 60% throughout work did not give significant result for percentage change in body weight, while rats treated with potassium oxonate 250 mg per kg by IP route for 7 days showed consistent decreasing in body weight. Groups of animals fed with 40% of high fructose diet and potassium oxonate and additionally treated with different doses of uric acid showed rise body weight, but it was not significant and gradual on day 42, 56, and 84. Then second graph represents serum uric acid. So at the beginning of study, serum uric acid level was in a range of 0.6 to 1.2 mg per deciliter in all groups. The animal fed with 60% high fructose diet showed significant increase in serum uric acid level as compared to those fed with 40% high fructose diet on day 14th in group 4, 5, and 6. However, 40% high fructose fed rats received uric acid on different doses, which is 1, 5, and 25, showed significant increase in uric acid level on the day 42 and 56 compared to those fed with 60% high fructose diet only. The rise was graduated in all animals. Additionally, received different uric acid level. Moreover, the rise was more significant on day 56 in rats those received uric acid. The third graph, which represent urine uric acid parameter, the urine uric acid level was approximately 1.18 to 1.40 mg per deciliter on day 0. Then 60% high fructose diet fed rats showed gradual decrease in uric acid, which reached to significant decrease in urine uric acid on day 56 as compared to rats fed with normal pellet diet. The normal pellet diet fed rats injected with potassium oxonate showed significant decrease in urine uric acid level as compared to other groups on day 14. However, potassium oxonate treated rats with 40% high fructose diet fed rats, additionally injected with different doses of uric acid 1, 5, and 25 for another 28 days, showed gradual increase in urine uric acid level after day 28. Moreover, the rise was more prominent on day 42 and 56 in rats injected with uric acid as compared to other groups. So the level was achieved approximately to 3.01 mg per deciliter. Here, graph 4 is representing xanthine oxidase activity. So at the starting of study, xanthine oxidase activity was found nearly equal in all groups rats, which is 7 to 10 IU per minute. Feeding of 60% high fructose diet to rats for 56 days significantly increase xanthine oxidase activity as compared to rats fed with normal pellet diet and rats those injected with potassium oxonate. However, the xanthine oxidase activity was more prominent in rats fed with 60% high fructose diet on the day 14, 28, and 42. Administration of different doses of uric acid for another 28 days significantly increased the xanthine oxidase activity as compared to rats injected with potassium oxonate and rats fed with normal pellet diet on day 42. The contraindicated results were observed in rats additionally treated with different doses of uric acid. The doses are 1, 5, and 25 mg per deciliter. The lowest dose of uric acid significantly increased the xanthine oxidase activity as compared to medium and high dose of uric acid on day 56, as well as the activity was observed to be increased, although not significant, as compared to rats fed with 60% high fructose diet. Now, uricase activity, graph 5. On day 0, uricase activity of all groups was in range of 24 to 28 IU per minute. And group 2 and 3, in group 2 and 3, uricase activity were decreased. The fall in uricase activity was more significant with potassium oxonate injected rats on day 14 as compared to rats fed with normal pellet diet only. Activity increased after day 14 in potassium oxonate treated rats indicating decreasing in effect of potassium oxonate after termination of 7 days of treatment of potassium oxonate. Administration of low dose of potassium oxonate and 40% high fructose diet also decreased the uric acid activity. Effect was observed on day 28. No significant difference was observed between group 3, 4, 5, and 6 on day 28. However, administration of different doses of uric acid for another 28 days showed significant decrease in uric acid activity on day 56 as compared to rats fed with 60% high fructose diet and rats injected with potassium oxonate. Moreover, dose-dependent effect of uric acid was not observed. Last graph represent serum nitrate level. On day 0, nitrate level was observed to be in range of 50 to 55 micromolar per liter in all groups. No significant change in nitrate level was observed in rat fed with normal pellet diet, which is group 1. Feeding of 60% high fructose diet significantly decreased in serum nitrate level as compared to rats fed with normal pellet diet on day 14, 28, and 56. Similar result was also observed in rats injected with potassium oxonate. Administration of different doses of uric acid to 40% high fructose fed and potassium oxonate treated rats also decreased the serum nitrate level. The fall was graduated but not significant. However, significant fall was observed on day 56 in rats. Additionally, administration with different doses of uric acid compared to rats fed with 60% high fructose diet and rats injected with potassium oxonate. So, after that, animals from each group were utilized for validation of development of model. They were kept on their respective diet for another 28 days and the administration of potassium oxonate and different doses of uric acid, which are 1, 5, and 25 mg per kg by oral route, were discontinued. The validation was carried out to observe the reversibility or maintenance of the developed hyperuricemia and to assess the complications associated with hyperuricemia. Various validation parameters such as serum uric acid, urine uric acid, xanthine oxidase activity, etc. Were measured on day 0 and 28. The effect of high fructose diet, potassium oxonate, and uric acid was evaluated for validation purpose by calculating significant changes in following parameters. So these graphs are for the resulting parameter for validation of model. So the first graph is indicated serum uric acid. The established model for hyperuricemia was validated by measuring serum uric acid level to check the reversibility or reversibility of clinical condition. Rat fed with 40% high fructose diet showed significant increase in serum uric acid level compared to those fed with normal pellet diet and 60% high fructose diet. The percentage change was highest in 40% high fructose diet fed rats that previously with uric acid. Then second graph which represent urine uric acid. Urine uric acid level in rats fed with 40% high fructose diet was significantly higher as compared to other groups on day 0. Highest level was observed with 40% high fructose diet fed and oxonate treated rats additionally injected with uric acid. After 28 days of respective diets, levels of uric acid in different groups was similar that observed on day 0. suggesting the maintenance of disease condition. Moreover, developed disease model was found to be irreversible in nature. The third is for xanthine oxidase activity. During validation period, xanthine oxidase activity was decreased in rats fed with 40% high fructose diet. However, the fall, although not significant, was observed as compared to day 0 in all groups. And the next graph indicates that uricase activity. Feeding of respective diet for 28 days during model validation phase did not alter uricase activity. On the day 28, activity of uricase in a different group was similar to be found on day 0, indicating the maintenance of model or uricase enzyme saturation. The next graph represents serum nitrate. Discontinuation of potassium oxonate and different doses of uric acid significantly increased the serum nitrate level as compared to day 0 on day 28. The serum nitrate level was also increased in rats fed with normal pellet diet and 60% high fructose diet. However, the rise was not significant on day 28. The next graph represents creatinine level. Hyperuricemia generally leads to renal dysfunction. At the starting of validation phase, serum creatinine level was approximately in a range of 0.7 to 0.9 mg per deciliter. So rats fed with 40% high fructose diet significantly increased the serum creatinine level, which is greater than 1 mg per deciliter as compared to observed on day 0. Although level to serum creatinine was increased significantly, but it does not suggest the renal dysfunction. Then next is indicate the serum triglyceride. High fructose diet rapidly stimulate lipogenesis and increases triglyceride level. Feeding of 40% high fructose diet during model development phase significantly increased triglyceride level as compared to rats fed with normal pellet diet. However, the rise was significant in rats fed with 40% high fructose diet and additionally injected with different doses of uric acid. During validation period, triglyceride level was decreased in all rats except those fed with 40% high fructose diet and previously injected with uric acid. The reason for such contraindication results may be due to some kind of manual error occurred during triglyceride measurement or any physiological changes during study. Then next graph, which is represent serum cholesterol. Overproduction of cholesterol and decreased lipid catabolism were observed with fructose feeding. Similar results were obtained in a study. Rats fed with 40% high fructose diet significantly increased the serum cholesterol levels as compared to day 0. Moreover, total cholesterol was also in rats fed with 60% high fructose diet, but the rise was not significant. This may be due to the effect of previously administered uric acid. And the last graph represents the serum glucose. So during a model establishment study, administration of different doses of uric acid, which is 1, 5 and 25 mg per kg to high fructose fed and oxonate treated rats showed significant increase in fasting serum glucose level as compared to observed in normal pellet diet and 60% high fructose fed rats. However, discontinuation of potassium oxonate and uric acid significantly decreased serum glucose level, and the fall in glucose level was also observed in normal pellet diet and 60% high fructose fed rates, but it was not significant. So here are some references, and after this, we can conclude that chronic administration of uric acid is oxonate treated rate and fructose fed rates showed significant hyperuricemia. So the disease condition got worsened during validation phase and suggested the irreversibility of or developed hyperuricemia model. However, further studies required using different doses of uric acid and combination of potassium oxonate and uric acid treated rates. Thank you.
Thank you, Ms. Lipi.
Okay.
It was a very exhaustive work though. Yes, Dr. Pai.
Yes. Yes, Lipi, thank you. Thank you for the presentation. Lipi, can you tell me why you have estimated that serum nitrate?
Yes, sir.
Yes.
Dr.
Why
Pai
you have
wants
estimated
to know the rationale behind estimating nitrate.
serum nitrate?
Serum nitrate.
Hello.
Lipi,
Sir,
can you hear me?
am I audible?
Yes, can you hear me?
Yes, sir. So serum nitrate level indicates that hyperuricemia induced endothelial dysfunction. So for model important that we have to measure serum nitrate level.
Okay. Thank you, Lipi. All the best.
Thank you, sir.
Okay. Thank you, Dr. Pai. May I now invite Dr. Yadav to examine the candidate?
Yeah, just again, one query from my side also, that it's a very exhaustive work as we have already seen. So Lipi, you could have actually kept the most important results. Like what happens is hearing it like over a period of more than 20 minutes, and then your graphs, most of them seem to be the same. All are almost in black and gray shades. It's not very impactful to show what were the more significant results. Slightly more difficult when you stretch, and in which you're also including data which have not shown results.
Yes.
So I would only suggest is work is good, and it seems you have done a lot of things, but only thing is more concrete things could have been highlighted, and the length of the talk could have been reduced by at least 5 minutes. That's it from my side. Thank you so much.
Sure,
Yeah,
sir.
this is a very important suggestion made by Dr. Yadav, sir. Yeah. Okay. Thank you, Ms. Lipi. I declare that the scientific competition this year, this edition, 5th edition of SNJB PharmaCon comes to a conclusion. There had been 25 presentations, 25 candidates who have presented, successfully defended their presentations. They were examined by, in all, 7 judges from different corners of the country as well as abroad. And I thank them all for their contribution. Now it's time to appreciate the efforts taken by our invited judges, Dr. Sridhar Pai from Manipal and Dr. Kushwant Yadav from Mumbai. They have spared their valuable time, although it is a Sunday, but there shouldn't be any Sunday or any resting time for scientists. I do appreciate their efforts in giving us more than 3 hours of their daily schedule, and their suggestions would be very, very helpful for young scientists to direct their thinking. I may now request my technical team to appreciate these 2 people, these 2 judges. On behalf of SNJB PharmaCon and SNJB family, our team at SSDJ College of Pharmacy appreciate your efforts in judging the competition. And on behalf of our principal also, Dr. C.D. Upasani, I request Dr. Sridhar Pai to accept the certificate.
Thank you. Thank you very much. Thank you.
We appreciate wholeheartedly your contribution to the conference.
Thank you for giving me an opportunity. Thank you.
Yeah, and we will be very much happy to have this association growing.
Sure. Thank you. Thank you, Vivekananda. Thank you.
Thank you.
Thank you.
Thank you, sir. Could you please display the other certificate? Yes, Dr. Kushwant Yadav, we are wholeheartedly thanking you for your kind gesture in sparing more than 3 hours of time. Hello.
Thank you, sir. It's my pleasure. Thank you, one and all. Thank you, Patil, sir, for sending me the invitation. Thank you, Pasni, sir, and thank you, Dr. Vivekanand. It was a pleasure. Thank you so much.
Yes, Dr. C.D. Upasni wants to share something.
No, I just wanted to say thanks to our supporters, Dr. Pai and Dr. Yadav, for sparing their half of the day, that to Sunday for us. It is really a difficult task to sit for 3 hours and evaluate the research and comment them and motivate them with your suggestion. So I am really honored with your presence, and I am really thankful to you, Dr. Kushwant Yadav and Dr. Pai. I am very much thankful to you. Thank you.
Thank you, sir. Thank you very much.
Thank you, sir.
Thank you, Doctor. I may declare that the scientific competition has concluded now, and the results will be declared tomorrow at 2 p.m. in the valedictory function. Over to you, Dr. Atish Mundada.
Thank you, sir. So here we come to the conclusion of day 3. As a coordinator of SNJB PharmaCon 2021, I would like to thank each and every one for being there with us on this Sunday. In the morning, we had a great session delivered by Dr. Srinath, who spoke about the applications of dendrimers in ovarian cancer, and he has elaborated his research, which he has carried out using dendrimers as one of the drug delivery approach for treating ovarian cancer. And after that, we had a marathon lecture delivered by none other than our beloved Dr. Saranjit Singh sir, and many delegates has actually enjoyed that Sunday feast being delivered by our Saranjit Singh sir. In the noon, we had technical presentation. That was the last round of technical presentations which were being delivered by various presenters, and we are really happy to have Dr. Pai and Dr. Kushwant Yadav as an evaluator for today's technical presentation session. In all, we had around 28 or 29 presentations for this technical presentation competition. We received many abstracts, and out of those abstracts, we could select only 30 abstracts according to their criteria and according to the depth of the research which they presented in their abstract. We have accepted 30 abstracts, and those abstracts were presented in last 3 days. The competition was very tough, and results will be generated and will be presented tomorrow. We'll be announcing the winners of this presentation competition in the valedictory function tomorrow. Tomorrow will be the concluding day of SNJB PharmaCon 2021. Tomorrow in the morning session, we will be giving our ears to Dr. Raj Suryanarayanan, who is professor with University of Minnesota, and next session and last technical session will be delivered by Dr. Ravindra Gui from Pune. And in the noon session, we will be going for valedictory function, where esteemed entrepreneur Navlin Khurana will be the chief guest. So this is all from day 3 of SNJB PharmaCon 2021. Thank you each and everyone. Thank you delegates. See you tomorrow morning at 10 a.m. sharp. The links will be shared to you on Telegram group as well as through WhatsApp. Thank you, everyone. Thank you very much.
Thank you. We may sign off now.
मुला मुलींसाठी स्वतंत्र वसतिगृह असलेली
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्रातल्या नाशिक जिल्ह्यातलं चांदवड इथलं SNJB कॉलेज
MBA,
इंजिनिअरिंग,
फार्मसी,
डिप्लोमा इन फार्मसी,
होमिओपॅथी मेडिकल,
पॉलिटेक्निक,
ड्रेस डिझायनिंग अँड मॅन्युफॅक्चरिंग,
इंटिरियर डिझाइन अँड डेकोरेशन,
आर्ट्स, कॉमर्स अँड सायन्स,
B.Ed,
व्होकेशनल कोर्सेस या सर्व शिक्षण शाखांचं सर्वोत्तम शिक्षण देणारी
मुला मुलींसाठी स्वतंत्र वसतिगृह असलेली
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्रातल्या नाशिक जिल्ह्यातलं चांदवड इथलं SNJB कॉलेज
MBA,
इंजिनिअरिंग,
फार्मसी,
डिप्लोमा इन फार्मसी,
होमिओपॅथी मेडिकल,
पॉलिटेक्निक,
ड्रेस डिझायनिंग अँड मॅन्युफॅक्चरिंग,
इंटिरियर डिझाइन अँड डेकोरेशन,
आर्ट्स, कॉमर्स अँड सायन्स,
B.Ed,
व्होकेशनल कोर्सेस या सर्व शिक्षण शाखांचं सर्वोत्तम शिक्षण देणारी
मुला मुलींसाठी स्वतंत्र वसतिगृह असलेली
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्रातल्या नाशिक जिल्ह्यातलं चांदवड इथलं SNJB कॉलेज
MBA,
इंजिनिअरिंग,
फार्मसी,
डिप्लोमा इन फार्मसी,
होमिओपॅथी मेडिकल,
पॉलिटेक्निक,
ड्रेस डिझायनिंग अँड मॅन्युफॅक्चरिंग,
इंटिरियर डिझाइन अँड डेकोरेशन,
आर्ट्स, कॉमर्स अँड सायन्स,
B.Ed,
व्होकेशनल कोर्सेस या सर्व शिक्षण शाखांचं सर्वोत्तम शिक्षण देणारी
मुला मुलींसाठी स्वतंत्र वसतिगृह असलेली
सर्वोत्तम शिक्षण संस्था
SNJB कॉलेज चांदवड. महाराष्ट्रातल्या नाशिक जिल्ह्यात